Good morning. I'm Lorraine Steele, and I'm the Chief Executive Officer of the American Liver Foundation. Since joining ALF in January of this year, I've been looking forward to participating in the 30th anniversary of the annual Irwin M. Arias Symposium, Bridging Basic Science and Liver Disease. More than 45 years ago, a small group of physicians from AATD-LD recognized the devastating impact of liver disease on America's men, women, and children. Together, they created the American Liver Foundation, the nation's leading nonprofit voluntary health agency promoting liver health and disease prevention. Our mission is to promote education, advocacy, support services, and research for the prevention, treatment, and cure of liver disease, and we remain steadfast in our mission today. Partnering with Dr. Arias and his team, as well as the amazing researchers speaking here today, is a vital opportunity for our ongoing liver education. Through your ongoing research and this annual event, we can bridge the advances of basic science with better diagnosis, treatment, and prevention of liver disease for our patients and caregivers. Okay, we now have a Zoom housekeeping slide. This program is being delivered on the Zoom webinar platform, and it will be recorded. During today's presentation, if you have a question, please hover your mouse over the bottom of your screen and click on Q&A. Then simply type your question into the Q&A box. Again, please note that this program is being recorded. It will be on ALF's YouTube channel for future viewing, so spread the word. Okay, now my great honor. The American Liver Foundation had the pleasure of honoring Dr. Arias at our recent 45th anniversary Legacy Gala in New York last month. I am equally honored to introduce the special and passionate supporter of the American Liver Foundation again today. Dr. Arias is a senior scientist emeritus at the National Institutes of Health, professor emeritus, Department of Medicine in Gastroenterology and Liver Disease at the Albert Einstein College of Medicine, professor of physiology and of medicine emeritus at Tufts University School of Medicine in Boston. He has been the recipient of many national and international honors, which he has received in a long and continuing career in liver-related science, medicine, and educational programming. It was in the spirit of fostering community, advancing medicine for liver patients, and highlighting the power of basic science that the ALF voted to hold an annual symposium named for Dr. Arias over 30 years ago. Each year, this program honors Dr. Arias, a founder of the American Liver Foundation, who was a longtime board member of both the ALF New England Division and the ALF National Board of Directors. The goal of this annual program, like Dr. Arias, is designed to bridge the remarkable advances in basic biology and engineering with the understanding of liver diseases and their treatment. Dr. Arias has contributed substantially to the mission of the American Liver Foundation, particularly by supporting young PhD and physician scientists interested in the pursuit of liver disease. In the early 90s, around the same time that the symposium was created, the American Liver Foundation established the Irwin M. Arias MD Postdoctoral Research Fellowship Award. Since 2005, 15 postdoctoral awards have been given from this endowment. And I must just share, Dr. Arias is always the researcher. In our pre-conference call, he was grilling those on the call about basic science and the latest and greatest. It was already an education for me. And Dr. Arias, we appreciate your continued creative mind and your pursuit of science and knowledge. So with that, it is my great honor to introduce to you the distinguished and much honored and much respected Dr. Irwin Arias.
Wow. Thank you, Lorraine. It certainly is a pleasure and exciting to see the remarkable program that Sangeeta, Wolfram, and others have put together for this 30th annual symposium. It was 30 years ago, at the time of a significant birthday of mine, that the New England chapter board wanted to celebrate and suggested having a party or a dinner. And I thought that the time was right to have a symposium that really reached deeply into basic biological and engineering sciences as the sort of the fountainhead leading ultimately to translation into better care for patients with liver disease. And that's how the program started. And it has been sustained, amazingly enough, to now the 30th year. If you look at the list of our participants from around the world, there are over 300. You will easily recognize there are at least 12 Nobel laureates and a huge array of outstanding scientists in almost all areas of related biology and medicine, and individuals who are outstanding clinical investigators as well. I trust that this list will be made available, if it isn't already, on our website, because I think just reading the names will give you an idea of the extraordinary opportunity that we have had through the years to hear outstanding individuals speak about their work and then to benefit from it. And there has been great benefit from it, because apparently we're doing the right thing, because the attendance at these symposia have been substantial through the years. Now, this is the first year that, due to COVID-19, we are truly international in our reach. There always were many individuals who participated in speaking in the course, in the symposium, but they were transported to Boston for the purpose. But now, due to the wonders of electronics, we are truly an international program today, with speakers from Europe, the Middle East, Asia, as well as the United States, and an audience which is international as well. Although there are great differences between a virtual conference and an on-site one, and particularly regarding the ability of young investigators to directly communicate with the speakers, somehow electronics permits us also to facilitate that kind of an interaction. So I would conclude by thanking everyone, particularly those who worked so hard to put this outstanding program together. And to thank Heather Fleming for her amazing persistence in seeing that everything will work out well. And to all those who took part at various levels in producing today's program. And most importantly, to thank all of you who are going to join with me in having an incredibly exciting day dealing with the science of liver structure, function, and disease. We are very, very grateful, and primarily to the American Liver Foundation, which through the years has supported this venture. To my knowledge, this is a unique event. I don't know of any other such symposium directed toward that type of a goal, to bridge advances in biological and engineering sciences with human health. So thank you all. We can all sit back and enjoy and learn from a remarkable group of speakers. Thank you.
Thank you, Win, and good morning to everyone. I'm Wolfram Goessling, and together with Sangeeta, Bhatia, and Heather Fleming, we have prepared what I think is going to be a truly exciting day today. I'm a liver doctor and a liver cancer doctor at Mass General Hospital in Boston, where I directed a gastroenterology division. I'm also the director of the Harvard-MIT Division of Health Sciences and Technology at Harvard Medical School. And I wanted to really express, as Win has already said, my excitement about the fact that we have such a large and truly international audience and speaker panel. When we, Sangeeta, Heather, and I started to organize this, this symposium several years ago in person at the Broad, our goal was really to both bring the local community of liver researchers and liver enthusiasts together, but always with an eye towards connecting to our community at large. And I think as much as COVID-19 has really pushed us all to the limits over the last 20 months, it has offered new opportunities. And I think this is really one where we take advantage of us being forming a community digitally across the globe. And that's really exciting to see and to have you all here and to share the science that we're going to hear about. Today, we will have 4 outstanding science sessions that will each be consisting of 30-minute live talks, as well as micro talk sessions picked from the abstract that we have received. And in the lunch break, we have a special treat. We will have a digital fireside chat with Dr. Arias, where he can reflect more on not just the 30 years of the history of the symposium, but really on what the bridges that he and others built in science in and around the liver have been able to achieve. I also want to say thank you to Lorraine and the entire staff at the American Liver Foundation. This has really been a great partnership, and I think it allows us as liver researchers to see the value and connect to what really the goal of all of our work is, and that is to affect and improve patients' lives. And we feel that connection through our work with the ALF particularly. And with that, I hand it over to my colleague and friend and co-organizer, Sangeeta, Bhatia.
Thanks, Wolfram. Hi, everybody. I'm Sangeeta Bhatia. I'm a liver bioengineer. My lab is based at MIT, and together with Wolfram, I'm the co-host today. Some years ago, Wolfram and I both participated in the symposium, and Wynn then passed the baton to us, actually physically at the symposium, to take over as co-organizers, which we very happily have done. Over these few years of co-hosting, we have tried to broaden the reach and the excitement of the liver to also include colleagues from industry. So you'll see some industrial speakers today, along with the basic science researchers and the physician scientists. So we're really excited about the program, which Wolfram described. Just to sort of remind you again, in spite of the fact that we're virtual, there is an opportunity for questions. So you can type those questions in the Q&A box, and the session share will get those to the speaker. We also are excited about cultivating the next generation of liver enthusiasts. So we have some 3-minute micro talks scattered throughout the programs. These are 2 slides only, just snapshots of hot topics. So we're excited to share those. And Wolfram and I will see you back with Wynn at the Fireside Chat. So with that, we're gonna start the day. You'll hear from our sponsors, and then we will have the first session chair kicking us off, Kirsten Sadler-Adepli from NYU Abu Dhabi. So sit back and enjoy the science, and we look forward to our virtual connection.
Thank you, Sangeeta. Good morning, everyone. My name is Sherry Singer, and I am the National Director of Development and Fundraising for the American Liver Foundation. Welcome to all of our global attendees. Throughout the morning, we will be sharing short videos from our amazing corporate sponsors. Without their ongoing support of the ALF mission, programs like the annual Arias Symposium would not be available to us today. At this time, I'd like to introduce you to our first sponsors. They are Intellia and Exact Sciences, who represent our presenting sponsor level. Thank you.
Liver cancer is the 6th most common cause of cancer death in the U.S., and its numbers are rising, on track to make it the 3rd leading cause of cancer death by 2040. But the 5-year survival rate can increase dramatically when liver cancer is caught early and curative treatment options are available. 85% of diagnosed liver cancers are hepatocellular carcinoma, or HCC. Fortunately, routine HCC surveillance improves early-stage detection. Unfortunately, the standard of care for surveillance, ultrasound, with or without an alpha-fetoprotein blood test, has poor and variable sensitivity for early-stage HCC. What's more, scheduling and logistics can make it difficult for patients to comply with surveillance testing. As a result, fewer than 1 in 3 patients eligible for HCC surveillance are compliant with surveillance testing. This contributes to the fact that 2 out of 3 cases of HCC are detected in the late stages. The OncoGuard liver test is an innovative new tool for HCC detection, including early stages. In a clinical validation study among at-risk patients undergoing surveillance, the OncoGuard liver test demonstrated high early-stage sensitivity and high specificity for detection of HCC. The OncoGuard liver test, a simple single blood test that may help more patients by enabling early-stage detection of HCC. OncoGuard liver, surveillance. Simply advanced. Contact your representative for details or visit us at oncoguardliver.com.
Good morning, good afternoon, good evening, everybody. It's really a pleasure to be at this symposium, and I will kick off the first session. My name is Kirsten Sadler, and I am dialing in from my position at NYU in Abu Dhabi. Before I introduce the first speakers, I just want to say what a thrill it is to be on this call with you, Win. I have been at several of these symposia over the years, and I've always found them to be among the best research symposia in the liver field. So I'm really grateful to have a part in this symposium today. So we'll start the session with a talk by Takanori Takebe, who is the Director of Commercial Innovation in the Center for Stem Cell and Organoid Medicine at Cincinnati Children's. He is an Associate Professor and Endowed Chair of Organoid Medicine, also at Cincinnati Children's, as well as a Professor at the Institute of Research at Tokyo Medical and Dental Institute. The topic of his talk will be Organoid Medicine for Precision Hepatology.
All right. Thank you so much, Kirsten, for the kind introduction. So can everybody see my slides okay?
It's all good.
Great, thank you. So it is, extremely honored to be a speaker for this amazing symposium. And I'd like to thank Sangeeta and Wolfram, Heather, and then also Dr. Arias for giving me this, fantastic opportunity. Really, truly honored to give an update on our liver program using the pluripotent stem cells. I also appreciate the American Liver Foundation to make this exciting program possible. So my name is Takebe, working at the Cincinnati Children's Hospital Medical Center, but also affiliated with Tokyo Medical and Dental University. So today I am connecting from Japan. So it's 11 p.m., but I'm really excited to really speak here at this phenomenal symposium. So I was originally trained in the liver transplantation field, but decided to move back to the bench side to pursue the different approach to really focus on generating the liver tissues, which is our most interested organ systems, as many of the speakers and participants are thinking about. So the really, very ambitious goal is to really try to generate this, beautiful, organized liver structures in the dish for the future studies of the human organogenesis and disease, and in the longer term towards the human therapeutic applications. But I think this is obviously a really challenging task, although we have lots of exciting stem cell science forward, including the ESL technologies or iPS technologies. But I am always interested in learning something from the past, which is about the history of organ transplantations. So as many of you know that, organ transplantation surgery is currently really, golden standard therapy for the patients with, fading organs. But I think, until being, able to standardize this, surgical process, I think it's been like almost like 100 years, and 3, Nobel Prize discovery was, made throughout the process of establishing the organ transplantation process, probably towards the regeneration of human organ systems, really requiring almost like the same time window and same levels of, important discoveries to really move forward. I would think, the embryonic stem cell discoveries and induced pluripotent stem cells really, eye-opening discovery, and really opened up the possibility to pursue this, really challenging goal of trying to generate the functional human organ systems. But right now, today, we are still in the process of understanding the mechanisms of how the organs are formed in a normal process. And in knowing that process, I think we need to translate the molecular or cellular principles in in vitro principle for generating the organs of interest. And obviously, I think, directed differentiation technologies or direct reprogramming technologies, or most recently organoid technologies, is really, open up the possibility to pursue, this exciting challenge moving forward. But still, I think we need to have real, therapeutic protocols or methods to really make this approach possible in a patient setting. So to do that, I think we still need to have to do lots of research before using it as a clinical applications. So when I started my career right after the medical doctor programs completed, I performed a very simple experiments to get some sort of insights to this goal, which is listed in these figures. So what I have done at my early career stage is very simple experiments of transplantations. So what I'm going to explain here is to show the fetal liver transplantation experiments and into the adult mouse. So original fetal liver is actually comes from the E30.5 days of the mouse and labeled by red color. And after transplantation into the adult liver, so adult mouse recipients, I have found that very exciting, development of the fetal liver into the adult liver-like tissues in the native locations. So when you look at the simple internal structures by the confocal live imaging analysis, you can nicely acknowledge the sinusoidal vessel formation as well as beautiful hepatic cord-like structures. And at the edge of the tissues, there are some functional vascular anastomosis connected to the recipient circulations. Plus, biliary drainage system is also connected. So looking at this, very exciting preliminary data sets, we decided to pursue this challenge of how to dilate the fetal liver tissues, or at this stage, what we call liver butt organogenesis in a dish using the human stem cell culture system. So that was the initial goal of our approach, trying to use the human stem cell culture systems. So liver butt tissues actually establishing a very early process of the organogenesis around E10.5 embryos. So the process is really, well studied by the, pioneering developmental biologists in the past, primarily using the animal model systems. So there are, lots of critical events, but to simplify this process, I would explain today dividing two major events of the liver butt formation process to be modeled in the stem cell culture. So initial step or initial process of making liver is actually the fate patterning process that is taking place around E8.5 embryos. So at this stage, foregut and midgut tissues or hindgut tissues is labeled by SOX2 and CDX2, and liver region will be specified as a PROX1 positive regions right after 1 day of the embryonic days. And at the boundary between the SOX2 CDX2 foregut midgut regions, we see the PROX1 specification at the border of these two tissues, primitive gut structures, which is the liver primordium or liver progenitors. And right after subsequently, this liver specified endoderm is delaminating from the gut to form a more, bigger tissue mass called the liver bud tissues. So essentially what we want to try to achieve is really, understand the process of these type of, critical events and translating these events in the culture principle. So today I'm going to highlight in my first part, looking at the budding or some inductive cue for this liver formation process in culture. So first questions is, there are a number of pioneering studies, including Gonzalez's work and other pioneers, identifying the vascular population or endothelial progenitor population is really key for inducing the liver bud formation process in animal model studies. So what we set out as an initial goal is to develop the liver-specific endothelial progenitors by recapitulating the normal embryonic process in a culture dish. So to tackle these questions, Noli, really talented postdoc in the laboratory, decided to look at the single cell RNA sequencing data sets published by other groups in identifying the critical signal cascades inducing the liver-specific vascular progenitor specifications. So those who are not familiar, panel A is to show you the process of liver vascular network formation at the early embryonic stage. And in the middle part, this is the liver primordium lesions, and right adjacent to the liver primordium, vitamin veins connected from the sinus spinosus region is existing, and then right after that, liver specifications, they are migrating towards the liver regions to populate the sinusoidal networks are formed. So knowing that, anatomy representations, we next looking at the single stranded sequencing analysis and deconstructing the different cell states and analyzing what would be the critical cellular signatures or signaling pathways inducing the progenitor state to the liver-specific endothelial cell state. So to make a long story short, we found that relatively low activation of VEGF-A pathways as well as oncostatinum signaling pathway is really driving the process of the maturing the liver-specific endothelial progenitor populations. So this is from the human fetal samples. So we next decided to recapitulate the process in the iPS or ES cell culture of humans. So this is the 20 days of the culture protocols. At the middle stage, this is really published protocols. But for after day 6 or day 8, we have optimized the culture ingredients to really specify into the liver-specific sinusoidal endothelial populations to be derived from the iPS cells. So in our previous literatures in 2013 or 2017, we've already shown that vascular population can be formed in our liver organ culture. But at that time, we are aware that these vascular progenitor population is predominantly arterial cell populations, but lacking the sinusoidal characters or portal vein characters. So to support this, this previous, published protocols is giving rise to almost like homogeneous CD34 positive or CD43 mid positive populations, but lacking a CD32, for example. And this is really important marker for the liver vasculatures. But after optimizing the culture protocols with the low VEGF as well as OSM conditions, we found that at the end, 95% of the CD32 and CD54 double positive populations induced from the human iPS cells. And looking at the immunostaining signatures on the panel B, AEC is a previously published protocols expressing CD31, sort of pan-endothelial markers. But our revised protocols labeled as IV-HEC as well as ISEC populations. They are not just expressing CD31, but also CD32B double positivity detected in both of the populations. And on the bottom panel, this is also looking at the factor VIII, which is synthesized by the sinusoidal endothelial cells. So in our middle progenitor also started expressing the factor VIII, but also after complete maturations, more stronger factor VIII synthesis is confirmed in ISEC populations, which is very lacking in arterial cell culture protocols. But we do see in a primary ALF cell culture. In looking at the panel C, this is the EM analysis in looking at the surface of the 2D culture of the IV-HEC populations. And as you can acknowledge here, there are small, tiny holes on top of the surface of these endothelial cells, which is really similar to the primary LSEC EM analysis, but not really existing in arterial cell culture. So taken together, our modified culture protocols likely inducing the liver vascular progenitor populations, which has the capacity to make sinusoidal endothelial cell populations. So next, we wondered how we can, coordinate the progression of the liver organ culture progressions. So to do this, we have labeled the two distinct progenitor by different color. Green is hepatic populations, and red color is the sinusoidal progenitor populations. And over time, as you can acknowledge here, there are exciting self-organization findings detected by confocal live imaging analysis, forming a beautiful sprouted vascular network formed. So in these culture conditions, we have several tweaks to make this happen. So to this purpose, we have invented the protocols termed Imali culture, which is representing inverted multi-layered air-liquid interface culture, which is a little complicated. So what we do is applying the transwell filter to apply high level of oxygenation access to the tissue culture. But also we have placed not just arterial cell population, but also sinusoidal progenitor populations layered on top of the liver progenitor or mesenchyme arterial cell mixture. And with this geometry, we are able to maintain these a couple of different progenitor populations self-organized over time. And looking at the panel B, this is the hormone staining of visualizing the sinusoidal markers. And this is like 5 millimeter tissue in size, and entire vascular networks is CD32B positive. And at the periphery, CD31 single positive population, likely portal veins population is also existing. And what we are most excited about is having that, sinusoidal networks. We have witnessed more improved engraftment as well as maturation of the hepatocyte populations labeled by red color ASGL1s, comparing with the previous one. There are, populated hepatocytic populations with the CD32B positive sinusoidal networks. And in support of this staining patterns, looking at the hepatic serum protein productions like albumins or complement protein secretions in our revised culture, with the Mali conditions labeled by red bar, as you can acknowledge here. All of the hepatic protein is also highly upregulated as a consequence of these sinusoidal populations. So we next wondered how sinusoidal cells is helping the hepatocyte to mature. So to tackle these questions, we have analyzed a single cell RNA sequencing signatures, complicated culture conditions, and identifying Wnt2 signal is really provided by the sinusoidal progenitor populations, whereas the receptor components is expressed by the hepatic progenitor populations. So we hypothesized maybe from this Wnt2 supplied from these sinusoidal progenitors might be helping hepatoblast population to mature. So to support this hypothesis, we have specifically knocking down the Wnt2 in the sinusoidal progenitor population and cultured them in a way we have done in the previous slides. And as expected, hepatic population is extremely diminished in the SI-based Wnt2 knockdown organoid culture. And not shown in here, but also we have confirmed the hepatic protein production is really impaired in the presence of Wnt2 knockdown endothelial culture conditions. So taken together, multicellular communication is really crucial for maintaining the hepatocyte phase on the right track in the normal organoid culture conditions. So as a final part of the functional characterizations, we have also tested the secreted coagulation proteins particularly from the sinusoidal vessel is functional or not, tested in a factor VIII knockout animal model. So factor VIII is responsible for hemophilia phenotype, and if you knock out the factor VIII, there are continuous breeding phenotype is confirmed. And the current standard of therapy is human recombinant factor VIII, and this treatment is really preventing the breeding process. But to a greater extent, our organoculture-derived proteins collected from the culture supernatant is also have an active capacity to inhibit the breeding phenotype on the right. So this figures is actually confirming the functional coagulation activities provided by the organoid derived proteins. So C and D is real transplantation experiments into the liver, and like my initial mouse experiments, fetal liver experiments, our liver organoid transplantation is really successfully engrafting in the native liver locations with predominant vascular networks. And with these conditions, we can also reverse the factor VIII defective phenotype as shown in the AATD data sets here. So this is a 5 months after transplantations, and in the normal conditions, AATD is very short. But if you knock out the factor VIII, the AATD time is significantly increased, and this can be reversed by the recombinant human factor VIII. But in our transplanted model, almost like comparable level of improvements is maintained even after the 5 months. So taken together, our organoid culture with the sinusoidal networks has the capacity to produce the functional coagulation pathways. Okay, so next part of my talk is really focusing back onto the patterning process, because we know that to maintain the functional capacity of the liver, we need to reconstruct the vital connection to the neighboring organ systems, which is including the biliary systems as well as duodenum. So the next question we have in mind is not just making liver, but also we need to connect with the multi-organ components, including biliary systems. So to tackle this challenge, we again looking back the what's going on in the process of making a pattern of this, multicellular structures. So to really simplify the process of early patterning events, this illustration is providing you nuances of how the liver, biolesions and pancreatic region is separated out from the primitive gut structures. So liver, biliary, pancreatic tissues is actually specified at the posterior or ventral foregut regions. But always the geometry of this specification is happening at the border of the foregut and midgut or CDX positive populations. And in our hypothesis at the time was not just making liver, biliary, pancreas separately and trying to connect, but we rather prefer to make foregut and midgut boundary culture to try to intrinsically stimulate the patterning events in the culture process. So that was the initial hypothesis. So we actually do this by in vitro stem cell culture-based approach. So Hilo and Ken really contributed to these efforts. So starting from human iPS culture, we transferred into the 2 different wells, continuing over 5 to 6 days to make an anterior gut endoderm and posterior gut endoderm population separately. Then we make our sort of tissue, small tissue culture mass and combining them in the same well plates to try to apply the boundary stimulation in culture, and then transferred into the air liquid interface culture, providing a lot of nutrients and oxygen access. But what we see is at the boundary between the foregut and midgut tissues, we see the reproducible specification of liver pancreas progenitors. And over time, the segregation or separation of different organ domains in the right locations and right time. So this system is very helpful model to study this patterning events in our culture protocols. So this is actual imaging data sets of the reporter-based visualization of the liver biliary regions by Plox1 reporter systems. And this is the boundary organoid culture. And over time, you can see the red color label liver domains is really invaginating to form a bigger tissue mass. And day 26 through day 38, you can nicely acknowledge the liver region is really connected to single tubular structures, which is the main biliary systems. And lower color lesion is probably, ventral pancreas populations. And no Prox1 positive lesions is actually the gut or duodenal structures. And to support this, we have stained the different organ domain marker systems and on the right, and you can nicely distinguish just by eye, morphology is quite different between, for example, the pancreas and liver. And these tissues can be sectioned to analyze the connections of the different organ domains. So this is a macro view, which is around 5 millimeter in size again. And on top of that, liver is located, but right adjacent to the liver domains, biliary to ventral pancreas population is existing or connecting. And when you zoom up the liver to biliary regions, you can nicely confirm the AFP positive hepatoblast population is connected to EPCAM positive biliary regions. And same for pancreas as well. This is purely ventral pancreas populations, but tip and trunk progenitors is really separated out or segregated, as we see in the normal in vivo organogenesis. But we wondered how this, completely synthetic model is reflecting the actual in vivo phenomenon. So to test out, we decided to try to recapitulate actual human disease or animal disease by genetic loss of function experiments, a very simple approach. So to do this, we have knocked out the HES1 genes, because if you mutate the HES1, it is already reported that, biliary progenitor is converted to the pancreas progenitors, so predominated by pancreas tissues. So to test if we can model that, that was already found in an animal model. So red and green is each labeling the biliary to pancreas. And if you knock out the HES1, you can nicely see the pancreas dominance in a knockout model system. So all taken together, we think that, this multi-organ specification is somehow representing the normal organogenesis. But, still there are lots of limitations in doing making a real liver bud structures. But even with these current formats, we can nicely, confirm the connections to the biliary system by, for example, providing the fluorescent bioacid-based assays. So you can nicely see the liver accumulation at the beginnings and secreted out to the main biliary trees, as you can see here. So based on these sort of simple assay system, we can potentially evaluate the narrowing of the biliary system, for example, by the primary sclerosing cholangitis or biliary atresia, which is still really difficult disease to cure. So we are collaborating with a number of clinician scientists to get the human iPS cells from these patient population in trying to model these disease. And also towards the real liver bud formation, there are still some missing cues, including, for example, the quality of mesenchymes or septum mesenchyme populations. And then also, fetal liver is really supported by hematopoietic populations. So we are currently working on, continuing on making more complicated liver bud organogenesis by supplementing these additional cues. And also, obviously, we probably need exciting transplantation process or surgical process. So to do this, we are collaborating with bioengineers and surgeons to try to help out this, liver transplantation process to go around. But as a couple of final slides, I would like to give a little bit more important messages for the people who are interested in using organoid-based systems. So there are a number of different levels of complexity we can potentially, approach by organoid-based or 3D culture model system. But I think not always, complicated one is the best one, because I think, the more complicated one, having multiple different organ system is a little bit irreproducible or scalable. So depending on the final context of use or intended use, maybe I think the selection of the complicated organ model system would be different. So as a biologist, we are always interested in making more complicated one, but in a real patient application, I think depending on the application, we should simplify the model for the reproducibility and scalability. So I would highlight two applications we are currently in progress. For example, we are recently working on the drug-induced liver injury model, which is a side effects of the pharmacological treatments. So to really predict the drug-induced liver damage, we are currently scaling up the process and establishing the 384-well-based simple screening system to particularly predicting the cholestasis induced by the drugs. So to this model, we are actually using the very simple hepatocyte organoid-based model system lacking other lineages. Whereas on the right, this is more of the precision medicine approach. So we are working on making liver organoids with the mesenchyme, some macrophage population together with the hepatocytes, and testing out the population level donor screening analysis, comparing with the genetic signatures and knowing the potential genomic architectures, defining the risks of developing drug-induced liver injuries. So in these papers, we've described the polygenic risk score, aggregating number of SNP alleles, is really predicting the potential toxicities in the later life when treated with some of the drugs for the cholestatic toxicities. So this is the final slides. So we are currently working on the stem cell on the organoid model system, and this opened up the possibility for the human biology studies. But also we are excited to foresee the future application of this system for the precision medicine, drug development, and ultimately organ transplantation approach. And to do this, I think we need to, really thinking about the question of how simple is complex enough to really test your questions. So that is the key message as an organobiologist. With that, I would like to thank all my colleagues and happy to take questions. Thank you so much.
Dr. Takebe, that was a beautiful talk. A bunch of the questions have been coming in, and I'll just read them out to you, and then I'll let you answer them one by one. So the first question is from Valerie Guan-Evans. Hi, Valerie. She says, amazing work, Taka. How many HLBO organoids do you need to transplant in the liver directly to rescue coagulation in the F8 knockout mouse? And technically, how deep can you transplant those organoids in the liver?
Right. So I think we are transplanting like 10 to the 6 or close to million cells included in HLBO. Which is like 1 or 2 HLBO culture. So probably of the total cell number perspective, I think for the sinusoidal endothelial cells, that is containing like hundred thousands of cells existing in transplanted mouse to rescue the phenotype. So how deep can you transplant? Well, in our model, we are just transplanting on the surface of the tissues. So we do not implant in a really deep locations of the liver, but I think we can certainly test out. But in a factor VIII knockout mouse model, I think it's a little bit challenging because they are breeding significantly. So in a normal, just immunodeficiency mouse, maybe we can go into deeper to transplant the organoid tissues.
Thank you for that. Another question posed by Isaac Oderberg says, very exciting work. Does the addition of vasculature to your hepatic organoids create any kind of central portal zonation in hepatocytes?
Yeah, that's really exciting questions. Actually, we are currently looking at the single cell RNA sequencing signatures, not really completed yet, but there are periportal and pericentral hepatocyte population close to the primary periportal and pericentral population. So I'm pretty sure there are some, cues for inducing more zone-specific type of hepatocytic populations derived from the human iPS cells by adding the sinusoidal components. But we are still trying to understand why we are seeing this.
That's really cool. So we have, I think, time for one or two more questions. This is a question from Luba, and she asks, does pancreatic bud in multi-organ culture have endocrine cells?
Yes, but we do not really force the differentiation of endocrine lineages, but we just culture them and let them, survive in like 2 months or 3 months, and then when we measured insulin or C-peptides, we actually see the secretory functions in our multi-organ based model system. So there are some endocrine components induced, but we do not supplementing any of the endocrine maturation media in our multi-organ culture. So we do not have any specific answer to that. But I think at least in the spontaneous level, there are some endocrine components existing in our organoid culture.
That's really exciting. And then we have one more question, and then we'll move on to the next speaker. So this is from Meri, Huch, and she says, very nice work, Taka. From the first part of the talk regarding the LSEC network and the interaction with the hepatocytes from the liver bud, do you see the architecture of hepatocytes sandwiched between LSEC cells?
Yeah, it seems like we do see, but this is very dense tissue structure. So without, for example, tissue clearing, I think we cannot really conclude the real geometry of the hepatocyte sinusoidal contacts. But I think this is, quite exciting experiments we need to work on. So we are currently working on to make optimized tissue clearing methods to really, analyze the fine spatial orientation of these two distinct progeny populations, but they are touching each other.
So that's really exciting, and hopefully we'll hear more about these developments at a future Arias Symposium. So I'd like to thank Dr. Takebe and move on to our next speaker of the session. This is Maria Mota. Dr. Mota is the executive director of the Instituto de Medicina Molecular in Lisbon and is the leader of the research group Biology and Physiology of Malaria. And she's going to talk to us about a really interesting and important topic of hepatocyte as a household for Plasmodium parasites. Maria?
Thank you very much. I'm starting now I will share now my screen. First of all, let me just put on presentation mode. First of all, I want to thank all the organizers for inviting me to be here to be able to present our data. It's going to be very different. The liver for us is really the household of the parasite that we love very much, that is Plasmodium, so it's the malaria parasite. So I want to thank really all from Goessling, Sangeeta, Bhatia, Heather Fleming to really, supporting work that is, of course, the liver is very important for us, but, it's not that we are not really like experts on the liver, okay? I'm talking today from Lisbon, Portugal, a beautiful day outside. It's just afternoon, and thank you again for listening to me. So basically, as I was telling you, I'm going to talk to you about the liver parasite, the Plasmodium parasite, that is the parasite that causes malaria. And malaria only occurs when this parasite reaches the blood and infects red blood cells in our blood. But prior to this parasite needs to be transmitted by a mosquito and obligatory needs to go through the liver and multiplies inside hepatocytes. What is very interesting to us is that, we are very much interested to understand why do people die of malaria, but really the major question that we have been having in the lab for the past 20 years is, why are hepatocytes so special? And they must be really special. In fact, we know that the mosquito, when transmits parasites, some of these parasites are also being, housed in the skin, and some of them can never leave the skin. But in fact, in the skin cells, they can start replication, but they are never able to do what they do here inside of the hepatocytes, that in each of hepatocytes, each parasite replicates into 20,000, 30,000, sometimes even 50,000 new parasites. And here in this image, taken by one of the students in the lab, Aparajita Lari, basically what she did, it was cross-section, so you have a Z-stack, and you see, the liver structure that you all know much better than me. Then you have these parasites here, and then another one appearing here that replicates inside one single hepatocyte. And really this replication is amazing, because really the size that really rich needs to, the hepatocyte needs to increase what we calculated between 10 to 14 times the volume of a normal hepatocyte. Here, basically what you see is exactly what you have seen before. Of course, this has been already computerized, but every single nuclei of every single cell in the liver has been colored, and as well as every single nuclei inside of the parasite. And so really, you need, I think it's easy to appreciate that the parasite must have something from these cells that allows the parasite to do this. And in fact, for the last 20 years, we have been having this question, but we have not been able yet to answer it. Not that we have been stopped, because when we started, really nothing was known about this stage. A lot of people were working on this stage, but more from the perspective of a vaccine and the immune response that this stage could elicit. But in fact, nobody knew exactly what was going on and what was necessary from the hepatocyte, from the liver itself, and what type of interactions they exist. And so throughout the years, we have been publishing and really, telling a little bit of the story, and taking a little bit of what I call pieces of the puzzle that we are trying to put together, and try to see if we understand what exactly makes this environment so special that allows the parasite in this type of replication. So what I will do today is to tell you three parts of the story, okay, that basically is independent stories, but I divided in three parts that really all tend to answer this big question: why and how are hepatocytes so special for the liver stage malaria parasite? And one aspect that we have been really thinking about in the past years is about this membrane that separates the parasite here in red from the hepatocyte. And basically this membrane we call the parasitophorous vacuum membrane initially is formed because of the invagination of the plasma membrane of the hepatocyte when the parasite is invading. But very quickly, the parasite really modifies this membrane. And so this membrane, for example, here is stained in green because we have an antibody against the parasite marker that specifically the parasite immediately puts away and really, markers this membrane. This membrane has been written in many different reviews that must really play a very important role. It's really the interface between the parasite and the host cell, and probably it's important to feed the parasite, probably to protect the parasite in many aspects. But the truth is that until very recently, we didn't have any function assigned for any of the proteins that really that are present in this membrane. And I'm going to tell you a few stories that we know now, and we have now in the past few years studied about the proteins that reside there. One of the first proteins that we study is this protein from the parasite that again is in the membrane, this membrane, and is called YS3. And we know that is a very important protein, because when we produce parasites that are deficient on this protein, the parasites cannot make it. They really can invade hepatocytes, but very soon, in a few hours, the vast majority of the parasites disappear, and they never replicate in this beautiful, what we call shy zone that has thousands of parasites inside. But postdoc in the lab, Ileana Rial, some years ago, realized that if she had host cells that were deficient in autophagy, the parasite could make it the same way as wild-type parasites. So, in fact, we realized that probably this protein was in the parasitophorous vacuole membrane to somehow impede or, really avoid the autophagy response that seems to be able to eliminate the parasite, or at least this observation was suggesting that. By the same time that we were having this observation, two independent labs, one in fact here in Lisbon, Miguel Seabra's lab that we were collaborating with, and another one in Germany, Volker Essler's lab, basically they were showing that another protein that was in the parasitophorous vacuole was a host protein, but from the host autophagy machinery, that was LC3. And in fact, what you can see here is that both the and LC3, they seem to be more or less in the same place and in the same type of region in this membrane. So we hypothesized that maybe they could interact, and somehow in this way the parasite could really avoid the autophagy machinery and being avoided to be eliminated. So really by in silico studies, structural studies, because we had the structure of both proteins, the parasite YS3 and the host LC3, we could really try to check if they could interact, and in fact, the computer tells us that yes, they seem to be interact, and provided us even some of the amino acids that they thought would be the ones that would be important for this interaction. So through a series of biochemical studies, we proved that indeed some of these amino acids were really important in both sides, in both the host protein and the parasite protein, and that indeed these proteins were really interacting. And by doing that, when the parasite protein interacts with LC3, basically was stopping the autophagy machinery, and we proposed that this would be a way that parasite had to avoid the autophagy machinery and basically avoid elimination by the host cell autophagy. So then we asked, could this really be a drug target? Could this interaction, if we block this interaction, could we really, try to really eliminate the parasite? And then Sonali Setua, postdoc in the lab, joined the lab, and basically, she, again, in an in silico screen for millions of compounds, she really screened for compounds that could really disintegrate, really could impede this interaction between the LC3 and the YS3, more from the point of view of interacting with the parasite protein without really directly blocking the autophagy machinery of the host, but instead just blocking the interaction between these two proteins. And so this was done just in silico, and 15 compounds were selected that they could be really, synthesized and could be used in a screen. And we have performed this screen from this for these 15 compounds, and found that in vitro screen with the parasite infecting hepatoma cells, and basically we realized that we had several compounds that could really, decrease infection, being one, the C4, the one that we selected because had a lowest IC50. When we calculated that the IC50 was around, a little bit below the 200 nanomolar. So we went on and really started check where exactly how this compound was interacting, if it was really, the effect that was having on infection was disrupting this interaction between YS3 and LC3. And we had several studies that show that yes, that indeed C4, this compound, was interfering with plasmodial liver stage infection, really by blocking the autophagy pathway, and on interferes when the host cell had a sufficient autophagy system, but in fact it did that without affecting the host cell autophagy. So basically this C4 is the compound, is a proof of concept that we can block the interaction without blocking the host cell autophagy that will be there working for other systems that cell does, but at the same time, because we block this interaction, the parasite is eliminated by this host cell autophagy. So basically, this is one of the stories that I want to tell you. But of course, when we started to do this, we started to be really curious about other proteins, both from the host side and from the parasite side, that were in the parasitophorus vacuole membrane and were there. So what are really their functions? And I'm just going to give you two more examples. None of this is published, okay? We are in the process now of putting together for publication. But in fact, it seems that the hepatocyte environment seems to be a dangerous environment for the parasite. And a lot of systems, in fact, all cell intrinsic systems seem to be really attempt to eliminate the parasite. And one of them is the cytoskeleton of the whole cell. So we have observed before, and now in more detail, that host cell actin presents an extremely dynamic structure around the parasite. And in many of these circumstances that we have observed, this actin cytoskeleton seems to really, really make a mesh around the parasite, as you see it here, and in many cases it really forces the parasite either to go out of the cell or to eliminate the parasite in ways that we are still trying to understand. But what we realize is that within the parasitophorus vacuole membrane, there is one protein that interacts with host cell actin. And most interestingly, when we had parasites that were deficient for this protein, basically we have many more of this phenomena. Although, in wild-type parasites, we can observe approximately 25% of the parasites that we see in movies that really have these very dynamic actin structures around the parasite. In fact, in parasites that are deficient for this protein, basically we can reach 80% of the parasites that are surrounded by this, and in fact they are eliminated by this actin, as we show here that parasites that die within, many more die without this, with this protein, and almost 95% of the parasites that die because they have this very, extremely dynamic and, in fact, capable of eliminating the parasite from the host cell. So again, seems that proteins here in this membrane are very important to really maintain the parasite in a safe environment and avoid cell intrinsic mechanisms that are capable of eliminating the parasite. But then we had another interesting observation, and this one was done by Aparajita Lari, a PhD student in the lab, and basically she discovered, doing a screen for different host factors that could be at the PVM, she found one of these factors that is called APPL1, and APPL1 is a adapted protein that is localized in a subset of RAF5 positive of positive endosomes, very early endosomes, and this protein is known to be able to either recruit other binding partners or regulate vesicular trafficking. So we were very curious to see exactly what was going on. So in many different experiments, and I'm just going to show you here a slide, but basically by competing with between the RAF5 of the host and RAF5 of the parasite, because Aparajita was able to show that in fact this host protein was really being localized to the parasitophorus vacuole membrane, because the RAF5 of the parasite was working like a kind of a sponge and was competing with host RAF5 for APPL1. And she showed this, and by really doing experiments that compete one side or the other, we could show that, if really we have systems that this protein is not so much anymore into the parasitophorus vacuole membrane, then the parasite does not replicate very well here. So you have less APPL1 in the parasitophorus vacuole membrane, and the parasite replicates much less. On the other hand, if you you compete out and you have a parasite protein that allows even to attract more APPL1 to the parasitophorus vacuole membrane, now what you have is a parasite that develops much better. And so we were wondering exactly what was this protein doing there and why it was so important for the parasite to recruit this host protein to the parasitophagous vacuum membrane. And as I told you, this protein is known to regulate vesicular trafficking, but also endosomal signaling. And we thought about some previous report that we had made some years ago, where we showed that the parasite plasmodium must really, and the plasmodium infection must really down-modulate phosphorylation of AMPK from the host AMPK. If it is not able to do that, the parasite becomes much smaller. And what we think is going on is that parasite is able really to down-modulate the phosphorylation of AMPK because really recruits the APPL1 to the vacuole and doesn't allow to do its work and to activate AMPK. So really connecting these two stories. And as I told you, all these stories are, as we see it now, are still some pieces of the puzzle that we are trying to put together to really understand and fully understand what is going on in this liver stage of infection. But while we were doing this, and a couple of years ago, 3 years ago, in fact, we have been in contact with a collaborator of ours, Shalev Itzkovitz, from the Weizmann Institute, and that we'll listen to him later on. And because we were very much curious about, in the liver stage infection, is really a parasite able to do the same in every single hepatocyte? So we all know that hepatocytes are different. So does it matter or not that the parasite ends up in one or another hepatocyte, or does the parasite select which hepatocyte it invades? And so through single cell RNA sequencing, Shalev and a PhD student in this lab, Mishai, been done beautiful work, and I'm just putting here just one slide. This is also not published work yet, but in fact, they provide an in vivo spatial temporal atlas of both host and parasite transcriptome. But much more interesting for us is that this work shows that indeed the parasite arrives to the liver, it goes through several hepatocytes until it finishes in one of them, but doesn't select. So it seems to be totally random which is the hepatocytes that parasite ends up. But what is not random is that, in fact, it seems that the parasites that end up in some more periportal or more pericentral hepatocytes, the outcome can be very distinct. So, in fact, the parasites that end up in a more periportal zonated, more periportal region, these infections tend to be abortive. So basically the parasite, a bigger proportion of parasites cannot make it to the end and cannot make this wonderful schizone that I showed to you that have all these beautiful and amazing number of parasites that tend to go to the blood stage and cause disease. So this is really something very interesting. I think is a lot of data there. We are just getting now the tip of the iceberg, and in the future we'll be able to show you much more about this. But going again, and just the last part, to our major question. Why are hepatocytes so special? And as I told you, we have been putting many pieces together, thinking that this is going to be probably a cocktail and many aspects need to be put in place for the parasites to be successful. But in the past few years, in fact, we made an observation from something that has been done by others from a very, very old days, but in fact, gave us clues that one piece of this puzzle might be it might be key and might be really, really important. So of course the question is, why was the liver selected by mammalian infectious Plasmodium species, and what allows the high replication of Plasmodium inside of liver hepatocytes? Because one thing that I didn't tell you, but is Plasmodium parasites that cause malaria can infect almost any vertebrate on the face of the planet Earth, okay? But only when these Plasmodium species reach mammalians as hosts, they really start to go to the liver. Prior to this, like Plasmodium species that infect reptiles or infect birds, they don't go to the liver. They also have a pre-erythrocytic stage, so meaning that the parasite stage that is transmitted by the mosquito is not able directly to infect red blood cells. They need to infect some other cell before, but while in all mammalian species, including man, you basically basically they go to the liver and is an obligatory. In other Plasmodium species, they infect some kind of, reticulum endothelial type of cells, macrophage-like cells, and other types of cells. They infect these cells, and in fact, in these cells, they need to go through several rounds because they really replicate in so few parasites. So instead of the 20,000-30,000 parasites that they replicate here in the mammalian liver here they probably make 50, maximum 100. So then, and this is known for several decades, but was when we start to think about this that we thought, okay, so probably what's selected to be here is the reason that allows the parasite to be replicating so much. And so we have gone through several possibilities, and we think the answer to this question lays in the uniqueness of the mammalian hepatic methionine metabolism. So methionine is really an essential amino acid for us, for the host, but also for the parasite. And so the parasite also needs to use to obtain this methionine, and it really goes through a cycle that here in gray, you have all the enzymes from the mammalian side, from the host side, and in blue, the enzymes from the parasite side. And the parasite this is an eukaryotic organism, correct? So basically, it has already all the capabilities of really synthesizing, metabolizing methionine, and really, using it. And in fact, many of these enzymes have been shown that they are really essential for the parasite when we really, trying to knock them out during the blood stage of infection, because the parasite really uses methionine. In fact, inside of the red blood cell, the parasite uptakes a huge amount of methionine that is used during the blood stage. And if we try to knock out any of these enzymes, we cannot make it. But one interesting observation that we did, it was that in fact, when we try to knock out one of these enzymes, this SAMS, the one that really is responsible from the first step of metabolizing methionine into SAM, basically, and it's called SAM synthetase. Basically, when we try to knock it out in blood, it was essential. But if we do a conditional knockout and then are able to go to the liver, basically, is completely non-essential. We can completely knock it out in the liver stage, and parasite still completely replicates into the same thousands of new parasites. So this was very surprising because it's essential on blood stage, but in the liver stage, somehow the parasite can completely, or probably already shuts off this enzyme and this cycle, and relies completely on the whole cell capability of metabolizing methionine. And so we start to think, so what is going on? So we know that the liver has a specific MAT1 enzyme, one of these enzymes that is capable, again, from our side, to metabolize methionine into SAM. And in our cells, we have two types. One that is only present in hepatocytes, and the other one that is present in other cells. In other cells, this enzyme is really, the activity of this enzyme is controlled by the level of product, by the level of SAM. When you have enough SAM in cells, basically it blocks the enzyme and you don't metabolize any more methionine. However, in hepatocytes, this is not the case. Really this enzyme really, it can be blocked, but at much higher levels. So really, you always have a huge amount of SAM in these hepatocytes. And so if we compare with this, yes, the parasite can also invite other cells, but when it invites other cells, really doesn't replicate much in its cells. But then when we have hepatocytes, it replicates much more. So we ask if this could be the reason to really have, an impact on the replication of the parasite. So basically what we now show, and telling a very long story very short, is that yes, the parasite in the liver is also dependent on methionine. It requires methionine. If we have a methionine deficient medium or a methionine deficient diet in mice, basically the parasite liver load is strongly decreased. The parasite really replicates much less. But as I mentioned to you before, if we remove the enzyme that is responsible for this metabolization in the parasite the parasite still can make it okay. But now if we really make a mouse that is deficient on this liver-specific enzyme, the parasite cannot make it the same way. So it's like if it is really in the absence of methionine, the parasite cannot replicate in the same number of parasites as it replicates in a wild type pathocyte. So then we show that in fact this dependence on methionine can really completely be substituted by adding exogenous SAM, the product of this. So what we think is happening is that the pathocytes really have something very, very special for the parasite. They have really almost an unlimited source of SAM that is the major donor of methyl donors, is of methyl groups, and so the parasite relies very much into that to be able to reach this level of replication. So just, to finishing, to thank, of course, the people that throughout years have been funding us, and also all the members of our group. This is already a picture taken after the pandemic, after is a relative term, but after having a big part of our population vaccinated in the country. Portugal has 87% of the population vaccinated, so being in a much better position these days. And of course, thank you all for your attention, and we are open, feel free to ask anything. So thank you.
Maria, thank you. That was really an incredible talk. I have some questions, but there's one that has come into the Q&A, so I'll read that out to you. And it is from Jim Boyer. Hi, Jim. Might it be possible to inhibit the entry of Plasmodium to the hepatocyte?
To block the entry? Yes. So we don't know yet what is the receptor of the host for the parasite to enter, but we know that there is in fact there are couple of molecules that from the parasite side that are very important. Especially one of them that is circumparasite protein, CS, is the most famous parasite protein. In fact, the vaccine that has just been now basically that WHO has considered that should be administered now in Africa. This vaccine is based on this protein, CS, covers completely the parasite when it's transmitted by the mosquito. And we know that if we have antibodies that block this protein, we can block invasion. Yes, we can block invasion. The only problem is that parasite invades very, very quickly. We are talking a question of seconds or minutes. So yes, if you have a huge amount of antibodies, you can block it. Probably never block 100%, and you don't need much. You just need one parasite to make it into the liver, then to cause a full malaria infection. So, or you have something that works 100% or doesn't work if we go for invasion. Yes.
The next question is again from Meri, and she says, very interesting talk. In the part where you see actin surrounding the parasite, do you know if the parasite is eliminated actively? For by instance, do you see actomyosin contractility or P-myosin that could suggest active elimination?
Mm-hmm. So really, if we block the actin-myosin complexes, we block elimination. So yes, so it seems that is through that complex, yes. Mm-hmm.
So if I may, I have one question for you. I was interested in the last part of your talk when you were talking about the high SAM environments in the hepatocytes. Do you know if people or mice are exposed to another agent that reduces SAM or creates a high SAM demand, does that then act as a protective mechanism to infection?
Yeah, so we don't know that, but it's something that we want to pursue. This is really very recent data that we are obtaining so far, and really, we really think this one could be something that in evolution was really being selected, by the parasite to go to the liver. But we don't have the answer to your question yet, but it's something that, yes, we would love to do that. What we know is that, another protein in the cycle of the host is GNMT, that is the next step, and really immediately uses SAM very quickly, okay? And if we block it and we have extra some, so it really cannot renovate. Basically, we also, the parasite cannot make it as well. So it's quite complex. It's not going to be as simple as we think it is, okay? So, I try to make it a simple story, but it's going to be extremely complex of how the methionine and its products are being used by the parasite.
Thank you for that. I will reach out to you because I think we might have a way that depletes SAM from hepatocytes, but I'll connect with you on that offline. So in the interest of time, I think we need to move on, and I'd like to thank Maria for a really beautiful and interesting talk. The last full talk of this session is Graham Heap. He is the global program leader at Takeda Pharmaceutical, and his talk is going to be on ARO-AAT TAK999 in patients with alpha-1 antitrypsin deficiency associated liver disease. Graham, over to you.
Thank you so much for the opportunity. Give me 1 second, I'll pull up my slides. Okay, how do we look? I'm not seeing any shaking the heads, so I assume we can see okay. Oops, and let's turn the subtitles off. 1 second. Okay, there we go. So thank you very much for the opportunity to present here today. I've got a few disclaimers before we start, as you might expect. First of all, I am an employee of Takeda Pharmaceuticals, and I do own stock in the company. Secondly, this talk was prepared by Takeda. The slides are going to provide an introduction to the mode of disease for alpha-1 antitrypsin liver disease and an investigational pipeline product. Efficacy and safety of this pipeline asset is currently under investigation. Regulatory approval of this asset is dependent on completion of the study program and review by regulatory authorities as we go forward.
Graham, I'm sorry to interrupt, but I'm hearing an echo when you're speaking. I wonder if there is a microphone that might need to be adjusted.
Give me a second. Let me try reducing the volume. Okay, is that any better?
Now you're a little bit quiet, but maybe we can proceed, and I'll jump in if it starts to echo again.
Let me know. It's just there's nothing fancy. It's just a laptop microphone. Okay, so we're here today to talk about alpha-1 antitrypsin liver disease. So alpha-1 antitrypsin is a neutrophil elastase inhibitor secreted by the liver that acts to suppress the activity of endogenous elastases throughout the body. Its primary mode of action, as it's seen in pathology, is its action in the lung. It also has significant actions in the skin and in other organs, where your body's being continually exposed to environmental insults, environmental pollutants, causing neutrophil elastase to become upregulated and active. The liver production of alpha-1 antitrypsin is a homeostatic regulator to prevent your elastase becoming overactive. When you have alpha-1 antitrypsin liver disease, which I'll talk through in a few moments, it manifests in two primary ways. It manifests as a chronic obstructive pulmonary disease with early onset emphysema, and it manifests with a slow, insidious fibrosing liver disease, eventually leading to cirrhosis. The mechanism of action of the lung disease related to the lack of alpha-1 antitrypsin in the serum, leading to attack by neutrophil elastase, and the mechanism of action of liver disease I'm going to talk through in a couple of moments. So AATD is a co-dominant disease, which essentially means when you have two alleles, they're both expressed. So unlike a recessive or autosomal dominant disease, where only one of the alleles is expressed and mediates the phenotype, both of these alleles are expressed at one time. This leads to a range of phenotypes of which different clinical manifestations can be observed depending on the genotype. The wild type allele is termed the M allele. The most severe mutation is termed the Z allele, and then there's an S allele, which is an intermediate phenotype. There's a range of genotypes which lead to a range of different effects on both the production of the serum alpha-1 antitrypsin and the degree of its misfolding and its subsequent activity. If you look at the serum AAT level, the wild type patients who are MM patients have serum levels ranging from between 20 to 50 micromolar. Alpha-1 antitrypsin is an acute phase reactant, as you probably know, which means these levels can increase significantly during disease, during infections and inflammation. But as a kind of baseline standard level in a healthy patient, we tend to see levels between 20 and 50. MS patients, so they have a single S genotype, but also a single wild type allele, display relatively normal levels, as you'd expect because it's codominant. When you have an SS or an MZ phenotype, so these are the intermediate phenotypes, you start to see reduced levels of alpha-1 antitrypsin. These patients tend to be phenotypically normal, but they do start to develop a risk of emphysema. It's still relatively low, and it's still not significantly increased above the background population, but you start to see some abnormalities. When you start to look at SZ patients, again, their emphysema risk starts to increase, but it's not until you get to the ZZ patients that the characteristic manifestations really start to show. So these are patients with very low levels of alpha-1 antitrypsin, below what we think is the protective threshold of about 11 micromolar. These ZZ patients present with early onset emphysema, but they also start to present with liver disease. There's some emerging evidence that some of the heterozygote phenotypes might be risk factors for the progression of fibrosing liver disease. I think this is still somewhat understudied, but there's some evidence to suggest that concomitant diseases such as NASH on top of an MZ or an SC phenotype display worse outcomes. The final phenotype of interest really in this disease is what's called the null genotype. So these are patients that have a specific mutation in the gene, which means they don't produce any alpha-1 antitrypsin. So these patients essentially have 0 levels. Sometimes you can detect levels around 1 to 2 micromolar, but it's normally absent. This is a very rare phenotype. There's only a small case series of these patients described, but they appear to have elevated risks of emphysema relative to the ZZ patients, suggesting that even low levels of alpha-1 antitrypsin have some protective effect on the lung. What's interesting about these null patients is they don't develop liver disease. The ZZ patients, however, do develop liver disease. If we look at the genetics of this population, it's a founder mutation that originated from northern Scandinavia. Most of the prevalence is found in outgrowth of this population around the world. So it's predominant in Europe, North America, Canada, Australia, and a scattering of cases across Latin America and the Middle East. But predominantly the most common geographic location for this is within Europe or North America. It is one of the most common single locus genetic diseases worldwide. It's prevalent in approximately 1 in 3,500 to 1 in 5,000 patients as a ZZ genotype in Europe and North America. This gives us a worldwide prevalence of around 250,000 patients. So it's still a rare disease, even if you accumulate the entire worldwide population. So alpha-1 antitrypsin itself is produced by the SERPINA1 gene. Single point mutations in the SERPINA1 gene are what lead to the S, M, and Z phenotypes that we see. We know that the ZZ genotype is the genotype that predisposes to liver disease, and it's this Z mutation which has been the subject of most study. So when you have a single point mutation that's encoded by a single SNP in SERPINA1, you produce a misfolded version of alpha-1 antitrypsin. This is termed Z-AAT. When the Z-AAT is produced, it's transcribed and translated in the ER. As it's secreted, small amounts of it are secreted through the Golgi into the serum. we saw a couple of slides ago, even patients with this mutation have low levels. It's just that it's present in subtherapeutic or levels that don't inhibit the elastase sufficiently in the lung to prevent lung disease. The challenge with this mutation is it causes polymerization with the endoplasmic reticulum. So the majority of this protein is formed. It forms in the ER and it starts to misfold. Some of it is subject to proteosomal degradation through the ERAD system. Some of it, the monomeric portion, enters the autophagosome and lysosomes and goes through their autophagic degradation system. But large amounts of it starts to polymerize with the endoplasmic reticulum. This polymerized Z-AAT forms characteristic PAS-D globules when viewed on histopathology slides. These PAS-D globules then appear to invoke hepatocellular injury, which leads to a pathway of inflammation, apoptosis, chronic regeneration, and subsequent fibrosis and cirrhosis, with the end-stage liver disease being the natural conclusion of this pathway. What's interesting is this is not the outcome for all patients. This process occurs in all patients, but environmental and genetic insults add on to this process and affect why some patients develop a more characteristic lung disease and why some patients develop a more characteristic liver disease. Alpha-1 antitrypsin has the bimodal distribution, as I'm sure many of you know. We see it as a predominant cholestasis in neonates. These patients are usually relatively easily recognized. It has a very high diagnosis rate. They present with high AAT, cholestasis, and most of these patients spontaneously resolve with appropriate supportive care. A small number of them go on to develop more severe liver disease that progresses through childhood and adolescence, and a very small number of them develop severe life-threatening liver disease and require transplants. Only small numbers of these patients are transplanted each year, though, especially in the US. That's not the outcome for most patients, though. Most patients have a benign childhood course and are diagnosed in adulthood. If you look at the overall population of alpha-1 antitrypsin patients with the PiZZ mutation, most of them don't have any clinically significant liver disease diagnosed. We know that about 35% of them, though, have F2 metavir fibrosis or greater. Some of these are picked up with portal hypertension. Some of them are diagnosed as end-stage cirrhotics. Some of them are not detected until they have liver transplants and the subsequent sequelae that goes with them. If you look at autopsy studies of alpha-1 antitrypsin lung disease patients, up to 50% of those patients have cirrhosis at autopsy. So we think there's significant underdiagnosis of this liver disease within this population, which is not really surprising considering, as with other chronic liver diseases, F2 and F3 fibrosis and even early stage cirrhosis are asymptomatic. So when we think about why these patients develop fibrosis, the pathogenesis suggests that it's the Z-AAT protein production that is the inciting insult for the development of the inflammation and subsequent fibrosis cascade. If you look across a panel of patients, and this is work that Ginger Clark's produced out in Florida, with a spectrum of different fibrosis markers, levels, from F0 to F3, there's a significant difference in the concentration of PAS-D granules that you see within those hepatocytes. We know that patients who have earlier stage fibrosis tend to have few or rarer incidents of PAS-D granules, and as we go up to patients with more severe fibrosis stages, the PAS-D concentrations increase significantly. There's lots of reasons you could hypothesize this might be. Some patients might have more efficient clearance mechanisms. It might be a consequence of other insults. It's all subject of a lot of intensive study at present. But what we do know is that patients who have high polymer loads don't survive as long as patients who have low polymer loads. If you just stratify people based on a very simple assessment of their polymer load, you can see that by the time you get to 1,000 days out, the survival of patients with very high degrees of polymer within their liver is markedly reduced from those much lower levels. So all of this put together, Z-AAT, the polymeric protein, is an intriguing target for development of a therapeutic for alpha-1 antitrypsin liver disease. One of the processes that has been investigated for reducing this protein is an RNAi-based approach. So there's a few RNAi therapies that are being developed at present. There's a few for some rare diseases that have actually made it onto the market. Fundamentally, the way they work is by inhibiting the transcription translation cascade. So the SERPINA1 gene is transcribed as normal. The messenger RNA is produced, but an RNAi is introduced which degrades the mRNA and prevents it being translated into protein. The mRNA itself is just degraded by the RISC complex. So the RNAi technology is a small double-stranded RNA-based approach that's introduced into cells. The strands bind into the RISC complex. The antisense strand, which is the guide strand, remains in the RISC complex. The sense strand is discarded. That RISC complex then has a sequence-specific target on it, and it goes around and looks for the messenger RNA that it wants to degrade. When it meets the SERPINA1 message, the SERPINA1 message is then degraded, and the RISC complex remains active and continues searching for other messenger RNAs that it can degrade if it meets its target. So TAC999, which you might also refer to as ARO-AAT, is a small interfering RNA therapy. The aim of these kind of targets is to make them as specific to cell types of interest as possible. So this investigational product is actually GalNAc conjugated. The addition of the GalNAc means that we try to make it as hepatocyte specific as possible. So the product is taken up preferentially by hepatocytes, and the theory is then that we can try and degrade the alpha-1 antitrypsin protein specifically within the hepatocytes. So that's the theory. If we then start to look at, does this work in practice? The first animal models we use are transgenic mice with a humanized PiZ transgene. Obviously, because the PiZ is not a completely conserved sequence, there's not much point in us testing a humanized therapy in a mouse, so we have to transgene develop humanized mice first. So what you can see here is a 5-week-old PiZ mice treated with a investigational TAC999 product by subcutaneous injection, treated every 2 weeks for 8 weeks, versus mice that were euthanized at baseline, which was at 5 weeks of age. So if you look at both baseline and the saline control, there's normal levels of Z-AAT messenger RNA, and then the untargeted RNAi control, so this is just a non-sequence specific RNAi, also has normal levels of Z-AAT. The sequence-specific TAC999 message, however, appears to be giving profound reductions in the message expression of the Z-AAT protein. So this is nice initial proof of principle that the agent is degrading the messenger RNA through the RISC complex in the way we expect it to. But obviously that's only message. So if we then move forward and look at what happens to the protein, this is the same cohort as before with the same experiment. This is looking at monomeric Z-AAT. So at baseline, we see patients, or mice, sorry, who have what would be normal levels of the monomer. 8 weeks later, with the saline control, again, very similar levels of monomeric protein, and the RNA control on the right with no significant difference between those two, but very profound reductions with treatment in the monomeric protein. The monomeric protein, though, we don't think is necessarily the pathogenic agent in this condition. The pathogenic agent that we're really interested in trying to reduce is the polymer. On the graph on the left, the baseline is again mice euthanized at 5 weeks. The saline control is then 8 weeks later. So you can start to see the amount of polymer that's accumulated in that 8-week time frame. You don't see it in the monomer as it's cleared from the liver relatively rapidly. And with therapy, we're starting to see reductions in that polymeric formation. This is probably due to a combination of reduced Z-AAT production and some of the hepatocyte being able to regenerate and clear some of the polymer as a result of the reduced insult. We start to see reductions in the number of PASD globules. We start to see a reduction in the amount of the liver that has these globules in it. And we start to actually see a reduction in the size of the globules relative to controls. So once we've been through the process of, we've got a therapeutic hypothesis, we've tested it in mice, it appears to be working in the way we predicted it would work in mice. The next approach is to think about how this might perform if we administered it to humans. So I'm just going to show you one slide on clinical data. This is the phase 1 study that was conducted in healthy volunteers. There's 2 pieces to this study. There's a single ascending dose portion and there's a multiple ascending dose. In the single ascending dose study, patients were administered 35 milligrams of TAK-999 versus placebo, and then open label 100, 200 or 300 milligrams of TAK-999. The results of that are at the graph on the top right. If you look at the serum AAT, these patients, if they're administered placebo, continue to have normal levels. Remember, these are normal healthy volunteers, whereas all 3 doses of the investigational product reduce serum AAT within a deer down to around probably 8 weeks. We had no deaths or serious adverse events during this study. No adverse events were rated as severe. And there didn't appear to be any pattern of adverse events that changed with dose escalation. The most common adverse events, so those seen in more than 25% of individuals, were upper respiratory tract infections, headache, and injection site reactions. The injection site reactions do include vascular access, bruising, hematoma, pain, and swelling. And importantly, there was no statistically significant difference in FEV1 changes from baseline for either placebo or TAK-999. But I think you have to remember this is a very short duration of therapy. I think what's interesting when you look at this is the duration that this appears to stay knocked down for. We do start to see some recovery, but not until you get out well beyond 6 months worth of duration, even after a single dose. So in summary, alpha-1 antitrypsin is a rare condition that increases the risk of lung and/or liver disease. We know that not all patients progress the same way. Some patients present with lung disease, some patients present with liver disease, some patients present with both at the same time. At present, there's no pharmacological therapy for AATD other than supportive care and ultimately liver transplantation. And as you've seen here, there's specific therapies under development, and we hope to provide more information on these in the future. Thank you very much.
Graham, thank you for that exciting talk. That's really significant progress for these patients. I think we're going to have time for one question because we are running a little bit over in this session. And I'll read it out to you. It's from Tobias, and he asks if you have thought about using LNP-mediated CRISPR approach to modify AAT DNA and eliminate AAT RNA production.
I think the short answer is yes. There's a number of therapies in development for alpha-1 antitrypsin liver disease. I think when you think about what therapies you could look at, there are approaches to knock down the message. There's therapies to try and modify the protein to form it to form back into a normal shape. And then there are gene therapy based approaches to try and reinsert a, I'm going to put in inverted commas, normal alpha-1 antitrypsin gene, or to base edit the existing genes to correct the mutation. All of these are being investigated. Some are at different stages of development than others. And I think we continue to see which one will eventually prove to be safe and effective and hopefully provide a therapy.
Thank you for that, Graham. I'd like to move on to the next part of the program, which is really quite exciting. And I would like to ask all of you to remember back to the time when you could go to a conference and there would be coffee waiting outside the lecture hall, and there may be posters up around the coffee area like there were at many of the Arias Symposiums that we've been to in the past. And just before we break for the coffee, there would be 2 perhaps micro talks or just poster teasers given by some of the poster presenters. But unfortunately, we don't have posters at this session or at this meeting today, but we still do have these micro talks. So I will introduce these very rapid talks given by 2 scientists who have very exciting data. The first one, it will be Dr. Joon Jung, who is a senior scientist at Camp 4 Therapeutics in Cambridge. Dr. Jung?
Can you allow me to share my screen? Thank you. I'll share. While you guys having coffee, I'll updating. First of all, thanks for giving me an opportunity to share our recent work. And while you guys are waiting for the coffee and drinking coffee, I will talk about the novel way of upregulating gene expression for treating OTC deficiency. In the nucleus, the expression of mRNA is precisely controlled by, let me turn on the laser, regulatory RNA, such as promoter associated RNA or enhancer RNAs or naturally occurring antisense transcripts. All these happens in 3D DNA structures known as an insulated neighborhood. Regulatory RNAs, in short, reg RNAs, are transcribed within the insulated neighborhood to control the gene expression. With the power of next-gen sequencing and proprietary machine learning system, CAN4 can efficiently map reg RNAs and design and screen the potent RNA actuators to upregulate specific mRNAs. Urea cycle is a biochemical pathway to detoxicate ammonia to urea for elimination. And it consists of 5 enzymes and 1 cofactor. When patients have a loss of OTC function, it leads to built up ammonia that cause neurological defect. So our approach is increasing hypomorphic gene expression by targeting corresponding reg RNAs. Increasing OTC enzyme to at least 5 to 10% of normal level has tangible therapeutic benefit. Our platform identified active enhancer that impact OTC. Then we identified associated regulatory RNA that control the mRNA expression. Then we have been through multiple rounds of screening, including initial screening, tiling around the hit, and fine-tuning with a different chemistry. That leads to 4 OTC RNA actuator that when we treated these RNA actuators in the multiple donor cell, we observed that there is a upregulation in OTC mRNA in dose-dependent manner. When we treated these RNA actuators in human OTC patient cell, it increased ureagenesis along with expression of OTC mRNA. Very similar to human, we also identified separate RNA actuators that can increase mouse OTC mRNA in mouse OTC deficient hepatocyte. Currently, we are conducting an in vivo POC study in OTC deficient mice with the selective RNA actuators. In summary, we identified RNA targeting RNA actuators that can upregulate human OTC mRNA in dose-dependent manner. Our results indicate that our technology may provide a precise and potent therapeutics that can be programmed to treat thousands of different diseases.
Thank you so much. So in the spirit of micro talks, we are not asking any questions for the audience, but I will ask you to stop sharing your screen so we can move on to the next micro talk. And thank you for that very exciting advance that you shared with us today. Okay, so the last speaker of this session and the next micro talk speaker is Dr. Ma, who is a postdoc in the Yanish lab at the Whitehead Institute at MIT, and he will talk to us about human-induced pluripotent stem cells.
All right. Hey, good morning, everyone. Can you see the slides and can you hear me okay?
Yeah, all good.
Okay, great. First, I would like to thank the organizers for this opportunity to share our work. Today, I would like to share our study to look at the epigenetic mechanism regulating the human pluripotent stem cells, differentiate the hepatocyte, and what are the mechanism regulating its maturation. So I like to first acknowledge the people enable this work. I would like to thank my mentor, Professor Rudolph, with a couple of collaborators. So as a very brief background, this audience is well aware that the many functions of the liver and the hepatocytes. For example, it's mediating a lot of the disease with both genetic and environmental factors, such as type 2 diabetes, which is becoming a big problem right now with additional non-alcoholic fatty liver disease, for example. So with that, there been quite a lot of interest to understand the human liver biology and potentially to use a renewable source of a human hepatocytes, such as from the human pluripotent stem cells, including the work from Dr. Takebe's group we heard in this morning. However, there has been a known issue that a lot of the stem cell differential hepatocytes among other cell types display this immature phenotype, and it's been hypothesizing that potentially the environmental factors, including the nutrient metabolites and potentially hormone regulation, contribute to this maturation process. So in our lab, we're interested in epigenetics. We hypothesize that there might be an epigenetic underlying for this process. So to study this one, we develop a spheroid differentiation system composed of both 2D and 3D differentiation. And for this one, we focus on derived relatively pure hepatocytes. And with that, we subject this both the 2D and 3D differentiation and also the primary non-cultured hepatocytes as our positive control for both gene expression and epigenetic mapping with the genomic approaches. Just to summarize a lot of data, we found that if we look at the active components, regulatory elements, we show different enrichment of the thyroid hormone receptor beta, which is a major thyroid hormone receptor in the hepatocytes. As you can see, the blue bar from the stem cell differential cells, they have a lower enrichment compared to the primary cells. This is true for the ataxic enrichment and also for the H3K27 acetylation ChIP-seq. So, but the gene expression of the thyroid hormone receptor did not differ. So we hypothesize that potentially this regulation happened at the ligand availability level. So we provide the thyroid hormone T3, and we can see enrichment of increased expression of mature hepatocyte marker like CYP3A4 and reduce the fetal cell marker like AFP. Furthermore, we can see the increased enhancer levels at the key genes and also at the global level as shown up in this cyan dot. And we can also see this a correlating change in the chromatin accessibility, including the increased chromatin accessibility of CYP3A4 and decreased accessibility of AFP. Furthermore, we show that we can transplant them into non-damaged liver in immunocompromised mice. So in the last little bit, we'll try to understand what are the mechanisms by which the thyroid hormone receptor mediated these changes. So we use the IP mass spec to identify protein interacting with thyroid hormone. And we found multiple components of this chromatin remodeling complex called the BBAF, including the tumor suppressor, the liver cancer tumor suppressor ARID2, which will also confirm with Western blot. So just to summarize what we have here for the consideration of time, not go into every aspect, I just mentioned that we propose that the thyroid hormone through the thyroid hormone receptor beta, potentially recruiting this chromatin remodeling complex PDAF to regulate chromatin accessibility and then establishment of the enhancer that are responsible for the activation of the genes involved in the liver maturation. And I would like to thank you for your attention. And if you have a further question, we can definitely feel free to reach out to me. Thank you.
Thank you, Dr. Ma. And I'd like to take this opportunity to thank all of the speakers in today's session. One downside of having a virtual conference is that you don't get to hear the applause that everybody is doing when they're sitting alone in their own room. So on behalf of the audience, I would like to applaud the speakers. And then, thank you. And I would like to close the session.
Good morning, everyone. Once again, on behalf of ALF, I want to thank you each for presenting this morning. It was certainly an exciting first session. And as we move into our second session, I want to introduce you two short videos to our gold corporate sponsors for today, Eisai and Salix Therapeutics. Thank you so much.
Right now, millions of patients are suffering with GI disorders such as overt hepatic encephalopathy, irritable bowel syndrome, chronic idiopathic constipation, opioid-induced constipation, bowel preparation, and ulcerative colitis. At Salix, we have kept up with these unmet needs for more than 30 years by providing an industry-leading portfolio of high-quality products that address these conditions. Through our dedicated and innovative R&D team, we're also exploring potential new scientific breakthroughs. And we remain committed to important healthcare provider investments in education, charitable grants, and sponsorships. We will continue to invest millions of dollars in patient education and do our best to broaden access to Salix medications. Because our mission to improve the care for GI patients continues today. For us, there is no finish line.
Hi, everyone. Welcome to session 2. My name is Yasuko Iwakiri from Yale University. I'm really honored and excited to be here moderating this session. The Arias Symposium has been my favorite meeting since I was a postdoc, which was many years ago. Happy 30th anniversary. Now, it's my great pleasure to introduce our first speaker, Dr. Shalev Itzkovitz from Weizmann Institute of Science. Dr. Itzkovitz is a recipient of the Human Frontier Career Development Award and the ENBO Young Investigator Awards. In 2017, he was selected as an HHMI International Research Scholar. Today, he's going to talk about a really interesting topic on liver donation in health and disease. Dr. Itzkovitz, over to you.
Thank you very much. Can you see my screen? Is everything okay?
Yes, I can hear you well and can see the screen.
Okay, thanks. I want to thank the organizers, Sangeeta, Wolfram, Dr. Arias, for this opportunity to present to you today. As all of you know, the liver is the central organ in our body. It has a vast array of functions, from storage of nutrients and the regulated release, detoxification of blood, production of most of the carrier proteins in the blood, cholesterol, bile acids, a very vast array of functions. And all of these functions are performed by a single cell type, the hepatocyte. But the liver is not just a bag of hepatocytes that all perform these functions equally. It's actually highly heterogeneous. And this heterogeneity is related to the anatomical structure of the liver. As we know, the liver is composed of repeating anatomical units, these hexagonal shaped columns called liver lobules, 0.5 millimeter in diameter in mice and 1 millimeter in diameter in a human. And blood flows through from the corners of these lobules and drains inwards towards a central vein. And this graded blood flow creates gradients of oxygen, nutrients, hormones. So the hepatocytes reside on rows along the sinusoids. They take up material and release material. And this creates gradients. For example, oxygen is rich in the portal areas, but as blood flows inwards and hepatocytes respire, oxygen level decrease. And there's also morphogen sources. And so this is a highly graded microenvironment. And in response to these gradients, if you section the liver and start staining for different functions or cellular properties, you can immediately appreciate the heterogeneity. So here on the right, you see staining for messenger RNA of albumin. Albumin is sort of the marker of hepatocytes. And you can appreciate the heterogeneity. Expression of albumin in periportal hepatocyte is much higher than in pericentral hepatocytes. Same goes for glycogen content. It's non-uniformly distributed along the lobule radial axis, and for a few other dozen functions. So this was known for decades. It's a phenomenon that has been termed liver zonation. And in my lab, we wanted to generate a high-resolution spatial atlas of liver zonation, of hepatocyte zonation. And we thought that having such a spatial map for all of the hepatocyte genes would help understand design principles of liver tissue organization. And so to achieve this, a few years ago, we teamed up with Amit, a fellow lab in my institute, and we performed single-cell RNA sequencing of dissociated hepatocytes, basically perfused the liver, and we extracted the 1,500 hepatocytes, and for each one, measured the complete transcriptome. We did this matrices of 30,000 genes by 1,500 hepatocytes. And for every hepatocyte, we have the complete transcriptome. So this is great. This is a picture of hepatocyte heterogeneity. But the problem is that when we dissociate the tissue, we lose track of which lobule layer every hepatocyte originated from. So to retrieve this information, we combined the single-cell RNA sequencing with a different approach, a microscopy-based approach of single-molecule fluorescence in situ realization that allows us to visualize individual RNA molecules of any gene of interest in the intact liver tissue. And we designed single-molecule fish probe libraries for known zonated hepatocyte genes. And you can see an example here of glutamine synthetase only expressed around the central vein. CYP2E1 that has a gradient from the central vein towards the portal vein. Albumin, you already saw before, has an inverse gradient. And for each one of these genes, we segmented hundreds of hepatocytes while recording their position along the lobular axis. And we counted the number of RNA molecules and sort of created an internal barcode. So now if you give me an hepatocyte and you tell me that it has 300 RNA copies of ASS1, I can infer what's the probability that this hepatocyte originated from the pericentral layer, the mid-lobule layer, or the portal layer. Now we can use this information to retrospectively determine the location of every sequence hepatocyte. So in my table of 30,000 genes by 1,500 hepatocytes, I ignore all the genes and I only look at the genes that have mapped under the microscope, my landmark genes. For example, the blue periportal hepatocyte gene and the red pericentral hepatocyte gene. And if I see an hepatocyte with 34 copies of red, 2 copies of blue, I infer it originated from the inner layer, from the pericentral layer. If I see another hepatocyte with 4 copies of red, 50 copies of blue, I know that it must have originated from the portal layer. And we built an algorithm to map these 1,500 hepatocytes, 1,500 numbers into 1,500 numbers between 1 and 9. Basically, we associated every hepatocyte with a layer from layer 1, the most pericentral, to layer 9. And now for every gene that I've never measured under the microscope, I can average the expression of all the hepatocytes from layer 1, 2, 3, etc., and obtain a whole transcriptome zonation map of hepatocytes. And so this is the map you see here. Every row is a gene. Every column is a different layer along the central to portal axis. Yellow is high expression, blue is low expression. What we found is that the number of hepatocyte genes that were non-uniformly expressed along the lobular axis was much higher than was previously thought. So we had a few thousand genes that were significantly changing along the lobular axis. We had pericentral genes, periportal genes. We found a new family of genes that peak in the mid-lobule layers. Here's an example of Hump, a gene that encodes hepcidin, a hormone that's secreted by hepatocytes and that regulates systemic iron levels. Every bright dot here is an RNA molecule of Hump, and you can see that it's low around the portal vein, then it peaks in the mid-lobule layers, and then it decreases towards the central vein. Having a whole transcriptome map allows you to classify different functions into different layers. So we found that most of the genes that encode proteins secreted by the liver, like clotting factors and the complement system and carriers like albumin, were mostly expressed in the outer layers of the lobule, similar to albumin here. So you can see expression is higher in the portal layer compared to the central layer. And this makes a lot of sense because translating proteins takes a lot of ATP, a lot of energy. So allocating these functions, these processes to the portal layers that have more oxygen availability might be more efficient for the tissue. Other processes like liver detoxification were expressed mostly pericentrally, but there were some interesting exceptions, like CYP2E1 that you can see here on the left. And we also found some processes that were actually distributed along sequential hepatocyte layers. So this is an example here of bile acid biosynthesis, so cholesterol converted to bile acids through an enzymatic cascade. And we found that the genes that encode sequential enzymes were expressed in sequential lobule layers. For example, the two first enzymes were expressed in the innermost layer of the lobule, whereas the third enzyme, CYP8B1, was actually repressed in the inner layer and up and peaking in the second layer. So since we had the highest pressure resolution, we could resolve these kind of distributed cascades. So it's sort of like a production line pattern in a factory, where potentially intermediates might be transferred from one hepatocyte to the adjacent one. So we have a global map of hepatocyte zonation, and there's a few design principles that are emerging. I already told you about the allocation of energetically demanding tasks to the highly oxygenated part of the tissue. This goes for secreted proteins, but also gluconeogenesis, a process that takes up a lot of energy for hepatocytes, and it's also periportally zonated. We saw production line patterns. There's examples of recycling of material between different zones. We have very nice examples of transporters that that are zonated in a way that matches the ligand concentrations. For example, glucose concentration is high at the portal layers where it comes from the intestine. And the transporter of glucose that's better in high glucose concentrations, BLAT2, is expressed in the portal area. But then as blood flows inwards and hepatocytes take up glucose, glucose concentration decrease, and the pericentral hepatocytes upregulate GLUT1, which is a transporter that works better in low concentrations. It's really a vast array of sort of design principles. And if I were an engineer I would build an optimal tissue, I think this is the way I would build the liver. So that's it for hepatocytes in health. I want to also tell you that the hepatocytes are not the only cell type in the liver. Of course, there is a vast array of very important supporting cells, including hepatic stem cells, Kupffer cells, endothelial cells. These are static cells mostly that are exposed to the same gradients of hormones, nutrients, oxygen. And we wanted to know whether they are also exhibiting spatially dependent gene expression patterns, and whether these patterns can interact with the zonated patterns of hepatocytes. And so we started with single-cell RNA sequencing of these non-parenchymal cells. Every dot here is one of these cells, and closed dots in these T-SNP plots are cells that are similar in their transcriptome. And we colored them according to several landmark genes for epithelial cells, Kupffer cells, B cells, T cells, etc. But what we're interested in is whether, for example, endothelial cells have zonated patterns, whether endothelial cells around the portal node would be different than mid-lobular endothelial cells or pericentral endothelial cells. And so the problem here is that for endothelial cells, unlike hepatocytes that were studied for decades, we really didn't have enough landmark genes, known landmark genes to locate them. So there were only about 3 or 4 landmark genes that were known to be expressed by pericentral endothelial cells or portal endothelial cells. And endothelial cells are also very, very small. And so when you perform single-cell RNA sequencing, you get a few hundred copies. They are spread along 5,000 genes. And so most of the genes are expressed in 0 or 1 copy. And so having a small set of landmark genes is just not enough to localize these cells. So to circumvent this challenge, what we did is to develop an approach called paired cell RNA sequencing. So we already have single-cell RNA sequencing of endothelial cells and hepatocytes. Now what we wanted to do is to suboptimally dissociate the tissue to obtain pairs of an hepatocyte and an attached endothelial cell. And we sort these pairs into the same capture well. And so some of the RNA molecules come from the endothelial cells and some come from the hepatocyte. And we can use the hepatocyte transcriptome to determine whether these pair come from the pericentral layers, mid-lobular layers, or portal layers. And then we can switch to genes that we know are only expressed in endothelial cells and identify endothelial specific genes that are differentially expressed among pairs that are that originated from different lobule layers. So that's the approach, and the way that we do it is by isolating large cells, which are hepatocytes, that are positive for CD31. Of course, no hepatocyte is positive for CD31. Since this large cell is positive for CD31, this is a liver endothelial cell, and this is what we sort in a sequence. And from this paired cell RNA sequencing, we found that endothelial
Dr. Itzkovitz, I think
cells as well.
I think your voice was cut and we lost your slide now. Yeah, I can see now. And please unmute.
Can you hear me now?
Yes.
And can you see my slides?
Yes, we can see now.
Okay, I'm sorry about that. Okay, and so basically we obtained the zonation map for liver endothelial cells as well, and we validated many of these. For example, you can see a gene here that we predicted to be zonated delta-like 4, and the red spots here are RNA molecules of aquaporin-1, which is a pan-endothelial cell marker, and the green dots are RNA molecules of delta-like 4, and indeed our prediction here is validated that periportal endothelial cells have more RNA molecules of delta-like 4 compared to the mid-lobular endothelial cells and pericentral endothelial cells, and we validated quite a few of these. And the zonation map here gave us quite a few findings that were interesting. For example, it helped us characterize endothelial cells that surround the central vein that secrete very important morphogenetic cues like Wnt2, respondin 3, and other molecules that actually shape a lot of the hepatocyte zonation patterns. Okay, so I'll just summarize this part. I've told you about spatially resolved single cell reconstruction of hepatocytes and a paired cell RNA sequencing approach that helps us reveal zonation of supporting liver cells, and it can be applied to other cell types. Now I want to switch gears and ask what are the implications of liver zonation in disease, in pathological processes. And what I'll discuss in the next few slides is work by my very, very talented ex-PhD student. She just finished, Shani Ben Moshe. And she was interested in acetaminophen intoxication, and the work is described in this preprint we recently posted. And so one of the important pathological processes in the liver is drug-induced liver injury. So of course, acetaminophen intoxication is a classic model system for this. When we have an overdose of acetaminophen, all of the hepatocytes that express CYP2E1 die. They become necrotic. The idea is that hepatocyte use CYP2E1 to generate NAPKI, a toxic intermediate. And if the levels of NAPKI are too high, the hepatocyte dies. And so following an overdose of acetaminophen, you can appreciate that 2 days after an overdose of acetaminophen, all of the pericentral hepatocytes have died. So there are no nuclei here. These are necrotic hepatocytes. All of the nuclei are non-parenchymal cells, and this is 2 days after damage. So a very massive damage to the liver lobule, and it's a zonated damage. But then 1 day later, 3 days after injection of acetaminophen, you can see that the gap starts to close. So we have new hepatocytes coming in towards the central vein. And actually 4 days after injection, histologically, the lobule is almost complete. So it's a remarkable regeneration process. It entails very complex procedures, clearing up dead cells, building matrix, generating new hepatocytes that will hopefully resemble the original hepatocytes. Because remember, while histologically the liver here looks identical to the control liver, the pericentral hepatocytes here 4 days ago were mid-lobular or periportal hepatocytes. So they came from a completely different zone. And noting the vast differences in expression between different lobule layers, he wondered whether they remember their original identity, whether they efficiently reprogram, and how do they do that, and in what time scales. And so to address this, we first did bulk RNA sequencing of liver tissue in different time points after injection, 6-hour, 24-hour, 48-hour. This is a PC plot. Every dot here is a single mouse. And you can see that gene expression diverges at 6 hours, 24 hours, but then at 48 hours starts to go back. And at 96 hours, 1 week, 1 month, we're already back to control levels. Now we can also take from our single-cell RNA sequencing extract hepatocyte-specific genes, about 2,000 genes that we know only come from hepatocytes, and examine the expression of these hepatocyte-specific genes. And when we plot the distance in hepatocyte transcriptome between the injured livers and the controls, we see the divergence towards 24 hours, 48 hours. But again, 96 hours, molecularly, not only histologically, the hepatocytes seem to be going back to normal. And we also stained for zonation markers. And again, this is CYP2E1, the pericentral marker, and ASS1, a portal marker and controls. And 4 days after damage, the newly made hepatocytes that come from the green zone, they shut down the green gene, upregulate the red gene. Same goes for glutamine synthetase. So there's zonal reprogramming that's very efficient and happens within 4 days. And the question is how, in what way is it happening? So to address this, we perfused the livers of these injured mice, and we extracted the hepatocyte, performed single-cell RNA sequencing, and we used our hepatocyte landmark genes to determine the location of each one, the original lobule location of each one of these sequenced hepatocytes. So for example, here we stained the UMAP by the expression of CYP2E1, the pericentral marker, CYP2F2, the portal marker, and we can assign computationally a lobule layer for each one of the sequenced hepatocytes. And we also have time after damage. And so we see that in a control, we have a distribution of all the lobule layers. 48 hours after damage, we only have the portal hepatocytes, and then 72 hours, the pericentral identities start to come back. But now when we compare the most interesting population, which is the population of the most pericentral hepatocytes, the one that are pushed into the damaged area and need to reprogram, we call these interface hepatocytes because they're at the interface with the damaged area. So when we compare the expression there, we found something very interesting. Here, every dot is a gene. The X expression, the X axis is the expression in hepatocytes that are in the same lobule layer as the interface hepatocytes, and the Y-axis is the expression of these incoming most pericentral interface hepatocytes. What we found is that these hepatocytes don't simply shut down portal genes and upregulate the central genes. They express a complete program that's not expressed at all in adult livers, consisting of fetal genes that are expressed in the fetal liver or in hepatocellular carcinoma like AFP, alpha-fetoprotein, CDH17, the ALF-P, SPP1. Here's an example of fish validation. So we're talking about these interface hepatocytes, the most pericentral hepatocytes during this regeneration process. And you can see here that every bright dot is a single RNA molecule of AFP. So they specifically upregulate very high levels of AFP, CDH17, which is a Wnt facilitator. AFP is, of course, the fetal version of albumin, SPP1, osteopontin, and a whole cassette of genes that are basically a different cellular state than anything we see in a normal liver. So rather than having immediate transdifferentiation, we have sort of a de-differentiation into this interface hepatocyte state and then redifferentiation into a pericentral state. Having the single-cell RNA sequencing, having hepatocytes stratified by a lobule layer, we can also, just one more thing about these interface hepatocytes, they're not only different transcriptionally, they're also dramatically different morphologically. So you can appreciate that these interface hepatocytes send protrusions into the damaged zone. And when you measure their circularity, they're significantly less circular than the non-interface hepatocytes. And since we have single-cell RNA sequencing of these interface hepatocytes, we can ask what other programs they're upregulating. And what is very interesting is that these interface hepatocytes upregulate very, very substantially both ribosomes and proteasomes, and also oxidative phosphorylation. So the idea here is that these hepatocytes need to degrade all the periportal proteins and massively translate new pericentral proteins. So we think that's the reason why they upregulate both ribosomes that translate new proteins and proteasomes that degrade the old proteins. And they also upregulate the MYC targets and oxidative phosphorylation, because in order to translate proteins, you need to have a lot of energy. So just the fish validation for this, these are the interface hepatocytes and the non-interface hepatocytes. And you can see that the interface hepatocytes have many more red dots, RNA molecules of the proteasome genes compared to the non-interface hepatocytes. And they also have much higher levels of ribosomal protein encoding genes, the green dots, compared to the non-interface hepatocytes. Okay, one last thing about the hepatocyte single-cell RNA sequencing here. Since we have the hepatocytes stratified by zone, we can also ask which hepatocytes proliferate. And this was interesting because it turns out that at the peak of proliferation, which is 48 hours after damage, there is a slight zonal tendency for hepatocytes to divide that are pericentral, that are facing the damage. But actually, there's quite a lot of divisions also in the mid-lobular and periportal hepatocytes. And actually, 72 hours after the damage, division occurs equally across all zones. So every bright dot here is an RNA molecule of Ki67, a proliferation marker. And you can appreciate that even hepatocytes that are portal and are very far away from the damage are proliferating massively. And the idea here, we think the design principle here is that if only the interface hepatocyte had divided, then it would take 3 divisions to make 3 new cells. But then if all of the hepatocytes are dividing across the plates, they're creating new cells that create a massive mitotic pressure. And in one division, we can actually close the gap completely. So this perhaps serves to close the injured region as fast as possible. So just to summarize, the hepatocytes rapidly regenerate after acetaminophen intoxication. They proliferate throughout the lobular axis, and they undergo a transient change in gene expression and morphology with this interface hepatocyte cellular state. 2 last slides, if I have time. We talked about supporting liver cells. So how do non-parenchymal cells support this dramatic process? To address this, we complemented the study with single-cell RNA sequencing of about 20,000 non-parenchymal cells throughout the regeneration process. And we got this map of the different cell types. The colors here, black is control, gray is the peak of injury, 24 and 48 hours. And then pink towards you is the reversal phase, 3 days, 4 days, and 1 week after the damage. And so you can see some, I'll just describe, I don't have time to go deeply into this, but for each one of these populations, we also have the zonal coordinate because we use our landmark genes. So I'll just tell you some of the insights that this atlas gave us. So you can see that at the peak of the damage, we have a huge influx of monocytes and macrophages that you can see here. And then 3 days or 4 days after the damage, they completely disappear. Kupffer cells also have an activated state 24 hours after damage, and then Kupffer cells revert back to their normal state. Hepatic stellate cells have massive activation of collagen genes, but then 3-4 days later, they become quiescent again, which you can see here from this part of the atlas. There's changes in the endothelial cells. One thing that's very prominent in this atlas is that T cells and B cells look quite similar between the control and the injured state. So generally, we don't see more T cells or B cells during this regeneration process. And we also don't see different cellular states of T cells and B cells. And in conjunction with this, we see a massive decline in all the genes that are related to antigen presentation throughout this regeneration process. What we think is going on here is that when hepatocytes die, there's a lot of antigens that could be presented and could stimulate the adaptive immune system. And we think the system strives to keep the adaptive immune system quiet. And so there's a decline in antigen presentation. And as a result, perhaps no changes and no influx of T cells and B cells. So just to summarize, I've shown you a very interesting regeneration process that in 4 days completely rescues the tissue. We saw pericentral necrotic hepatocytes, interface hepatocytes that upregulate translation and protein degradation and become morphologically distinct. Portal hepatocytes that divide to create a mitotic pressure to seal the wound fast. I didn't tell you about it, but hepatic stellate cells also have zonal differences. Hepatic stellate cells in the damaged zone rapidly secrete collagen, whereas periportal hepatic stellate cells divide to give rise to new hepatic stellate cells that could then migrate inwards, perhaps to assist in collagen buildup. Endothelial cells express HGF, also, I didn't tell you about it, throughout the lobular axis. And this, we think, might explain the broad proliferation of hepatocytes. And Kupffer cells, macrophages downregulate antigen presentation. And pericentral Kupffer cells and macrophages have also a very distinct zonal program related to matrix protein remodeling. That's it. If you're interested in some zonation resources, please check out our webpage. There's a zonation browser for human and mice. I want to thank my lab, Keren Bar-Halperi, my amazing staff scientist, did most of the work in the first part with Rom Shenhav. Shani Ben Moshe and Tamar Veg did the second part. Funding sources, we have wonderful collaborators on our liver projects, including Maria with a very exciting project. And I want to thank you for listening.
Great, thank you so much for this amazing talk. So we are receiving many questions. First question from Dr. Genshen-san. Hi, Genshen. Beautiful and fantastic talk. Can you please speculate if zonation is involved in location of HCC initiation?
Yeah, that's a really interesting question. I would imagine there might be different mutational processes related to low levels of oxygen, xenobiotic metabolism, portal processes, central processes. I think it will be very interesting, to profile the mutational signature of hepatocytes at different zones, and then maybe relate that to hepatocellular carcinoma. Maybe you can then infer what is this zonal cell of origin. We haven't done anything like that, but I do think it's an interesting direction.
Okay, so next question from Dr. Aparajita. Thank you for the wonderful talk. Did you by any chance look at cholestatic livers or any other long-term disorders which you expect graded loss in zonation, or any preference in functional loss in certain layer versus others?
Yeah, it's a very interesting question. So it's very interesting if these interface hepatocytes, for example, is this a general program of hepatocyte that need to reprogram as they move zones? So this is on our to-do list. We haven't done that, but to examine portal injuries and to see if indeed the newly incoming hepatocytes after portal hepatocytes die come from the mid-lobule or the central region, do they also sort of upregulate this sort of fetal program, maybe to erase epigenetically some of the zonal genes and basically modulate the epigenome. So we don't know, but I think it will be interesting to examine that in portal injuries. We haven't done that.
Okay, so due to the interest in time, I'll take one question, although we are receiving many questions, so Shalev can reach out to them later on. From Dr. Udayan Apte. Hi, Udayan. Fantastic, amazing talk. The mid-zonal hepatocytes were recently shown to have an added regenerative capacity. Did you see this in your studies with APAP, acetaminophen?
Yeah, so yeah, in APAP, yeah, this is true. There were some beautiful studies lately that did lineage tracing and showed that the mid-lobular hepatocyte are dividing preferentially. We've also seen very interestingly, liver is also a polyploid tissue, and we saw that polyploidy advances more quickly in the mid-lobule zones. In APAP, we actually see a slight tendency for proliferation in the central areas in these interface hepatocytes at the very first time point. So proliferation, basically 24 hours after damage, the tissue is in a shock, the hepatocytes don't do a lot. Then 48 hours later, they start dividing. And division starts very centrally, but very rapidly it becomes uniform. So actually 72 hours after damage, all of the hepatocytes proliferate identically, presumably to create this sort of mitotic pressure.
Thank you very much again for this amazing talk. So the rest of the question will be taken care of by Dr. Itzkovitz individually. Okay, so now we're moving on to the next speaker, another outstanding speaker, Dr. Meri Huch. She is currently a research group leader at Max Planck Institute of Molecular Cell Biology and Genetics. So she's going to talk about multicellular tissue-derived liver organoids help us understanding cellular interaction during tissue regeneration. Dr. Huch.
Thank you very much, Yasuko, for the introduction. And I would like to thank the organizers for, so Sangeeta, Wolfram and Irving for inviting me to be here today. It's a pleasure. I was supposed to be here, I was invited like 2 years ago, and I had to cancel because I was sick. I had a pneumonia, and finally I'm honored to be able to give a talk today. I'm gonna try to put that on presentation mode, and see if it works. And I hope you can see my slides.
I can see that.
Let's see if I can find my pointer, which I've lost at the moment. Oh, I'm not sure I can find my pointer. Okay, well, I'm not sure I can find my pointer. Can I do something about it? Oh, yes, here it is. So, and I'll put the laser so it's maybe easier to follow. Yes, so without further ado, I just want to give an outline of what I'm going to be talking to you today. And I may probably actually move that from here. I'm just going to make a little introduction on organoid cultures from adult tissue, and I'm going to go directly into try to give you 2 examples of how we've been using them to understand mechanisms of liver biology, in particular liver regeneration. And it was nicely introduced earlier by, why is not working? This is not moving, okay. No, by Takanori Takebe, who actually is the world leading expert on generating liver organoids from pluripotent stem cells. So I'm not going to be discussing that today, because what my lab has been working, and I was working already when I was a postdoc in Hans Clever's lab, and afterwards when I established my own lab in Cambridge, and now here when I moved to Max Planck, is we've been working on using and developing liver organoid culture systems from the tissue, either from the adult tissue, this is how we started, and also recently from embryonic liver tissue. And recently, the adult tissue-derived or the liver organoids have been classified according to the cell of origin. So I've redefined that slide to fit that standard of a consensus group, whereby now the organoids that we first developed already back in 2013 are now called cholangiocyte-derived organoids. And I'll be mostly talking about these ones. There's also hepatocyte-derived organoids from the mouse. That's been the work of Hans Clevers and Roel Nusse, and some human fetal liver organoid, hepatocyte organoids from Hans Clevers' lab. I'm going to be discussing about this, and I'm just going to show you some of our data on hepatoblast-derived organoids. But just going back to the first ones, as I said, back in 2013, we actually didn't know the cell of origin of our liver organoids, as we call them back then. But now we know that indeed the cell of origin is a dactyl differentiated cell, and that's going to be important for the rest of my talk. It's a dactyl differentiated cell that both either coming from the mouse, and then we actually afterwards improve the system to be able to expand also human cells. We can isolate these dactyl cells from the mouse of the human, put them in culture, they self-organize, they start dividing, they make these 3-dimensional structures that we call now cholangiocyte-derived organoids, and we can split them and propagate them over time in culture, with the advantage that they retain the capacity to differentiate into dactyl cells, but also if we change the media conditions, we can generate hepatocyte-like cells in culture. I call them hepatocyte-like because these are not bona-fide hepatocytes. They have some of the functions, but they are not they don't have the same amount of functionality as a phase isolated hepatocyte or as what we know now as hepatocyte organoids. But it showed these cultures show that property, and that I will be coming back later on, that from one single cell now you can generate both cell types. This was reminiscent to me to what happens in development. And very early on, when I started my lab, I realized that the question of hepatoblasts being bipotent had always been addressed at the population level, meaning that actually, indeed, it was true that hepatoblasts were bipotent. When you look at the population level, they can make both hepatocytes and dactyl cells. But the experiment of doing the same experiment in vivo with one single hepatoblast had not been done. Chris Hinde and Nicole Pryor in the lab took this approach of making lineage tracing from single level hepatoblast very early on, and they showed that they can generate hepatocytes and ductal cells. And then they did the same with they asked the question in vitro, can we isolate this now that we know they are truly bipotent single hepatoblast? Can they retain this bipotency in vitro? And indeed, when we do that experiment, when we take this single hepatoblast very early on the mouse embryo, they retain the capacity of generating both cell types, hepatocytes and ductal cells. And I'll be happy to go into details if you have questions. I just want to continue, but as I said, very early on, already in 2015, we had transferred the technology of the mouse liver organoids into the human setting. And when I also started my lab, Laura Bhatia, fantastic postdoc, she asked the question whether we could do the same with liver disease. And Laura Bhatia showed that you can take human liver cancer patient material and directly from this patient biopsies or patient resection material, she could culture it in ex vivo using our organoid technology, and she could propagate the 3 subtypes of liver cancer, both cholangiocarcinoma, hepatocellular carcinoma, as well as the mixed subtype. She showed that these cultures, despite you grow them in the same media conditions, you can recapitulate the transcriptional profile of the original tumor. So here I'm showing you how this is a transcriptional profile of the tumor compared to the organoids. And since this was published some years ago, I'm going slightly fast. If you have questions, I'm happy to go over at the end. But basically, there's a strong correlation on the transcriptional profile of the corresponding organoid to corresponding tissue of origin. And what it was more interesting is that not only retain the transcriptional profile and the genetic alterations, but also the histological organization. Here we have the patient material this is a cholangiocarcinoma in particular, the patient material that gave rise to these organoids that we grew ex vivo and for months in culture, and Laura afterwards transplanted in a mouse, and then they generated this similar architecture. As you can see, these glandular domains surrounding this lumen, all formed by these cancer cells. Actually, the pathologist could not distinguish when we presented to her blindly the tissue and the organoids. She could not distinguish which was which one. We also showed that this culture system is amenable for drug screening as a proof of concept. So as I said, we've been working extensively on these cholangiocyte-derived organoids, and one of the reasons is because we thought very early on that they could help us unveiling mechanisms of liver regeneration. And to this audience, I don't have to give much background about liver regeneration, but as we all know, at the cellular level, when the tissue is damaged, the damage mainly occurs on the epithelial cell types, either hepatocytes or ductal cells or both. And depending on the extent of the damage, or even in homeostasis, liver resection, acute injury, and depending on the extent of the damage, the cells, and we've seen it very nicely from Itzkovitz group, hepatocytes undergo self-replication, ductal cells undergo self-replication, and then this is how the tissue repopulates when there is certain types of damage, in particular in resection as well as in acute injury. For me, what was important is that this way of repairing is maintaining the cell identity, the cell fate. However, in liver disease, in human liver disease, patients come to the clinic with chronic liver disease. Most of them have fibrosis, cirrhosis, or cancer. And it's very well known that human chronic liver disease, it comes from a chronic damage to the tissue. And many groups, including especially Stuart Forbes recently, and many others in the past, they have also shown that when you perform chronic damage into the tissue, in particularly or very, very severe injury, there is a process by which both ductal cells or hepatocytes acquire the new fate. In other words, they de-differentiate, acquire a bipotent state, and now they are able not only to make themselves, but able to make the other epithelial cell type on the tissue. Basically, a ductal cell now is able not to make only a ductal cell, but also an hepatocyte. And this was a question that puzzled me and my lab for a long time. How is this mechanism of changing cell fate being regulated? And as I said very early on, we realized that maybe our ductal organoids are a good model to understand that and to try to understand mechanism on that direction. As I said, either you start from a healthy ductal cell or from a damaged liver and you isolate the ductal cells, or from a healthy liver and you isolate ductal cells, even one single cell will generate a clone that now is able to make both ductal cells and also hepatocyte-like cells in vitro. So we first asked the question, is that indeed true? Can we use this system to address a mechanistic understanding of how this acquisition of new fate happens in the liver? And for that, we needed to benchmark our culture system. To that point, it was only a correlation. So that was the work of Luigi Aloi and Michael Mackay in the lab, who took the approach of comparing the transcriptional profile of the ductal cells before and after damage in vivo by performing DDC damage model, and comparing the transcriptional changes that occur during this transition from being in homeostasis or before the cells proliferate at day 2 or after the cells start to proliferate at day 5, and comparing the in vivo versus the in vitro organoid formation, doing exactly the same approach, taking the transcriptional profile of the ductal cells at time 0 or at different time points during organoid formation before the cells proliferate, which is around 40 hours, and after that time point. And much to our surprise, what they saw is that there is a 70% overlap in the transcriptional changes that occur during the differentiated to a progenitor state in vivo, and the transcriptional changes that occur in vitro in the process of going from a differentiated ductal cells to organoid state. So 70% of these transcriptional changes are retained, especially before the cells divide. And most interesting to us, among the many things that occur, we found that 55% of known epigenetic regulators are dynamically changing during this transition. And we found that very interesting. We wanted to go into this detail, and just cutting long, very story short, we did some siRNA screening, and we came up with TET1 as one of a very interesting hit of our screen. And that one was very interesting because that one is involved in the transition of a methyl cytosine, 5-methylcytosine to 5-hydroxymethyl cytosine. And this molecular change, actually this is only the change of a methyl group for an hydroxymethyl group in the cytosine, transforms this genomic region from being a repressive region that cannot transcribe to a permissive region for transcription. It's permissive, it means it facilitates gene expression. You still require all the transcriptional machinery to bind to the loci and activate the transcription. It was also interesting for us because we know it was very well known that to induce pluripotency, that proteins are very important and are required for this reprogramming. So we wanted to focus on that protein. And cutting long story short, that was recently published, we indeed showed that TET1 is very important for the transition from a differentiated to bipotent state in vitro in organoids, because when you deplete TET1 after isolating ductal cells, you deplete TET1 in vitro using a conditional allele by adding just tamoxifen into the media, organoids cannot be formed. But similarly, if you do that experiment in vivo, in the mouse model that was developed by Stuart Forbes, where you combine the P21 overexpression with DDC damage for ductal reaction, and in this damage model, you all know that ductal cells are bipotent and are capable of generating both hepatocytes and ductal cells. What we did in that experiment is we took PROM1 cream reporter mice, and we deleted specifically TET1 on the ductal compartment using this reporter, and crossed it at the same time with a rosa ZS green to visualize the mutant cells. And therefore, those ductal cells are green, and at the same time, if we use the mutant floxed allele, then the green cells are also depleted on TET1. And what we observed when we induced this damage model from Stuart Forbes is that TET1 mutants, ductal-specific TET1 mutants, cannot generate hepatocyte clones. Neither the ductal clones are also very tiny, with a very small clone size compared to wild-type clones that can generate up to 200 cells of a clone. What is the molecular mechanism? What we found is that actually TET1 is involved in, as I said, is involved in this transition of a methyl cytosine to an hydroxymethyl cytosine. And actually TAT1 is depositing hydroxymethyl cytosine marks in many, many genes, most of them involved in regeneration, including mTOR, including YAP-EPO pathway. And we are pursuing some of these hits. And I'll be happy to go into details if you are interested. At the moment, as a summary, our working model is that when you are in homeostasis and this regenerating signature is switched off, then upon damage and before the cell divides, there is a peak of TAT1 expression. This activates and it starts depositing hydroxymethylation, changing the methyl cytosine for hydroxymethyl cytosine in critical genes involved in proliferation, but also involved in regenerative pathways like YAP and MAP kinase. And this facilitates the cell to go from the differentiated to activate proliferation. So in the second part of the talk, I want to talk about the microenvironment. And actually, Professor Itzkovitz gave us a very, very nice introduction. So I will go very straightforward to the point. But all what I've been talking is just about the epithelium, the ductal cells in that case. But we know that these cells do not work alone, and there is many other supporting cells, mesenchymal, endothelium, immune cells. Very early on, Lucia Cordero in the lab, talented PhD student, in collaboration with Anna Doubach in the lab, they realized that actually next to the ductal epithelium, as we have here, there is this mesenchymal population that here we have it marked by PDGFR-alpha, and also desmin, that are kind of embracing the ductal epithelium. I think we can see it better here. We see that this mesenchymal cell in red is embracing the ductal epithelium and is indeed expressing also the other marker SCA1. And that allowed us this will allow us to do the following experiments. The first observation, though, that Lucia had is that these cells change their numbers during the damage regeneration response. Basically, Lucia and Anna spent a lot of time quantifying the number of ductal cells and number of neighboring mesenchymal cells next to it during the different phases of the damage repair. And what they saw is actually the ductal cells, here they did an homeostasis and upon damage and during recovery after the cells have regenerated, or the tissue is starting to regenerate. And what they saw is that ductal cells proliferate first. But this particular mesenchymal population, actually, it doesn't proliferate after you remove the drug from the toxin from the mouse. And then they catch up and they recover their numbers. And they go back to homeostatic at the end. This means that actually these cells are in contact. And what it happens is that actually during the damage response, they lose contact. And they regain the contacts during the during the regenerative phase, in the recovery phase. In other words, if you look that into the ratio between the two populations, and that number is going to be important afterwards, what you see is that the ratio between the two populations in homeostasis is 0.3 mesenchymal cells per 1 ductal cell. This drops to 0.1 during the damage peak, and it regains at 0.5 to 1 during the recovery phase, terminating in the same values, more or less, as it was starting. So we wanted to understand, we had the hypothesis that this cell contact was an important mechanism to drive the regenerative process. But we had no way to understand that in vivo. It would need to do in vivo live imaging. And we turned to our organoids, but our organoids had the problem, as I said, that they are only epithelia. So we had to devise a strategy to be able to recapitulate this structure of the ductal mesenchymal, portal mesenchyme in vitro in a dish. And after quite some iterations, Lucia and Anna figure out a way to combine the ductal cells now in green with this mesenchymal SCA1 PDGFR-alpha positive population. And as you can see, this is one example, but in a high proportion of the structures, we always see the mesenchymal cells surrounding the ductal epithelium. When we obtained these first structures, we actually found a very interesting observation. We started filming them, and I will need you to focus on the green arrowhead and the yellow arrowhead. They are pointing to two different structures. The yellow is pointing to a structure that is touched by one mesenchymal cell in red. The green arrowhead is pointing to a structure that is close to, but not touched by, a mesenchymal cell in red. And the green is marking the ductal epithelium. And we performed that experiment in the absence of any growth factor in the media. And what we see is that while the contacted organoid collapses, the non-contacted organoid grows. So that was kind of telling us that these cells have a dichotomy. They are totally opposite phenotype according to whether there is a cell-cell contact or whether there is no cell-cell contact. And we wanted to address that question. So the first thing we did was to separate the two populations. And we did that in several ways. Just showing you one. Lucia and Anna grow mesenchymal cells, and they generated conditioned media from these ones that they used to put on top of as a media to grow ductal cells in the absence of any growth factor in the media. When we don't have growth factors in the media, nothing grows, basically. When we use the conditioned media, actually, organoids grow, and they grow to the same extent as when we add the complete cocktail. And now we know that this population is expressing some key factors, for instance, HGF, FGF7, or are responding. Then what about the other phenotype, the cell-cell contact? And I'm getting to the end of my talk. The cell-cell contact was suggestive of a totally opposite phenotype, that actually contact was inhibiting. But to address that question, we needed to be able to play around with the ratio of the two populations. And that's what actually Lucia and Anna did. And what they found is that there is a sweet spot. When you have 0.1 mesenchymal cells per one ductal cell, actually the ductal organoids can grow very nicely to the same organoid efficiency as when we put the full media, almost. Not entirely, but almost. But that drops very quickly, just with two more cell contacts, 0.3 to 1. And up to the point that this organoid formation is totally abolished when you get to two mesenchymal cells per one ductal cell. That was very interesting, but it was even more interesting to see that this cannot be rescued when even we dump all the growth factor cocktail into the medium. The cell-cell contact inhibition is much stronger than any growth factors that we add into the medium. We found that this is indeed because there's an increase in proliferation and a decrease at the low ratios and an increase in apoptosis at the high ratios. What is the mechanism of this cell-cell contact? We think there is many mechanisms, not only one. I'm going to just show you one, and we are studying still some of others. And here I'm just focusing on a chemical mechanism, biochemical mechanism. We found that was easy because we found that ductal cells were expressing Notch receptors, and the mesenchymal cells were expressing the Notch ligands. So we decided to inhibit Notch receptors on the ductal epithelia only, and then combined these Notch-inhibited ductal cells with the mesenchymal cells. And indeed, we could rescue totally the phenotype. I'm showing you here the Notch inhibition with a chemical inhibitor, but the same is true if we do siRNA. And we indeed found, using confocal microscopy, indeed, that these mesenchymal cells, when a mesenchymal cell touches a ductal cell here in white, it activates HES1 GFP reporter, which is the reporter for Notch signaling. And that gives me the working model that we have at the moment, whereby we think that the homeostasis is maintained in a 3:10 ratio, or 0.3 to 1. During damage, this ratio drops because the number of cell-cell contacts drops. And this allows proliferation of the ductal epithelium, but mesenchymal cells catch up afterwards during the termination phase, and they restore back homeostasis. And that brings me to my summary slide, where I've shown you in the first part that we think that to go from a differentiated to a bipotent proliferative state, there is a massive epigenetic reprogramming. I told you that more than 3,000 genes acquire de novo hydroxymethylation marks at the promoter region. And this is a massive, and yet some of these genes are specific and they belong to a known classic regeneration pathways important for liver regeneration. I've also shown you that the number of cell-cell contacts or number of contacts matter. And I think this is very timely with the COVID times, but it's actually true. What we see is that the number of cell-cell contacts between the neighboring portal mesenchyme and the ductal epithelium dictates the behavior of the ductal epithelium. And that brings me to my most important slide, the acknowledgements. I've already cited some of the people along the lines. Most important work of Michael Mackay, Luigi Aloia on that one project, and Anna Dobach and Lucia Cordero-Espinoza on the mesenchymal project. Of course, I would like to thank all my collaborators and our funding. I'll be happy to address your questions. Thank you.
Thank you very much, Meri, for this innovative and beautiful work. we are receiving many questions now, so I'm going to start with this interesting question from Dr. Tobias Rabe. What happens when you overexpress TET1?
Oh, that's very good. So if you overexpress that one in a wild type, we did it actually, we overexpressed that one in wild. If you overexpress that one in the mutant, you can rescue the phenotype. If you overexpress that one in a wild type, actually ductal cells don't like it and eventually they die. The organoid culture is gonna die, which doesn't happen. So with the time, you lose the culture. So we think that actually the TET1 protein levels are very tightly regulated. We don't know how, but the TET1 protein levels must be very tightly regulated because even a 1.45-fold overexpression over the normal levels actually kills the ductal organoids.
Okay, so the next question from Dr. Apajita. Thank you, Meri, for this lovely talk. It is super curious that these mesenchymal cells cause the ductal colony to dissociate in culture. Do you see something similar in vivo? Would depletion of these SCA1 positive cells leads to large ductal niche?
Actually, this is an experiment we really loved. We would have loved to do because of COVID reasons, and it was not possible at the moment. We're now starting to breed animals to do this particular experiment in vivo. In vitro, I want to emphasize, maybe I was going too fast, is actually this depletion of the ductal colony or the ductal organoid happens only when you have the 1-to-1, so a particular ratio over the non-homeostatic ratio. When you go to the 1-to-1 ratio, when you keep the homeostatic ratio, you can keep the structure nicely. When you have a low ratio, actually you allow the proliferation.
Okay, so we'll take one more question before moving on to the next session. This is from Dr. Konstantin Zoanas. Could you share your thought on comparing or contrasting the context where bipotent cells versus zone 2 hepatocyte contribute to regeneration? Different reports have implicated each, and I'd be curious for your thought on integrating these observations phenomena.
I see. I think it really depends on the type of damage. That's my understanding from the literature. So for instance, the model that Professor Itzkovitz just showed us is that it's very similar probably to the carbon TET model, where if you kill a zone, so pericentral hepatocytes, then but you still have preservation of the other zone, of the other hepatocytes are relatively healthy, so they can recover the tissue. The model where you have senescence, so the model of Stewart-Forts, for instance, when you get this bipotency, in this model, what you do is you induce senescence to the hepatocytes. So basically, on those models where the tissue is not allowed to induce proliferation itself, because in that case, for instance, you have senescent hepatocytes, they cannot enter the cell cycle. When you induce damage, then the tissue has to decide either to regenerate from another cell type or to die. So I think actually the ductal cells have this capacity to be facultative. And then in that particular model where you have senescence of hepatocytes of massive severe injury or very chronic injury has been shown by the group of Ding and also by the group of Paul Monga. In these models where you have massive or hepatocytes cannot regenerate, then the ductal cells acquire this bipotent state. I hope I could answer that. And maybe someone else on the audience or panelists can answer as well, or maybe better.
Thank you. Before we move on to the next session, next event in this session, I'd like to take this opportunity to thank Dr. Huch and Dr. Itzkovitz for these fantastic talks and also interesting questions from the audience. So now we move on to the our last event of this session, which is the Fireside Chat. Now, in recognition of the 30th anniversary of Arias Symposium, Dr. Bhatia and Goessling will chat with Dr. Arias and reflect together on our field and the history of this series of events. Sangeeta and Wolfram, it's all yours.
Thanks, Yasuko. Hi, Win.
Hello, my, isn't this fantastic?
It is fantastic. It's great to see you. And we are so thrilled with the science and the community and the conversation already today. Just to kick things off, I thought I would get started and then we can just chat. But I was kind of reflecting on your original vision for building bridges between basic science and clinical medicine. And, you know, we were, in putting together the program today, we were going through some of the older programs just to kind of remember what had been presented previously. And it's just really striking to see, you know, a program in the 90s talking about new ways to knock things out genetically, and a program talking about HCV, viral replication. And, you know, today we're talking about stem cells. And so the science has just moved, and there's so many breakthroughs now coming, you know, for patients. So, you know, I'm just curious how you see it over the course of your career. How are the advances changing that you note, and what do you think we'll be talking about in the next 10 years?
I don't know. I just want to be here to hear it. No, I think what you said, Sangeeta, is correct. Absolutely. The composition of this symposium through the years really has been the same as the explosive pattern of discovery and advances in basic biological sciences and also in engineering. And to me, the exciting part of it is that much of the topics and conversations, the presentations that took place in all of the meetings, then about 4 or 5 years later, you see that they have some really direct impact on liver-related research and on ultimately liver disease and even development of therapies, vaccines, drugs, small molecules, and so forth. And I think the point of it is that there's this gigantic gap which gets bigger and bigger and bigger between what happens in science and it's bridging into what you might call clinical medicine. I think of it as the scientific advances are sort of on a logarithmic scale, and their application or entry into medical into medicine is more arithmetic. So the gap gets bigger and bigger and bigger. And there are many reasons why that happens. And there have been many, many efforts to try and do many different things to sort of bridge that gap, because I really think that's the major challenge confronting medicine and human health. There's such a vast amount of information and exciting research, and it takes a long time before much of it gets into an area that affects human health. And there, as I said, there are many reasons for it, but the symposium is just one small step in a big, complicated process of how you deal with bridging this gap. At the time the symposium started, to my knowledge, the only thing that was remotely comparable in the world were the Falk symposia, which were being held, I think, every other year which really dealt with basic science. There wasn't that much going on in many other areas you might expect. So in the beginning, this was unique. And it still is unique, but there are many other avenues that are now developed, including contract research and pharmaceutical companies and all. But the huge problem is where do young people stand in terms of this kind of career? And that's a very complicated issue, particularly for MDs.
Yeah. Well, one thing I think that's been shown a light on this is actually COVID. And I know, Wolfram, you have some thoughts on how this maybe is a framework for thinking about the future.
Yeah, Wynn, I wanted to sort of ask you, because I actually feel during COVID, as challenging as it has been, I think, on a societal and a global, on an economic, on any level, on a personal, on a mental health level, I think we have seen that science and clinical application does not have to lag behind. You know, I mean, we were presented, the global population, the world was presented with a clinical problem, and science together with the clinicians solved it, right? The clinicians solved new ways to intubate patients before we had a vaccine or an antiviral, and now we have vaccines and antivirals. So do you think, you know, when we think about the gap and the bridges that we want to build, and I know when Sangeeta and I started this, and our approach was really try to put this together, right? And so we invited industry speakers and you know, research groups there who actually, you know, the goal is to have something at the bedside, give something in the hand to the clinicians. Contrast that with some of the basic and fundamental work we've heard today that is just as important. But I think even if you just look at the topics of these first two sessions, going, you know, from infectious disease to thinking about regeneration to genetic liver disease, right? These are all real problems. NASH, forgot to say NASH. Everyone has NASH, right? These are the real problems that our patients have, and I feel we're really focusing on that. So how do you think we can sort of tighten that, or, you know, either build more bridges or faster bridges or bigger bridges?
Well, you know, that takes much smarter people than I am, or I suspect all of us are. I mean, you put your finger on one of the great tragedies, which is that amazing discoveries. I'm thinking particularly of hepatitis C treatment and hepatitis B vaccination. I mean, these are really incredible, affecting millions and millions of people. Yet the fact is that the public health systems throughout the world, and I guess it's the political systems as well, take a back seat to a great degree in trying to bring these very effective treatments to the public, not just in the so-called developed world, but throughout the world. So that's another part of a different gap. And that's where you go from science to medicine to public health. And I don't know how one, I have no real comments about that, because I just wish it were different. I think, honestly, I think it's unconscionable that our global societies cannot take such effective treatments for these two lethal diseases, which are both major causes of all kinds of things, transplantation, cancer, so forth. I think it's terrible that these vaccines and drugs can't be made available to effectively control this. And it applies too to other diseases, but most dramatically to the HBV and HCV situation. I don't know. What are your thoughts?
Well, I think, one thing, Wynn, that you've shown us is that I think an important part of all of these bridges is just having a conversation and forcing people to be in the same room. This symposium is a virtual attempt at that. And I thought maybe you could tell some people about the class that you started, the Demystifying Medicine class. I think it's a great example of how to have conversations, whether it's policy or medicine. Before you do, I'll just say to the crowd that Wynn obviously is a node and has all this historical knowledge. And even today, if I find something in the literature that I have a question about, like I was just reading about ectopic livers that have been found at autopsy, I emailed Wynn, have you ever seen this? And, within 2 days, like we had a conversation going. So I think it's about conversation and having avenues for those. So maybe Wynn, you could talk about your course.
Yeah, well, I think you hit the point. I think that today, so much of the rapidly developing science, particularly when you get into the informatics area and so forth, you're bringing in huge disciplines that most physicians, for example, even those interested in science, you cannot fully master. But what you can do is to know enough so as to be able to communicate. And that's, I think, is really the key thing. To be able to learn somewhat of a common language between, I think that at the present time in science and medicine, it's like different parts of a pizza pie. We all speak our same languages, and what the other person speaks is something we don't understand, so we stay away with. Well, the Demystifying Medicine course at the NIH in January begins its 20th year. And it started out with the idea that there are 4,000 or 5,000 PhD, mainly postdocs at the NIH, and close to 800 medical students spending a year doing research. And then there's the hospital and the whole clinical arena. And there's very little communication. And so the idea was to somehow bring all these people together into the same room and discuss, hear what's going on. So we reinvented the wheel. The wheel was old-fashioned grand medical rounds. So there are many topics, about 20 in each year. And when possible, we start with a live patient who very briefly describes what his or her illness is like in layman terms. And then a physician, who's usually a physician scientist, sort of talks to this heterogeneous audience and explains sort of what the disease is about, diagnosis, treatment, and why that person is, for example, doing research at the NIH. And then a basic scientist who's really at the crest of the wave of where the big questions are. And all of this is together and people communicate in the same room. Now that we're virtual, we are global, and we're getting up to 3,000 to 4,000 people watching and taking part in electronic connections. You might find it interesting too. You can just Google Demystifying Medicine and you'll see the upcoming program for 2000. Covers a wide range of medicine and other areas like science in space, science at the bottom of the ocean. So the whole point is, if people can hear and what excites individuals as physicians, as PhDs and whatnot, that's the whole idea, to get people to be excited and to listen and learn that it's the big issues are challenging, but they're not that impossible to communicate. And the last part of it is serendipity. And that is, if you sit down with people of different disciplines and background and just talk to them about what you do and what they do, it's amazing how good things happen. So I don't know, maybe that's oversimplistic, but I think the purpose of the Demystifying Medicine course, which by the way now is replicated to a great degree in 38 different countries and a large number of North American institutions. And all of the programs for 20 years are archived and readily available. And you don't have to watch it at the time the program is going on. You can just Google it and hear what it is. At any rate, I think things like that help because they get people excited. And once you get excited about something, that's the first step in motivation.
Yeah, I totally agree. I think you know, that example of having a patient in your class, I think those of us have been in med school. I mean, you always remember that. You always remember the first patient that you met with a disease that stays with you as a real sort of spotlight for the real problem. So I think that's right. I wonder, one thing that strikes me, and it may strike you more or less, over my career working on the liver, which is not as long as yours, I feel kind of like we're having a moment. I feel like, you know, when I entered into this field, there were very few therapies, right? There was, first there was the HPV vaccine, but it was pre-antivirals for HCV. There was no interest in NASH. There wasn't even a name for NASH. There was no CRISPR. There was no AAV gene therapy. There was no RNAi. There was no iPS-derived hepatocytes. So it feels like if you sort of fast forward 20 years from where I started, like we found ourselves in the middle of this really exciting convergence of technologies and opportunities for patients. And I wonder if it feels like that for you, or if it comes and goes, or how does it feel for you, like looking back at the changes?
I just wish I were 50 or 60 years younger so I could be part of it, because I really believe that the next 5 years, 10 years are going to see really amazing advances in our understanding of basic biological processes and how they apply to disease and health, not only of the liver, but in medicine in general. And I think that sometimes we're too short-sighted in thinking about what the future is going to hold. I think this is one of the most exciting times that a young person can become involved in research, whether it be more in a laboratory or at a patient's bedside. I don't know, one of my old professors once said that whether something is basic or clinical is not determined solely by where the research takes place, but it's really the importance of the question. And I've often found that useful to think about, because I think a lot of research that goes on now, particularly in the clinical area, I'm not so sure that the question, the important question, you know, is being addressed. But at any rate, that's
so, So Wayne, let me follow up on that. From as you watch what young people do in the field, what would be your advice for young scientists, to with regard to not just, identifying the big questions? That's easy to say, right? But it's very hard to do. But both Sangeeta and I have obviously trainees in our labs, and we see them both grow and be successful, but also being presented with a multitude of options these days, both within academia and in biotech and pharma. And clearly they want to have and make an impact. And from your perspective, with all this this experience, what would your advice be for them?
Yeah. I don't know. It's easy to give advice, but I'm not so sure that it's always helpful. When I first got involved in research, believe it or not, it was in the late 1950s, and in that era, what was rate limiting was ideas and not money. We were reasonably smart and had done reasonably, good work, and wrote a reasonable proposal. You could be quite sure that you were going to get funded. And this whole, the explosion in science then spilled over, so everything becomes much more competitive and all, and it's difficult. But if you're really turned on by the excitement of the big questions that you're asking and seeing things, there's probably nothing more exciting than suddenly realizing, my God, I may be the first person on earth who ever saw this thing and began to think about what it means. It's a long shot. And there are no easy answers to it. I think there was a hue and cry about maybe 30 odd years ago about the decline of the physician scientist. And at that time, various methods, the MD, PhD programs and others were created. And they've been helpful. And I think it's important. I like the bridge analogy because a bridge is made up of many different parts. And all of these problems have many different approaches. There's no single one that's going to solve everything. And we just have to work hard to try and be imaginative and convince our institutions and all, and the pharmaceutical companies and others, foundations, to support these young people and to get them started.
Wayne, one thing that I've observed about you, and I wonder if you share this perspective, is that you have a pretty unique balance of really expansive knowledge base. So like I remember when I first talked to you about bipotential cells, you said to me, the mollusk has a hepatopancreas. And, you just know, it seems everything. I was like, oh, we're really interested in heat generation in the liver. And you said, like, oh, somebody stuck a thermocouple in the liver after the fed state in, 1955. You remember every experiment. So I think that's amazing. But you also have like a very specific, pretty focused view based on the science that you're interested in the moment. And so for example, you and I interacted a bunch around AMPK and cell polarity. And I felt like every conversation I had with you was going to end up at cell polarity. And I love that because I think it's a combination of like a specialized lens and then like a broad, expansive knowledge base. Did you do that deliberately, or that's just how you're just curious and that's how you think about the world?
A lot of it is serendipity. I was very fortunate when I was, 14 years old, 15. I worked in a laboratory in New York, founded by IBM after the World's Fair in 1939. And they put together a group of about 40 individuals. I remember there were only 5 women. And we were selected idea. Now, I don't think that anybody, those research ideas, to my knowledge, none of them particularly blossomed. But the people blossomed because they were treated as mature adults. Lots of consultation with experts in the field. There were 4 Nobel laureates came out of that group, and God knows about, I think, 20-odd members of the National Academy. So that was the orientation of, and you meet people like that, you become friends with them almost for life. And I think that was purely chance. And a lot of things happen by chance, but they only pan out if you take advantage of it. If you're too busy doing your own thing and you don't talk to somebody who may be sitting next to you who's, I don't know, an immunologist or a plant person or, you know, whatnot, there's an excitement in the bigger area of science and biology. And if you're in an academic institution, you have a great opportunity. You don't have to go spend vast amounts of time. But I've always found that listening to what other people are doing and thinking, it's profoundly influenced ultimately what we did in doing our own research. Knowing both of you has influenced very much, you know, much of our thinking and what we do. And I mean, hearing talks like today, just amazing. I mean, Shalev's talk and Meri. I mean, how can you not be excited? You don't have to be the expert, but you certainly can be excited and think of it in terms of whatever is the big question that you're interested in. And I do think you have to have a big question, not a little one.
Yeah, when I totally agree. I wanted to ask you, or you know, and maybe this is how we can end, to give us your thoughts on, as you mentioned, you know, the importance of both, you know, men and women doing science, and highlighting who you had interacted with in the past. I will date myself. You know, I went to, my first symposium was in 1993. I think that was number 2 or so, as a student. And they were all men, both at the speakers and in the audience. And you know, I think both Sangeeta and I, we made a pact when we started the symposium to have actually equal gender distribution, not just like a token woman here or there. And if you look at the last list of speakers, we are at 50% across the years, and we haven't run out, you know, of talent in any way. And I think if there is a change in these last 30 years, that this is that this aspect of who does the science, because it actually has to reflect, you know, the people with the disease as well. I think that's important. And so I know you, as you just said, right, have been really a strong advocate for that. And maybe we can end with those thoughts around that topic by you.
Well, you know, I'm glad you mentioned that. I mean, it's so impressive to see the array of speakers and the topics and really the beauty of the science that they presented. And as it happens, wonderfully, a large group of talented young women. That's long overdue. And I think that there's no question. You don't, this science is, good science is not just done by one gender or another. But I would point out something, Wolfram, a couple of years ago when we had it on site at the Broad, several of the speakers were from undergraduate colleges, biology departments. And when I spoke with many of them afterwards, they were just blown away by the fact that now they were mixing it up with people who were in different areas and who had different interests, particularly as regard to medicine or regard to health or regard to science. And I think this is a very important point that I hear, and that is that we have this artificial barrier. I don't know where it came from. You don't teach anything about medicine until somebody gets to medical school. God forbid you should teach something about medicine, not general health, in high school. Why did that came about probably in Victorian times, but there's no reason why it should be that way. Many of our high school kids, I know my grandchildren could draw DNA while I was still struggling to try to understand where the bases were. And they were, in public elementary, not elementary, but high school. So by reaching out into education at undergraduate colleges, even in high schools, and particularly in high schools, perhaps specializing in science, I guess. But we could do a lot more to encourage and support those young people. The great ideas don't all come from everybody who's just been given everything. And these are huge challenges, more bridges.
That's a great note to end on. When we need more bridges going forward, we need younger exposure, more gender and racial diversity, all talent bringing to bear on this conversation. Thank you so much for spending the fireside chat time with us. We have actually a little slideshow for those of you who want to keep your session live. But during the break, the science will resume at 1:30, but there's some lovely historical pictures and nice moments to share for those of you who are curious. So thank you, Wynn, for spending time with us, and thank you, Wolfram, for the conversation.
Thank you. Look forward to being with you.
Good afternoon, everyone. I hope you had a great break and a great lunch. And as we begin our session 3, our afternoon session, I wanted to introduce our final corporate sponsor. This is CVS, and they are one of our bronze sponsors for this program. Thank you.
When you hear the word healthy, it always feels a little out of reach. That's because the way we're thinking about it is all wrong. So we made a healthier song. For some folks, it's like baby steps. Maybe it's a jump or eating something green or taking mom to get that vaccine. Healthier means bringing stuff to the folks that really need it. Like Millie's meds straight to her door or carry it home, believe it. Sometimes it's healthier to laugh, other times it's healthier to cry. We'll work through it together. When it works for you, drop on by. Cause healthier happens easy when you just give people access. For Bob, it meant admitting that he needed reading glasses. Healthier comes in all these different shapes and sizes, and ages and races and faces and guises. Caring for them all means we're doing healthier right. So let's do it all together, people. Cause this is what healthier looks like.
So good afternoon, everyone, and welcome back. My name is Kristen Knaus, and I'm thrilled to be chairing the 3rd session of today's fantastic symposium. And so we'll begin with a talk from David Cohen at Brigham and Women's, and he'll be telling us about leveraging energy expenditure in the management of non-alcoholic fatty liver disease. So take it away, David.
Thanks, Kristen, and thanks to Wolfram and Sangeeta for the opportunity to speak. After the fireside chat, can you see my slides? Okay. I took out a few slides because I wanted to spend a minute appreciating Win from a personal perspective that I hope gives you a sense of what he's done for so many of us in the field. I think I'm the only speaker on this program who also spoke at the first symposium 30 years ago. I was a GI fellow and I was asked to make some comments at the dinner the night before, the dinner that evening, on what it meant to have an American Liver Foundation postdoctoral fellowship, which those of you who know that is a little salary supplement that made actually a huge difference in my life. But that connection with Win was even far later than the personal one that I had with him, which goes back, I hate to say, a little over 60 years, when Win and my father were successive chief residents under Irving London, who many of you know started the HST program at Harvard and MIT. And so our families were connected for many years. Years later, I had the privilege of being an HST student and being mentored by Irving London, who, when I was trying to figure out what to do with my career, he said, why don't you go talk to Win? And so that discussion with Win led to a job in the liver center at the Albert Einstein College of Medicine, which really changed my career and ultimately led to a return trip to Boston, directing the HST program, organizing this symposium with Win, which was, I have to say, among the most fun that I had in hepatology. And then ultimately editing now the journal that he founded. And so with that, and as my term as editor is just wrapping up, I will tell you that perhaps I can deflect some of the blame for rejecting at least some of the papers of those who are listening, because so many the journal is so competitive to get a paper in, that really speaks to Win's success in so many ways, in it really helping to establish the AASLD, founding editor of the journal, and all the amazing contributions that he's had over time. So when this has been a lot of fun. The difference is my hair is not gray, is now gray. Win is exactly the same age. He's got the sense of humor, the curiosity. That's exactly the same as the first time I can remember interacting with him. So this is really a privilege. And now I'll talk some science. So as comment has been made, fatty liver disease is really the premier liver disease now of our time, with about 25% of the world population being affected by too much fat in the liver. And we don't completely, there's a lot we don't understand about this disease, but what we do understand is that it's a disease of overnutrition. It's associated with the metabolic syndrome. It's associated with obesity, dyslipidemia, and diabetes. And although we don't have specific molecular mechanisms that we can drill down and identify one, this is all really associated somehow with an insulin resistant state that is initiated by the consumption of too many calories. So if we think about it in that context, it becomes a somewhat simple problem in the sense that energy intake exceeds energy expenditure. And so the management of this should be simple. It should be, we can do one of two things. We can decrease energy intake, or we can increase energy expenditure. Decreasing energy intake, for those of you and me who have tried this in the clinic, tends not to be very successful. It is the foundation of a multi-billion-dollar diet industry that really has had little, if any, impact on the trajectory of this disease. The other possibilities are increasing energy expenditure, and certainly this can be accomplished by exercise. But again, prescribing exercise in the clinic tends not to be a fruitful strategy, at least towards getting to the goal of alleviating fatty liver disease. So the question is, are there other ways to leverage energy expenditure? And one of them we've been thinking about for some years in the lab is the body's response to ambient temperature, because that really leverages issues of energy expenditure and mechanisms of energy expenditure. So what I've shown here is a plot of, in a mouse, if we measure energy expenditure as a function of ambient temperature, we can see that there's a rather striking relationship. At 30 degrees, the mouse is thermoneutral. It does not need to create heat in order to maintain its core body temperature. But you can see as we decrease the ambient temperature towards what's comfortable for us, a somewhat cooler temperature, and in an experimental cold room in the high 30s, you can see that there's a tripling of energy expenditure that goes along with that. So the thinking is, if we can drill down and understand these mechanisms that control energy expenditure, and the control is quite tight, each of these data points is the average and in standard error of 13 measurements. So you can see, you can't even see the error bars. This is a very precise and controlled mechanism for expending energy. So what causes that? Well, at least in a mouse, and now we understand in humans, we have a thermogenic tissue known as brown adipose tissue. In the mouse, this constitutes about 1% of body weight with a very high metabolic activity, so that in the cold room, up to 80% or so of calories consumed by the mouse are consumed in the interest of creating heat to maintain core body temperature. This is a tissue that's essentially mitochondria as heat engines, and it's characterized by the expression of a protein called uncoupling protein 1. So cold ambient temperatures do two things. They activate thermogenesis in brown adipose tissue, and they also upregulate a suite of genes that supports this thermogenic response. And it's a very strong and tight relationship. So what is brown adipose tissue? Well, that's shown here histologically on the left. We can see that at 22 degrees C, which is our room temperature, there are lipid droplets and some other material, but mostly lots of small lipid droplets. And if you cool the mouse down to 4 degrees, these lipid droplets get much smaller. And if you look on the right at the electron micrograph, you can see that the tissue is lots of mitochondria. They tend to be circular in appearance, at least on cross-section, and lots of lipid droplets. And as I'll show you, the mechanism really that generates most of this heat is that these lipid droplets, which are triglycerides as the lipid, are hydrolyzed, and the fatty acids are translocated into mitochondria for combustion. So the way that looks is that when there is cold exposure, centrally that is sensed, peripherally norepinephrine is released by neurons. This activates beta-3 adrenergic receptors on the surface of brown adipocytes, leading to pKa phosphorylation and downstream events that result in hydrolysis of lipid droplets, releasing fatty acids. The fatty acids are then activated, transported into mitochondria, oxidized, creating a proton gradient where there is this additional protein called uncoupling protein 1 that collapses this gradient, and we get a futile cycle that creates heat. So this is at least one of the major mechanisms for creating heat in brown adipose tissue. And a key enzyme of this is an enzyme that commits the fatty acids that are coming out of lipid droplets to combustion in mitochondria, and it's called ACSL1. And this enzyme, ACSL1, is a long-chain fatty acyl-CoA synthetase, which activates fatty acids. And if you think about fatty acid metabolism, when fatty acids are taken up into cells, They're esterified to CoA by a high energy thioester bond. And this enables fatty acids to be oxidized, taken up and oxidized as fuels. It's also the same event that allows them to be incorporated into complex lipids, such as triglycerides. So what about this thermogenic program that supports thermogenesis? Well, we know that from the work from the Spiegelman lab and many others, that the cold ambient temperatures that stimulate activity in brown adipose tissue also upregulate critical transcription factors like PPAR gamma, PPAR alpha, et cetera, to promote the expression of genes that support this thermogenic response. So about 20 years ago, in an unbiased screen, an early unbiased screen of genes that are upregulated in response to cold ambient temperature, Sean Adams and his group on the West Coast showed that the most upregulated gene under the conditions that he studied, which is a specific way of moving the animal from thermoneutrality into the cold, a gene called thioesterase superfamily member 1 was the most upregulated gene under their observation. And this is a gene we got interested in for a variety of reasons, but there really was nothing known about it. And this wasn't particularly well pursued at the time. It was just an observation by a group that was looking for genes that might become targets for obesity therapies. And considering that this gene was so upregulated, the assumption was that it was involved in creating heat. And we ultimately reproduced this. This is mRNA going up 100-fold. Depending on the rate of cooling, it can go up 1,000-fold or even further. So what is this gene? This is a gene that belongs to two protein families. One is an acyl-CoA thioesterase. The other is a steroidogenic acute regulatory transfer-related domain-containing protein. It's got some synonyms. The structure is down at the bottom. There are two enzymatic thioesterase domains, N-terminal and a C-terminal lipid-binding start domain. And really nothing was known about this. It was classified into this enzyme family. And this enzyme family are a family of genes or enzymes that deactivate fatty acids. So they take fatty acyl-CoAs and convert them to free fatty acids plus CoA. So this is the opposite of the reaction that I just showed you that creates heat by thermogenesis in brown adipose tissue. So again, these enzyme family was described but not characterized. So at the time, we turned our attention to this gene. There was ideas that it might regulate fatty acid metabolism. It could be involved in recycling CoA for energy metabolism in the cell, or even in some cells, generating free fatty acids for conversion to inflammatory mediators, such as cyclooxygenase and prostaglandin formation. So the hypothesis then was that this enzyme promotes energy expenditure. So the first thing we did, because it was very enriched in brown adipose tissue and very little in other tissues, was just to knock it out and see what happened. And what happened was quite surprising. So these are the body weight of mice on the vertical axis, their age on the horizontal axis. Yellow and green mice were fed a chow diet, and their weight curves tracked the same. The wild type animal was fed a high fat diet and became obese. 45 grams is obese for a mouse. And that's what we usually see in mice that we feed this very high fat diet. But surprisingly, the high fat diet fed knockout mouse really didn't get obese. It tracked more like a chow fed animal until very late in the high fat feed. And so here over on the left is a picture of the two mice. This is the wild type animal. This is the knockout animal, so much more lean. And what was even more surprising, both on the chow fed diet and the high fat diet, they were eating twice as much food. So eating twice as much food and not gaining weight, now that's an attractive proposition. They were completely resistant to non-alcoholic, experimental non-alcoholic fatty liver disease, which we see in these high-fat fed animals. That's why part of the reason why we give them this diet to model that. They did not become diabetic, as these mice tend to do on these diets. So overall, this was a good deal for the mouse. And so the hypothesis was maybe things were going in the other direction, and our original idea that this was involved in creating heat was incorrect. And in fact, when we put these animals in a calorimeter to measure energy expenditure during the 24-hour dark light cycle, we could see that there was always a 10, 15, 20% higher energy expenditure compared to the wild type control. So in the end, what we found by going after this gene was a surprising finding that THEM1 is highly upregulated by cold ambient temperatures, but it functions not to generate heat, but to suppress energy expenditure. So how does it do that? And is there any way we could get into this and see if we can leverage it for a therapeutic purpose? So again, here is the schematic that I showed you earlier about how the fuel gets to the engine, the lipid droplet, into the mitochondria, and this is the pathway. And so if we just, there's a lot of data that I won't show you supporting this, but if we look, here's the brown fat that I showed you before, and on the bottom is the brown fat from a knockout animal. So you see at 22 degrees, you can appreciate that the lipid droplets look a bit smaller and perhaps more sparse. And then if you take the animal to 4 degrees, you could see that the lipid droplets all but disappear. And yet the ultrastructure of the tissue, which is the mitochondria and lipid droplets, are really preserved. And then if we, by a variety of different mechanisms, if we measure fatty acid oxidation rates, and so this is one example of that, where we do it in tissue homogenates, there's a big step up in rates of fatty acid oxidation. So these and a lot of other data support a model in which this enzyme is working in reverse of the pathway that allows fatty acids to translocate to mitochondria for combustion to create heat. So THEM1 is working in that direction. And what we think is going on is that it's stealing the CoA from these fatty acids, turning them back into free fatty acids, and regionally, all around here is the ER. And we know there are other isoforms of acyl-CoA synthetases that reconvert fatty acids into fatty acyl-CoAs, which are reassembled into triglycerides and stored. And at the same time, these are potent suppressors of the rate-limiting enzyme adipose triglyceride lipase, which frees up the fatty acids in the first place. So this is a counter-regulatory pathway, if you will. And we were able to drill down on some regulatory information that turns out to be important by really studying the biochemistry of this enzyme. And so Sean Hahn, who was in the lab about 10 years ago, first characterized this enzyme. He appreciated that it was active as a multimer. It was allosterically activated by ATP. It had a lipid binding domain, the start domain, that we appreciated at the time was required for full enzymatic activity. And just this past year, working with a very talented structural biologist, Eric Ortland, and his grad student, Matt Tillman, we were able to appreciate that the lipid binding that goes on at the start domain really tunes the activity of the enzyme domains that are N-terminal to it. And what Matt did was try to figure out what lipids were binding and sort of did that make sense and how does it fit in from a regulatory standpoint. And so he made a recombinant protein of the start domain. He exposed that to lipids. He then repurified the start domain and looked at what was bound. The most abundant thing that was bound were free fatty acids, and in particular, the kind of fatty acids that were hydrolyzed out of lipid droplets. And the other thing that was bound was lysophosfatidylcholine, which is just a phospholipid missing one fatty acid. And where you find this phospholipid is in the coat that surrounds the lipid droplet. So the lipolytic properties that occur when lipolysis of the lipid droplet is going on in the setting of heat production are the lipids that regulate this enzyme. And again, not showing you a lot of data, what Matt was able to show was that free fatty acids, when they're bound to the lipid sensing domain, activate the enzymatic activity, and the lysophosphatidyl-CoAs tend to inhibit it. So the lipolysis event fine-tunes the activity of the enzyme. And then we really wanted to ask the question that I'd like to spend a few minutes showing you, which is that, so this is an enzyme that's markedly and rapidly upregulated by cold ambient temperatures, and it suppresses thermogenesis. And so why does it make sense to upregulate that gene at the same time that the animal needs to be creating heat to preserve its core body temperature? And so we decided to look at where is the enzyme and where does it go under conditions that promote thermogenesis. And so here is an immunofluorescence microscopy that shows you lipid droplets and the main lipid droplet protein, perilipin-1, and it also shows you THEM1 in red. And so where the gold is this tends to be around, is the co-localization of the lipid droplet protein and THEM1. And you see that it does seem to hang around lipid droplets, but it's not really a true lipid droplet protein, or this would be a gold rim that surrounds the lipid droplet. It's more punctate. And so we wondered if that was an event or a location that was transient, and perhaps it moves around in response to some stimulus. And the same stimulus that activates lipolysis is phosphorylation of a bunch of proteins. So we considered that. And we did that by going to a new database at the time called PhosphoMouse, which was started by Steve Gigi here at Harvard. And he stimulated a bunch of proteins in the mouse he just purified proteins from all different organs and did phosphoproteomics to ask what proteins were phosphorylated in what tissue. And so by querying that database, we found that THEM1 in brown adipose tissue and not other tissues is phosphorylated at 3 N-terminal serines, 15, 18, and 25. And informatics suggested that the kinase was protein kinase C. And so we verified that by doing our own phosphoproteomics as a function of time after stimulating a brown adipose cell line with PMA, which activates PKC, and we saw phosphorylation events. And then with collaboration with Susan Hagen, who's a longtime collaborator at Beth Israel, who's a wonderful microscopist, and our common postdoc, Yue Li, what we did was we first localized THEM1 and showed that this little punctate area by immunofluorescence was a region that was very close to mitochondria and lipid droplets, but not really part of either. And so you could see them in cross-section here and in three-dimensional representation there. And then if we stimulate with PMA, you can see diffusion of this protein throughout the brown adipocyte. So it did move in response to PMA. And then what Yue did was do a bunch of work with stimulators and inhibitors in phosphorylation cascades. And what he found was that the PKC that stimulates phosphorylation of THEM1 and its diffusion throughout the cell is downstream of norepinephrine through a pathway that kind of makes sense through PKA, ATGL, diacylglycerols, PKC, and then ultimately phosphorylating THEM1. And then what Yue went on to do was show that the phosphorylation sites that we had originally found in the database were really relevant, but two were more relevant than the others. So it turned out that serine 15 and serine 25 were the phosphorylation sites that really drove this diffusion of the protein away from these punctate areas in response to PMA. And again, in the interest of time, I won't take you through all those data. But what I wanted to show, which was particularly exciting, was what exactly these punctate were. And Susan is, again, is a remarkably talented and creative microscopist and was able to do what's called correlative light and electron microscopy. And so we could see, using a horseradish peroxidase, transfected cells with the three phosphorylation sites killed by doing alanine mutations. And we could locate these puncta under light microscopy and then also look at them by electron microscopy. And what we saw by electron microscopy was very well ordered structures that had no membrane around them. And these are biomolecular condensates or membrane-free organelles. And this was such an organelle, and it was located just exactly where you would want to see it for this, which is in between mitochondria and lipid droplets. And if we mutated the phosphorylation sites into phosphomimetic mutations, we abrogated these organelles because the THEM1 diffused throughout the cell. And what these turned out to be driven by was a disordered domain at the N-terminus that seemed to regulate this. And this is a map of disorder in the amino acid structure of THEM1. At the N-terminus, there's a predicted disordered structure at exactly the same sites where phosphorylation occurs. So this is a site that's organizing these things into these punctae that seem to be active in suppressing fatty acid oxidation. And we could show that functionally using oxygen consumption in tissue culture as a surrogate marker for fatty acid oxidation. And we could go back to the mouse in vivo and show that the same distribution event occurred in response to to stimulating pharmacologically brown adipose mediated thermogenesis. And so what we have then is this enzyme that's markedly upregulated in response to cold temperatures, that has the capacity to suppress thermogenesis, but at the time that thermogenesis is required, the same thermogenic stimulus inactivates the protein and moves it away from its active site. So then, to summarize, when it's in its active conformation, it's in this membraneless molecular condensate that organizes and drives fatty acids that were targeted for mitochondria back into the lipid droplet. Adrenergic stimulation leads to phosphorylation and distribution of this protein throughout the cell. And one of the destinations, and I won't have time to tell you the story, is that the protein goes into the nucleus and regulates transcriptional events that are appropriate for this phase of thermogenesis. So overall, what we have then is a pathway towards energy expenditure that's characterized by an acyl-CoA synthetase, and we have a pathway towards energy conservation that's characterized by THEM1. And so then the proposition is, can you target THEM1 with a small molecule to promote thermogenesis and mimic the phenotype of a mouse that can eat twice as much food, not gain weight, and not be subjected to non-alcoholic fatty liver disease? And again, I won't show you this story. Chris Crum, who is on the call now, has completed a high throughput screen and at least shown evidence in cells that this is a feasible proposition. So I'd like to stop there and thank those in my lab who have contributed to this story over the years. Thank our funding sources, which include the American Liver Foundation, and thank you for your attention.
Thank you so much, David. That was fantastic. We have time for one quick question. Feng Tao Chi asked, do the THEM knockout mice develop insulin resistance equally as the wild type mice under the long-term high-fat diet context?
No, they're quite resistant to that. And it's a good question because we are still sorting out the contribution of just simply the energy expenditure itself, or the fact that long-term high-fat feeding upregulates them1 rather remarkably in liver and in white adipose tissue. And in those tissues, there seems to be contributions to white adipose inflammation as well as steatosis and excess gluconeogenesis. And so what the contributions are, we don't entirely know yet, but energy expenditure seems to be at least dominant.
Thank you so much. So we will move on next to a talk from Sekar Kathiresan, who's going to tell us about how to read the genome from risk to rewriting it for cardiovascular health, so taking us a bit out of the liver. So thank you and welcome, Sek.
Thank you, Kristen. I think I'm trying to share my screen, but it says host disabled participant screen sharing. There we go. Got it. Okay, it's a delight to participate in this symposium. Thank you for the opportunity. I'm going to walk you through some of the human genetics work that we've done over the years to understand risk and resistance to cardiovascular disease, and then more recently, gene editing medicines we're developing to rewrite for cardiovascular health, rewrite the genome for cardiovascular health. My disclosures. So the problem we're focused on is myocardial infarction, and this is a disease of the heart arteries. It's blood cholesterol produced by the liver, clogging the heart arteries over time. And then ultimately, a blood clot can form within the blood vessel, leading to complete cessation of blood flow and death of heart tissue. And that can be detected by symptoms, EKG change, or elevation of cardiac biomarkers. This disease, leading cause of death in the world, it's a complex trait, has heritable and lifestyle components. And the best understood risk factors for this disease are lipoproteins, which I'll tell you about in a minute. If you think about this disease and the genome overall, and the average individual has average MI risk, but some individuals may have a genetic factor that increases risk. So the question that we've looked at over the years is, what's the genetic basis for risk? And then conversely, what's the genetic basis for resistance? So, because some genetic factors might, rather than increase risk, actually decrease risk. And so these are the two questions I want to address first before we turn to the idea of gene editing. Now, these are the best understood risk factors, lipoproteins. These are a lot of the action here is from the liver, and the lipoproteins are schematized here. They are spherical particles with a phospholipid monolayer. There's a protein embedded on the surface, either ApoA1 or ApoB. And the lipoproteins are named based on density, high density lipoprotein, HDL, or low density lipoprotein, LDL, or the lipid they carry. Here, triglyceride-rich lipoproteins refers to these lipoproteins where the neutral lipid triglyceride in blue is the majority of the lipid in the center. So these are, as I said, very well understood risk factors for coronary heart disease. Now, back in 1997, we started enrolling patients at Mass General Hospital who had a myocardial infarction at a young age to study the inherited basis for risk. We focused on early onset myocardial infarction patients because genetics plays a larger role when you have when disease occurs at an earlier age. So over the years, we've done a series of experiments with this collection of patients with and without disease, and uncovered a couple of different paths to early onset myocardial infarction, genetic paths. One is the monogenic model, where a single mutation is sufficient to lead to early disease. Then you have the polygenic model, where the additive effect of many variants in aggregate can lead to early disease. I'll walk you through the results of these two models. The monogenic model, The experiments done involve patients with disease, without disease, doing exome sequencing on all the individuals. And the result of that analysis is at least 5 genes where coding mutations confer large effects on MI risk. The genes are shown in the first column: LDL receptor, ABCG5, LPL, ApoA5, and LPA. The second column shows the carrier frequency of the mutations. The third column shows the blood lipid lipoprotein that's elevated as a result of the mutation. And the last column shows the clinical effect, anywhere from 2 to 4-fold increase in risk for disease based on having carried the mutation. So this is really the monogenic model. We've also worked out the polygenic model for disease, and here the question has been, do common DNA polymorphisms contribute to MI risk? Looking at thousands of cases and controls, doing genotyping and systematically searching for allele frequency differences between cases and controls at millions of polymorphic sites across the genome. The results of this kind of analysis are shown here. On the x-axis are all the chromosomes, on the y-axis is strength of statistical evidence. Each dot is a specific polymorphic site in the genome. There are a range of different sites in the genome that are confidently associated with disease risk. Each specific polymorphic site has a very modest effect on disease risk, maybe 20, 30% increase in risk, but there are a lot of these sites. And so it's possible now to add up these sites into a score, so-called polygenic score. So if there are n polymorphisms, 0, 1, or 2 copies of the risk allele at each polymorphic site, you can have a score ranging from 0 to 2n for each person. We and others have looked at these scores and asked how they predict one's future risk for heart attack, and indeed they do. More recently, we moved beyond just the sites at the top of these association lists to consider the full range of polymorphisms evaluated in the study and calculated polygenic scores involving millions of polymorphisms. In this case, 6.6 million common DNA sequence variants. And this is a distribution plot of polygenic scores calculated across 300,000 people in a prospective cohort study called UK Biobank. And you can see that the polygenic score calculated from 6.6 million common variants in each person has a beautiful bell curve distribution in the population. It's a Gaussian distribution, and it's essentially a new quantitative risk factor for MI. It's a single number which captures the genetic liability to myocardial infarction based on all these common DNA sequence variants. Now, some people are high in the score, some people are low, most in the middle. And the question becomes, if you happen to be high, how much higher risk are you compared to everybody else? And how does that contribute to risk for early myocardial infarction? So if you think about the two models, you take 100 patients with early myocardial infarction. It turns out that the monogenic model is relevant to roughly 2 out of the 100. And you can find a mutation in one of those 5 genes that I mentioned earlier. And the risk increase is about a 4-fold increase for risk for heart attack. Premature heart attack. The polygenic model is relevant in quite a few more patients, about 17% of the early MI patients. And again, the risk here is also about 4-fold increased risk. So this is really a summary of the genetic basis for risk. Now, one important point is that the high polygenic risk folks are currently unaware of their risk. And because no measured biomarker that we use in kind of clinical practice right now, like blood pressure or cholesterol, really tracks with this high polygenic risk. And one really needs to genotype across the genome to get this number. And that's an important opportunity for prevention and a work in progress. Now, with that on the risk side, let's move to the resistance side. As I said, there might be mutations that, rather than increased risk, confer resistance to disease. And so we and others have worked on this. And the main reason to identify resistance mutations is to guide the development of new medicines. So the best example of this whole paradigm playing out is for LDL. And there are a series of gene mutations in different genes that lower LDL cholesterol lifelong, and those mutations confer resistance to heart attack. And then there have been medicines developed to mimic those resistance mutations and then tested in patients, and those medicines indeed lower LDL cholesterol and reduce risk of heart attack. So that's really has established low LDL as the best validated causal pathway for resistance to myocardial infarction. Now, we wondered whether there are additional paths to resistance beyond low LDL. And this is, by the way, just an example of one of those stories, the gene PCSK9, very familiar to probably most of you. Individuals who naturally lack the gene have been identified. They are human knockouts. They have lifelong low blood LDL cholesterol levels as low as 14. They're healthy and they're resistant to coronary artery disease. Now, are there more protective pathways beyond low LDL? So we've worked out one, which is low triglyceride-rich lipoproteins through angiopoietin-like 3 and related genes. And I'll walk you through that story. So this goes back to this woman, Anna Fuhrer, we worked with back in 2010, 2009, 2010. She had this lipid profile, LDL of 37, triglycerides of 19, so really low lipid values. This was identified at a corporate health fair. The people who identified these low lipid values in Anna thought it might be dangerously low and sent her to a lipidologist, Gus Schoenfeld in St. Louis. He tracked down the family across 3 generations. So here's Anna, 2, 6. Several of her brothers and sisters have the very low lipid levels as well as are members of her mother's family, as well as her children. And Gus thought a single gene was responsible for this tracking of low LDL in the family, and spent about a decade trying to find this gene. He was able to link it to chromosome 1, but was not able to pin down the causal gene in the family. We collaborated with him to do exome sequencing when it was just becoming available as a technique, and then found the gene. The gene was angiopoietin-like 3, or ANGPTL3. And what we were able to sort out is this pattern in the family. So here's Anna, 2-6 here, generation 2, number 6. Her mother, generation 1, number 7, carried this nonsense mutation in the first exon of ANGPTL3. You can see that several of her brothers have that, and then that was passed down to several of Anna's siblings. Now, it turns out that the father married into this family with a different nonsense mutation in the same gene. They went on to have 9 children, and 4 of the children inherited 1 defective copy from the mother and 1 from the father. So these 4 individuals are human knockouts of ANGPTL3. What this family taught us is that lacking ANGPTL3 is tolerated. These individuals were healthy, but they had these very low lipid levels. We went on to show that if you had 1 copy deficient, that was prevalent, about 1 in 300 in the US. People had that null mutation in the ANGPTL3 gene. These individuals had lower triglycerides, lower LDL, and lower risk of myocardial infarction. So these data put angiopoietin-like 3 on the map as a therapeutic target for lowering lipids and atherosclerotic cardiovascular disease. So data from a couple of other genes in this pathway all point to lifelong low triglyceride-rich lipoproteins by enhanced lipolysis as a key pathway beyond LDL to confer resistance to myocardial infarction. So what I've tried to share with you is some of the genetics work that we've done to look at risk for premature myocardial infarction, and the monogenic model, the polygenic model, and these genetic factors help identify individuals who might be likely to benefit from early intervention. On the resistance side, these mutations that we and others have identified point the way to causal pathways and could inspire the development of new medicines. So that's what I want to close with, is really thinking through how the resistance mutations identified could be leveraged for the development of new medicine. So imagine if there was a single treatment that could durably and safely lower blood LDL cholesterol. Our human genetics research suggested that such a medicine would have the potential to treat and ultimately actually eradicate this coronary artery disease altogether. So this is the vision behind the company I founded called Verve Therapeutics, where I'm at right now. And I want to walk you through the gene editing medicines we've developed as a progress toward this vision. So can we transform the care of atherosclerotic cardiovascular disease, coronary heart disease, heart attack, from daily pills to intermittent injections to truly a once and done approach? So those genes that I mentioned, PCSK9, ANGPTL3, they're 2 genes that are basically cholesterol raising genes. And what the human genetics has taught us is these are genes that need to be turned off for health, and ideally permanently turned off, and as a way to lower LDL cholesterol and treat atherosclerotic cardiovascular disease. So we turned to a couple of different gene editing technologies for the ability to permanently turn off either PCSK9 or ANGPTL3 in the liver. And two of the technologies are shown here. On the left is Cas9, so-called molecular scissors that allows you to cut DNA, and then error-prone repair leads to disruption of a gene. On the right is so-called is base editing, a so-called molecular scissors, a molecular pencil eraser approach. Here you're able to basically chemically convert one base to another base. In the case of adenine base editing, an A to a G at a given spot in the genome. So these are two different ways to turn off a given gene. We evaluated these two technologies for the ability to turn off either PCSK9 or ANGPTL3 in cells, in mice, and most importantly in non-human primates before prioritizing the adenine base editing technology for our first two drug programs, VERVE-101 and VERVE-201. The drug product that we've developed is schematized here. It's the adenine base editor delivered as an mRNA, about 4,000 or so nucleotides, guide RNA, about 100 nucleotides, shown here in green. Both of these nucleic acids are packaged in a lipid nanoparticle, and this is shown as electron micrograph here, and this is the drug. This is infused into the bloodstream of mice or monkeys over a 60-minute period. And the ultimate intended effect after the medicine is taken up by liver cells and the mRNA and the guide RNA are released into the cytoplasm, is that there's editing happening in the PCSK9 DNA sequence, an A to G conversion at the canonical splice site at the end of exon 1 going into intron 1. And this splice site disruption turns off the gene, turns off either, in the case of PCSK9, for the first exon change, and then it can also, the same approach can be used to turn off the ANGPTL3 gene as well. So these are our two drug products in terms of VERVE-101 and VERVE-201. And what I want to this is the pipeline that we're developing these two products initially for familial hypercholesterolemia, the genetic disease, and then ultimately intending to develop it for atherosclerotic cardiovascular disease as well. What I want to walk you through is some of the preclinical data in non-human primates testing the adenine base editor technology. This data was published. Some of this data was published this past May here in this publication. This is the typical study design. As I said, the drug is delivered as an intravenous infusion. Two groups of monkeys, group 1 that gets drug, group 2 gets control. 3 endpoints, whole liver DNA editing assessed by sequencing, blood PCSK9 level and blood LDL level. These endpoints are measured at time 0, at time 2 weeks, and then the monkeys have been carried forward for now out to 450 days. So with drug treatment, this is what you get in terms of the first endpoint. Shown here on the x-axis are each bar is a monkey. On the y-axis is the level of adenine editing in the liver DNA at the PCSK9 target site. You can see the purple bars are the treated monkeys. There's 67% whole liver DNA editing at the on-target site. The consequence of that adenine editing at the on-target site is to turn off the PCSK9 gene and reduce the blood PCSK9 protein by about 90%. And the blood LDL level goes down by about 60%, all at 2 weeks. Now, the question is, how durable is this one-time treatment in terms of the turning off the gene? This is data now out to 450 days, so 15 months. On the x-axis is days post that initial treatment. On the y-axis is the percent change from PCSK9 from baseline for the PCSK9 level. And what you can see is that dramatic drop in PCSK9 that we got at 2 weeks has remained durably low now out to 15 months. And the LDL similarly, 60% drop at 2 weeks, and then 15 months later, still a 60% drop. So this is truly going to be a one and done therapy, a one-time treatment, permanent lowering of LDL cholesterol for the treatment of heart attack. Now, what we've been able to do is essentially make a single spelling change in nearly every liver cell in a living animal. I want to close with the second program, and that's the ANGPTL3 program. And here, we wanted to reach heterozygous FH patients, but also a specific type of FH disease called homozygous FH. So this is FH, familial hypercholesterolemia. This is a monogenic form of high LDL and premature heart attack. The dominant mutation is in a gene called LDL receptor. In normal liver, there's a full complement of LDL receptor on the surface of liver cells. In heterozygous FH, there's 50% deficiency. In homozygous FH, there's a complete deficiency. Standard lipid nanoparticles, like the one we're using for VERV 101, get taken up actually in the blood, from the bloodstream to the liver cells by the LDL receptor. This is an underappreciated fact, actually. With systemic delivery of lipid nanoparticles, they get taken up to the liver by the LDL receptor. So standard lipid nanoparticles work well in normal liver and heterozygous FH, but they do not work at all in homozygous FH. Okay, so this is a problem for us if we want to reach homozygous FH patients for gene editing in the liver. And so we had to come up with a solution. This is the data behind this concept that standard lipid nanoparticles don't work in homozygous FH. These are 3 different genotypes of mice: wild type, LDLR het, and LDLR minus minus, so homozygous FH mice. And the y-axis is adenine editing delivered through a standard lipid nanoparticle. And what you can see is that it works well. The standard lipid nanoparticle works well in wild type and LDLR het mice, but not at all in LDLR homozygous knockout mice. So we've solved this problem by adding a GalNAc ligand to the LNP. And the GalNAc binds to a different receptor on liver cells called ASGPR. And when you have a GalNAc LNP, that you can now see that we have complete rescue of this defect in delivery, going from this blue bar to the purple bar. The homozygous FH mice are now able to be edited in the liver using the GalNAc LNP, carrying the base editing tools. So this proprietary GalNAc LNP now we've taken to monkeys. So we wanted to test whether the GalNAc LNP efficacy will translate to larger animal models such as NHP, because data in NHPs for lipid nanoparticles and gene editing has been more predictive of human success than data in mice. So to do this, to evaluate this, we first actually had to create a monkey model of homozygous FH. I'm actually not sure if anybody's ever done this before, but we took this on. What we did was we actually knocked out the LDL receptor, somatic knockout, in monkeys using standard Cas9, standard lipid nanoparticle, and LDLR guide targeting LDLR. We deleted about a 50 base pair portion of the LDLR gene, and that's what's shown here, the study design. And the consequence of that is this is now looking at editing of the LDLR gene in the liver biopsy in those monkeys. You can see there's excellent editing here, 60-70% editing. That led to about a 94% reduction in LDLR protein expression on liver cells on biopsy. And then most importantly, it led to about a 6-fold increase in plasma LDL from 50 to about 300. So we've recreated the physiology, the pathophysiology of homozygous FH in these monkeys. So now we have a monkey model of homozygous FH, and we can come in and test to see whether the GalNAc LNP will work in this monkey model. Before doing that, we actually tested a standard LNP in the monkey model. The prediction here is it wouldn't work because there are no LDL receptors, and indeed that's what we saw. You can see the editing here. There's minimal editing on in the HoFH monkey model, and then there's no change in the ANGPTL3 blood level. Because these standard LNPs are not getting taken up by these homozygous FH NHPs that lack the LDL receptor. So, but then we came in with a GalNAc LNP, and what you can see is that there's this dramatic increase in editing, so real about 60-70% editing, and then the plasma ANGPTL3 level, the protein made by the gene, is completely shut down here, now lower by more than 90% after treatment. So the GalNAc LNPs achieve effective ANGPTL3 base editing, even in the setting of homozygous FH NHP liver. This experiment was repeated, and this is with a couple of different LNPs now, the GalNAc LNPs, and we're now able to reduce plasma ANGPTL3 level anywhere from 94 to 97% in NHP model of homozygous FH. So we're able to completely inactivate this gene in the liver. So let me close with a couple of slides, kind of taking you back to the big picture of what we're trying to accomplish. if you take a typical patient with homozygous or heterozygous FH, this is a heterozygous FH actually, they have high LDL starting at birth, myocardial infarction early in life, here shown at age 44. They get treated with standard of care, LDL lowering therapy. LDL comes down, but typically over the life course doesn't stay down. There's oscillation in the LDL over time because of issues of adherence, healthcare infrastructure needs, and also healthcare access requirements. And that lack of LDL control over time leads to recurrent heart problems like procedures or even a fatal heart attack. So this is the chronic care model for LDL care, and this model is broken. And what we'd like to do is replace this model with this model, a single course gene editing medicine given at the time, in this case, at the time of an MI, it would dramatically lower LDL and durably do so for the life of the person. So this is the vision for our medicines. So let me summarize then. This is the entire story in a single schematic, and I'll stop here. Thank you.
Thank you so much, Sek. It's amazing to see the progress that's been made in what seems like relatively short time. If anyone has questions, please put them in the Q&A box. One question I had was combining these two perturbations. What does that do to risk?
Yeah, so that's a great point. They actually the PCSK9 axis and the ANGPTL3 axis of risk are actually orthogonal to each other. So if you add one on to the previous, you're going to get additive lowering of LDL. And that's actually already been tested in patients with monoclonal antibodies, one that targets PCSK9, one that targets ANGPTL3. So there's a study done recently where take a bunch of patients with severe heterozygous FH, you put them on a monoclonal antibody targeting PCSK9, and the LDL comes down, but let's say it still was high at not 150 range. And then the monoclonal antibody targeting ANGPTL3 was added, and the LDL came down by another 50%. So that sets up actually a potential way we're going to develop the second program, the ANGPTL3 program. It'll likely be an add-on to the first program in patients who still have unacceptably high LDL levels after the PCSK9 product.
Gotcha, thank you. We have a couple questions in Q&A. So the first is from Randy Levinson, who says, but with genetic screening, would one need to wait until an MI occurs to treat FA? And I guess more broadly, that leads to this question of how are you going to define, is the target population everybody, or is the intent to narrow that?
Yeah, no, so we have a kind of a bullseye model of clinical development. The initial group that we're going to focus on is patients with FH. So that's a genetic subtype of basically high LDL and premature MI. It's a lot of people actually in the US. The prevalence is 1 in 250. It's about 1 million people in the US, 31 million people globally. So our treatment would be a genetic treatment for that genetic disease. That's the initial group, let's say the center of the bullseye. But the same medicine would be appropriate for garden variety heart attack patients. So stop patients with anybody with a heart attack for any reason, because the average person with a heart attack in the US is about 65 years old. They're facing about 20, 25 years of LDL care after that heart attack. So imagine a one-time treatment at the time of heart attack that would dramatically lower LDL, durably lower LDL. So this is the second group, and that's 22 million people in the US. The third group, the outer ring of the bullseye, is what you're getting at, is this ultimately could be truly a preventive medicine before somebody has a heart attack, because lowering LDL is the main treatment once you've had a heart attack, but it's also the main treatment, a main way to prevent a first heart attack. So you can imagine this medicine, once there's a sufficient safety database, that it could be given to people when they turn 30, 40 as a way to prevent a first heart attack.
Great, thank you so much, Sek. There's a few other questions in the Q&A. In the interest of time, we're going to move on, but I know you are all able to interact in the Q&A and in the chat if you have any further questions or want to communicate further. And so for the last part of this session, we're going to move on to the micro talks. And again, as a reminder, this is a very brief talk, so we're going to bypass any sort of question and answer in between the talks and just have everyone run through them sequentially. And we will begin with Arkadi Shwartz from Wolfram Goessling's lab at Harvard.
Okay. So I want to start by taking the chance to thank the organizer for letting me to present today my work, which deal with macrophages in liver development. And if we will stop and think for a second about developmental pathways, so there are a handful of pathways that execute developmental programs. And while in the early development, the key is the integration of those pathways together to generate those to execute properly the developmental program. What happened with those pathways when they need to be integrated into formation of a complex organ such as a liver, it's less known. And if you will follow the literature, you probably find that there are suggestions that those pathways are regulated by different non-parenchymal cells, such as endothelial cells. And in this project, we decided to look in a different non-parenchymal cell population, which are the macrophages. And there are two main reasons for this. One is that the liver harbors the largest amount of macrophages among the solid organ, and also that the macrophages are actually crucial for the maintaining liver homeostasis. So we decided to elucidate the function of macrophages in liver organogenesis by using zebrafish and the mouse model. So using the amazing ability to do live imaging in zebrafish, we were able to characterize the interaction between the macrophages and the developing liver. In this example, with the biliary epithelial cells, we found out that the macrophages spend longer inside the liver as much as development progressed. And also we found two different types of interaction: kiss and run interaction, when the macrophages are actually coming and going from the liver, and also those tight long-term interaction, which happened later in development. Using a genetic models to ablate macrophages, we found that the biliary tree that form in the absence of macrophages is truncated and also lacking its characterized branching complexity. And to look more into detail in this process, we switched to mouse model, and we found that upon ablation of macrophages in mice, we found that there is a significant reduction of SOX9 cell, which is one of the first marker for biliary epithelial cell differentiation. And this indicates that macrophages will participate in the induction of biliary epithelial cell differentiation. And again, using a zebrafish, we use the genetic tools that were available and found that TGF-beta pathway is actually the one regulated by the macrophages. And in this experiment, we ablating macrophages and hyperactivate TGF-beta in its background. We see that there is a significant amount of rescue to the biliary tree growth. So to sum up what I showed you today, macrophages can facilitate liver organogenesis, and they can coordinate biliary tree differentiation, in particular the biliary epithelial cells, and they can also modulate TGF-beta in the process of modulating TGF-beta pathway, and this can happen directly or indirectly. And another thing that I didn't show you today is that the initial liver progenitor, the hepatoblast, can mimic inflammation to attract the macrophages to the area. And with that, I'd just like to thank the Wolfram Goessling Lab, our collaborator, and thank you all for listening.
Thank you so much, Arkadi. We will move on next to Maria Mironova, who's from Daniel Stein's lab, also at Harvard.
Good afternoon, everyone. I'm glad to present our research. It's called Disparities in Performance of the Model for End-Stage Liver Disease Score Across Different Races and Ethnicities. My name is Maria Mironova. Cirrhosis is one of the leading causes of death in the United States, and the Model for End-Stage Liver Disease Score is used to estimate 3 months mortality in patients with cirrhosis. It's used for liver transplant prioritization, and it's also used as a proxy of in-hospital mortality. Little research has been done on the impact of patients' race and ethnicity on the score performance. And to do so, we analyzed the records of the patients with cirrhosis admitted to Beth Israel Deaconess Medical Center from 2008 and to 2018. And we analyzed the records of the patients self-identified Asian patients, African-American patients, Hispanics, and white patients. And as we see in the table 1, there was no difference in the groups in terms of age, gender, MELD score, mean MELD score, length of stay, and outcome, which is in-hospital mortality. We used areas under the receiver operating characteristic curve as a measure of discrimination. And we can see some difference. However, this difference was not statistically significant. It's shown on figure 2. But when we look at standardized mortality ratios as a measure of calibration, we see the difference. We compared actual mortality rates and the mortality rates predicted by the MELD score. And we found out that the MELD score systematically overpredicts mortality for African-American patients and Hispanic patients. What does it mean? It means that the calculated MELD score is probably higher than the real MELD score that the patient has. Some patient can benefit because they can get quicker on the liver transplant list, but it can also do harm because some patients can be considered too sick if they have a very high MELD score, or they can, or some essential procedures, like for example, TIPS placement can be postponed. And it raises concern about racial bias. Efforts have been done to overcome bias. Research on MELD 3.0 was recently published, and MELD 3.0 is adjusted for gender. However, racial bias is not an easy fix, and more research should be done in this area. Thank you.
Thank you so much, Maria. And last but certainly not least, we have Jason Guo from Hao Zhu's lab at UT Southwestern.
Hello, everybody. First, I want to thank the presenters, well, the organizers for giving me the chance to present today, and share my screen. Okay, so I guess I'll start off, as I'm sure many of you know, when you look at the histology of a liver, it's seemingly monotonous. Sorry, I am having a bit of problems with my slides. Okay, sorry about that. So as I'm sure you all know, when you look at the histology of a liver, it's seemingly monotonous, but in reality, the liver is actually fairly heterogeneous, consisting of different zones, zone 1 being closer to portal triads and zone 3 closer to central veins. And we kind of look at the linearized view of hepatic lobule, you can see that hepatocytes of different zones can often be segregated by metabolic function. And recently, our lab has been interested in studying these metabolic zones in the context of liver cancer. And one of the models that I want to talk with you all about today is studying activated beta-catenin and deleted ARO-AAT, which are commonly co-mutated together in HCCs. Now, to kind of study these mutations in a zone-specific way, we turn towards some new zone-centric Cre-ER mouse lines generated in our lab to induce these mutations, specifically in zone 1 using GLS2 Cre, or zones 2 and 3 using CYP1A2 Cre. And what we found is that we can actually track the fate of activated beta-catenin mutant cells using glutamine synthetase or GS expression. And what we saw is that over time in zone 1, these mutants tend to be relatively stable, kind of no real change in cell population of GS expressing cells over this time. But in zone 3, we saw a dramatic decrease in the number of cells ectopically expressing GS and thus activated beta-catenin. And while this is really interesting that these zone 3 mutants are disappearing over time, what we found to be really surprising is that this is where tumors more often arise, as you can see here in the liver pictures on the left and quantified on the surface tumor numbers on the right. And this is really interesting and kind of begs the question, what about zone 3 is promoting tumor formation even though mutants are disappearing in the zone? And to kind of answer this question, we shifted towards a different model using hydrodynamic transfection, or HDT. And kind of the main idea here is that given a panel of zone 3 specific genes, can we knock them out in the context of other oncogenes such as beta-catenin and MYC and kind of maybe find out what about zone 3 is causing this different cancer potential. And kind of our early results are pretty interesting. So looking at the relative liver to body weight ratio as a proxy for tumor burden, just 1 week post-HTT, here you can see kind of our non-targeting control or oncogene only model, our CDK1 knockout that shouldn't really have much cell proliferation. But when we knock out zone 3 genes, GSTM2 and 3, we see a reduction in the relative liver weight and thus maybe tumor burden. And kind of visually, when you take a look at these livers, they seem to be just a bit smaller, a bit cleaner than our oncogene only model. And while this is a really interesting trend, we kind of wanted to dig a little bit deeper into this. And at 2 weeks post-HTT, we're starting to see kind of a bit more of a change in the relative liver weight when we knock out GSTM2. And so furthermore, when we analyze these 1 week post-HTT livers using H&E staining, we can see a dramatic decrease in the number of early tumor lesions or microtumors when we knock out GSTM2 and 3, maybe suggesting a role of these genes in zone 3 for tumor initiation. And so while this is kind of some early data, the main takeaway that we want to give is more than just thinking about the paradigm of clonal dynamics in cancer formation, also maybe thinking about things like metabolic zonations when studying cancer. Thank you.
Welcome back to our final session. This afternoon, and the first speaker that we have is Professor Anna Musch. She is a professor of molecular and developmental biology at Einstein Medical College, and she is going to tell us about a matter of architecture, how to build epithelia with hepatocytic polarity. Welcome, Anna.
Thank you. Thank you very much, Sangeeta and Wolfram, for inviting me. I will start sharing my screen. Yeah, I want to start out by saying that it's really been a pleasure to hear all the speakers. And with regard to Win, I have followed Win Arias. Let me find this here. I have followed Win Arias' work on protein trafficking and polarity in hepatocytes for many years and have admired his fundamental contributions, and recently have also become to admire his tireless championship of the subject matter of polarity. And so Win, it's a particular honor for me today to try to promote our common interest in polarity at this symposium. So my interest in the liver, as I tried to allude to, is from the perspective of an epithelial cell biologist, because the liver really presents a fantastic model for how epithelial form is adapted to its function. So when we look at the two epithelial cell types in the liver, the bile duct cells and the hepatocytes, it is obvious that they have very different organization. So bile ducts are simple tubes that are simple conduits for bile, whereas hepatocytes are organized in cords that are sandwiched between blood vessels. And at the same time, between them, they have this adjacent to a canaliculi network of luminal domain, the bicanaliculi network. And this, of course, promotes the extensive exchange with the blood that hepatocytes need to accomplish. And at the same time, to also be able to excrete bile into this bicanaliculi network. So if we zoom in further into these epithelial cell types, we see that bile ducts have a classical epithelial organization that you find for epithelia in most parts of the body. That means they have a single apical or luminal domain, and each cell gives a single luminal domain to the central cavity, and that is opposite a basal domain that's parallel to the luminal domain and perpendicular to those lateral domains. Now, if you look at hepatocytes essentially can make a luminal domain at each side where it's in contact with a neighboring cell. So these luminal domains are essentially grooves that just interrupt cell-cell adhesion domains. So this particular hepatocyte has a luminal surface that it shares with one neighbor here and at least one other with this neighbor here. At the same time, the free surface that is the apical domain in a ductal cell in the hepatocyte is now used as a second basal domain. So hepatocytes can be thought of as multipolar with multiple luminal domain and two basal domains. And this, of course, helps to mediate the close apposition and the exchange with the vascular system. So the other difference between or uniqueness of hepatocytes compared to ductal or other epithelial cells is that epithelial cells usually deposit basement lamina that separates them from the endothelium. And hepatocytes are unique in that the space of this is generally devoid of a basal lamina, proper basal lamina. In fact, the endothelial cells are fenestrated to, again, facilitate the exchange of macromolecules between these two cell types. So this is all geared towards a maximum contact with the blood in the endothelium. So essentially, you have this very different architecture in which you have two basal domains versus one basal domain, multiple luminal domain that are perpendicular to the basal domains here rather than parallel in these two epithelial phenotypes. And both of them are generated from a common precursor, the hepatoblast. And this is really a fascinating question of how from one epithelial precursor you can generate two epithelial cell types with such vastly different architecture. And this is not only a basic science question, but it's important to understand because as we've seen that the organoids that are becoming increasingly popular from these epithelial cells we still have problems to properly recapitulate in those organoids the hepatocytic polarity. So this is the main interest in my group. And so for a few years or many years now ago, we came across or we established a system in which we can experimentally switch the polarity between these two phenotypes. And it is based on a serine-threonine kinase called PAR1B. So when you have a ductal epithelial cell, in this case, it's a cell line derived from the kidney tubule, MDCK, Martin-Darby-Kaning kidney cells, that nevertheless have this classical ductal epithelial polarity in which the apical surface is on top, as you see here. Lateral surface is here, tight junctions separate both. So if you overexpress PAR1B kinase in these cells, they now organize with the architecture of hepatocytes. They don't make long bicanaliculi, but nevertheless, you see now that the luminal domain is not on top. Indeed, you have two basal domains here, and rather the luminal domain is interrupting the cell-cell contacting domains here, as you see. So conversely, when we take hepatocytic cell line, a bona fide hepatocytic cell line with B cells that do present with those pseudo-canaliculi, so these are lumen that, again, established between neighboring cells here, and we express a dominant negative form of the PAR1 kinase, we switch their polarity to one in which they are now forming their luminal surface here in red on top. So the jury is still out if PAR1B is indeed critical for the branching of the polarity phenotypes in vivo. And the reason why this is so hard to establish is because PAR1 is one of 14 kinases that have substantial substrate overlap, and several of them also similar subcellular localization. So there's presumably a lot of redundancy going on. Nevertheless, this system has allowed us to study or allows us to interrogate and to establish some basic mechanisms that are govern these polarity changes. And so our first hunch was that the differences in the basement membrane organization or the differences in ECM signaling that are the consequence of a lack of basement membrane and presence of basement membrane respectively might have a role in this in these phenotypes. So when you look at the classical ductal cell like MDCKs, they deposit their own basement membrane here, composed of collagen 4 and laminin, predominantly here stained for collagen 4. And when you look up upon PAR1B overexpression in those cells that make this lateral lumen that you see here, you see that the laminin distribution or collagen 4 distribution is altered. So you have now less collagen 4 at the bottom, and you have some also on top, and just generally the asymmetry is is no longer as pronounced. If you take the hepatocytic cells, on the other hand, they, as you expect from hepatocyte, lack the deposition of collagen 4 and also laminin. And expression of dominant negative PAR leads to the deposition of some collagen 4 here. So that encouraged us to test in more detail the role of the extracellular matrix in this phenotype. So we decided to experimentally add culture these with B cells on collagen 4. And what we observed is that this is switching the polarity phenotype. So you see that these are the control cells with the lumina, pseudocanaliculæ. And here we see what we call a chicken wire tight junction staining and the lumina protein on top. So this is essentially the ductal polarity phenotype in culture. So just by plating these cells on collagen 4. Now, collagen 4 is known to usually regulate Rho signaling at the cell adhesion sites. And so when we look at the PAR1 overexpressing MDCK cells, we find that overexpressing PAR1 indeed reduces the total levels of active Rho. So this is a pull down with a, that pulls down active Rho, and also the main substrate of Rho for cell shape regulation, phosphomyosin light chain, is reduced, phosphorylated in the PAR1 overexpressing cells. So we found then that when we inhibit or deplete Rho-A from these MDCK cells, that this is sufficient to replicate the PAR1 overexpression phenotype. So basically getting rid of Rho-A, we make the pseudocanaliculæ in MDCK cells. And again, conversely, when we use a pharmacological activator of Rho in those with B cells, these hepatocytic cells, we now make them organize with this chicken wire tight junction and with the lumina protein on top. So it appears that Rho signaling has downstream of those ECM signaling might have a role in this process. So that essentially raised the question, how does signaling at the basal domain work to define apical domain polarity? So we considered essentially two pieces of knowledge. The first one is that it is well established that there's a crosstalk between cell ECM signaling and cell-cell adhesion signaling. Although the molecular details are still fully understood, it is clear that the increased traction forces at the cell matrix are balanced by cell adhesion forces between cells. So this was the first piece of data that we were considering. And the second piece comes from work in the polarity field, in vitro polarity field with this MDCK cell model, in which we know that when cells are naive, that means initially non-polar before they undergo cell-cell contact, that the first, when they make their first cell-cell contact, this cell-cell contact serves as a cue to recruit not only cell-cell adhesion protein, but also apical determinants to the sites of cell-cell contact. And only during the establishment of polarity then are the two kind of two determinants sorted apart from each other, and the luminal or apical proteins are displaced to the top. So we considered that there might be a weighing for dominance at these contact sites between the adhesion and the apical determinants, and that the adhesion strengths in turn determined by matrix signaling might be a way to regulate the positioning of the luminal domain. So to test this, so this is essentially the hypothesis that there's ECM signaling Rho activity regulating cell-cell adhesion, and that might then determine this kind of polarity branching. So there are three adhesion systems in epithelial cells. The most known one is the one E-cation with its associated catenins. Then there's one by nectins, and one by here by protein JAM-A, junction adhesion molecule A. So JAM-A eventually becomes a part of the tight junction. These other two end up at the zonular adherence, but initially they all mingle at the sites of cell-cell contact. So we first focused on ECAT in particular because it was already known to be linked, its adhesion linked to the establishment of Rho activity in the case of an adhesive actin contraction belt at the zonular adhesion. So the reason why ECAT can actually have mechanical force is because on one hand, the ECAT adhesion at the extracellular domain between neighboring cell occurs in trans, and on the other hand, you have a link bit of the cytoplasmic domain of ECAT via catenins to a contractile actin cytoskeleton. So that can cause a tension. And so to see if this kind of adhesion strength has any role in the polarization, we swapped the endogenous ECAT for an ECAT mutant that lacks the cell-cell adhesion domain. And we found that at least temporarily, these cells form luminal domains that organize like hepatocytes, like or these pseudocanaliculæ, like the PAR1 overexpressing cells, suggesting that ECAT adhesion does have a role here. We also tried to compare the adhesion strengths between these type of cells and these type of cells. So between control MDCK cells and PAR1 overexpressing MDCK cells. So to do that, we used a biosensor developed by Borghi et al. And so the authors introduced between the transmembrane domain and the beta-catenin binding domain that will eventually link to actin, an elastic linker with a FRET pair. And so if these catenin molecules are under tension, these two molecules are separated and there would be no FRET. And if the adhesion is poor, if they are slack, then there would be FRET occurring between the blue and the yellow molecule. So you can measure this essentially. The FRET is when the energy emission of this, upon excitation of the blue fluorophore, it's part of its emission is absorbed by the yellow fluorophore. So if you bleach the yellow fluor, the blue fluorophore emission will then increase if there is FRET indeed. And you can see before and after bleaching, there's an increase here in this heat map for PAR1 overexpressing cells, not such an increase in the control. So you see overall higher levels of FRET, which means less cell-cell adhesion when PAR1 is overexpressed in these cells when they establish this hepatocytic polarity. And importantly, we find a similar situation in the VIF-B cells, in the hepatocytic cells. So here, when we look at our control conditions, so these are the cells that form hepatocytic lumina. So when we either activate Rho or plate them on collagen 4, both conditions I showed you previously lead to the reversal of polarity, to this type of polarity. We see that their FRET activity drops. So now they essentially have stronger cell-cell adhesion. So means there is indeed a correlation between stronger cell-cell adhesion and this type of phenotype versus this type of phenotype. So we wanted to see if we could tease out some of such differences also in a more physiological model. And so when we culture mouse hepatoblasts from newborn animals, we can develop them into, in part into hepatocyte-like structures that have the hepatocytic polarity with kind of hepatocytic lumina. They have albumin. And in the same field, we often find also cells that develop into bile duct-like structure that lack albumin, that on the other hand have cytokeratin 19, the bile duct marker, whereas the hepatocyte-like structure lack it. So this presented an opportunity to now analyze the Rho signaling in these two types of structures in the same sample. So we haven't been able yet to express the ECAT biosensor, but we have analyzed Rho activity at cell-cell junction because high Rho activity correlates with this high ECAT tension. So again, we used a FRET biosensor that we expressed in this biadenovirus. And so this sensor now, the readout here is higher Rho level lead to higher FRET efficiency. And so if you look at cells that are bile duct cells and we bleach at cell-cell contact sites, we get a certain amount of FRET. And when we use these cells that have biocanalicular-like structures that we identify and then we bleach at the cell contact sites just next to them, we get another set of readings. And we find that clearly there is a significant difference in Rho activity at cell contact sites between these two. So essentially that suggests that Ikatian adhesion might be a downstream effector of this ECM signaling, and that could drive differential polarity development. So we also looked at the two other adhesion systems, the nectin and GEM-A adhesion system. So nectins come in there are 4 different nectins. So we depleted the common effector, which is a cytosolic protein via which nectin exerts all its signaling function, that is afardine. But depleting of afardine had no effect on the polarity decision. On the other hand, when we depleted GEM-A, we found that this also had a role in defining the hepatocytic polarity in our PAR1 MDCK cell system. So here again, we are back to the MDCK cells. We have under control conditions this classical epithelial monolayer. We overexpress PAR, which we do via doxycycline induction or doxycycline withdrawal in this case. So we induce the hepatocytic type organization in these cells. If we deplete GEM-A in these cells, however, we revert the polarity back to control conditions, as you see here. Now, interestingly, we can rescue this, this conversion when we also add a function-blocking antibody to beta-1 integrin. And the same was true when we added an inhibitor of Rho kinase, so we can also rescue this. So this actually suggests that there might be crosstalk between this adhesion molecule and the extracellular matrix as well. So, but unlike with the Ikatian condition, in this case, GEM-A adhesion appears to or might have a role in the signaling at the extracellular matrix, as suggested by these rescue experiments. So we wanted to see or study this more in a hepatoblast model. And so to go a bit more into this rodent hepatoblast differentiation model, by a protocol that Naoki Tanemitsu established in the early 2000s that we are following, what you see actually when you try to differentiate in vitro hepatoblasts, these are spindle-like cells. And the first signs of differentiation are actually sites of cell compaction. So you find these small areas where cells become more dense and lose their stretched appearance. You see that the nuclei become more round. And eventually those areas grow and become these areas of hepatocytic polarization. You can appreciate here rather extended bicanaliculi structure. They have albumin, they have HNF4 alpha, and they form these bicanaliculi, more elongated bicanaliculi than the cell culture PAR1 model. But if you look at GEM-A, you see that at the early stages where the compaction occurs, where these cells do not form yet any luminal domains, but just start to compact, that GEM-A is at those compacting sites. And once you see these islands starting to polarize, GEM-A becomes localized around the tight junction, circling the luminal domains. So now if you deplete GEM-A from the system, what you see is that there are fewer of those clusters forming. The overall cluster area that you find is lower, and the bicanalicular length that you can plot altogether is lower too. So it appears that the GEM-A's role is really to start this initial compaction step that is required for these, or precondition for this polarization to occur. Finally, we looked at this in a second model for hepatoblast differentiation. And this is a hepatocellular carcinoma-derived cell line, HEPA-RG. And we find a remarkably similar process differentiating or polarizing these cells. So they also start out as spindle-like cells when you just propagate them. But then the differentiation process involves also a compaction step, and that it's these very compacted areas here that then eventually yield these areas that have hepatocytic polarity. And so GEM-A is also here found at the earliest compaction steps. Curiously, before they show any bicanaliculite-like structures, GEM-A organizes like in ductal polarity with a chicken wire organization, and only reorganizes as these hepatocytic polarity sets in. So eventually they also have, albumin. Here is shown HNF4 alpha. So they have a whole set of hepatocytic markers and are generally considered a good model for differentiated hepatocytes. So again, when we deplete GEM-A in these cells, we find during that differentiation process, we find that they have much fewer lumina. So if we plot lumina per nuclei, they are less here in the GEM-A knockdown compared to the control. But also when we analyze the number of cells, so the cell density, which is an indicator of compaction, we find that they have fewer cells per area than the controls. So there's a pretty good correlation between these two phenomena, the formation of lumina and the compaction step. So basically what I'm trying to show you is that there are essentially two adhesion systems that crosstalk with the extracellular matrix. And E-cadherin is in the same, essentially, in the same direction as cell matrix adhesion, whereas GEM-A appears to be opposite cell matrix adhesion. So at least in our system, cell matrix adhesion can regulate E-cadherin adhesion, whereas GEM-A levels can somehow affect the cell spreading on the matrix. So how might this actually work? So it's conceivable, we think that the ductal cell that on a basement membrane, that the strong contractile acting ring that forms, or the tension that forms parallel to the basement membrane, might make it hard for contraction to occur at this perpendicular to this tension that would be necessary to establish a lumen, and that only when you have no basement membrane, low E-cadherin signaling, you might be able to accomplish this. And at the same time, for GEM-A, we think that GEM-A might allow cells to compact more and allow these ECM proteins to be, as compaction occurs, to be reduced, cell-cell contact to be stronger, and that allows closer cell-cell apposition, allows the lumen to form. There are clearly other mechanisms during differentiation that go on that lead to the altering of the integrin composition and basement and ECM composition that go on during differentiation, but we think GEM-A might be one contributor. So with this I finish, and I want to acknowledge the two people in my lab who have been for the whole time of this many years ongoing project, my faithful collaborators, David Cohen and Francisco Lazaro-Dieguez, some collaborators at Einstein, and the company that provided the HepRG cells. Thank you.
Thank you for that wonderful talks. Gorgeous images. There are some questions coming in from the chat. I'll read the first one from Shalev. Thanks for the interesting talk. E-cadherin is expressed by periportal hepatocytes, whereas N-cadherin is expressed by pericentral hepatocytes. Might that carry implications to polarity or luminal properties? Or are there corresponding zonal differences in ECM composition?
Yeah, I don't know about the EC. There are other people here who should be able to answer this. I don't know about zonal differences in ECM composition. Yeah, with regard to the cadherins, so we have the cadherins that are expressed by the HepRG cells is N-cadherin. And so for N-cadherin, there is an inhibitor that is used because it's also usually an EMT promoter. So there are inhibitors that we have tested and applied that could inhibit N-cadherin interaction, and that had no effect on hepatocyte polarization. So that appears to hold up even for N-cadherin in this case.
Right, I have a follow-up question, which is related to heterotypic interactions. So most of the data that you showed were in like cells forming junctions between each other. But, you know, Meri spoke earlier about the mesenchymal cells around the bile duct, and we've done a lot of work in vitro on co-culture systems. I'm curious if you've looked at any of those.
We have not yet. I mean, it's obviously, you know, a different level of complexity. So yeah, it would be wonderful to do it, and eventually we should branch out into this. But right now we have not. No.
Yeah. I mean, just to follow up, in our co-cultures, we see E-cadherin in homotypic interactions and N-cadherin on the hepatocytes facing the stromal cells. So I think there is something even within the cadherin family that could be interesting.
No, I'm aware of it. Yeah.
Great. Well, I think we are out of time. Thank you very much for that wonderful presentation. Wynn is clapping. We're going to move on now to our next speaker, who is Carolyn Bertozzi. Carolyn is the Baker Family Director of the Stanford ChemH Institute and the Ann and Robert M. Bass Professor of Humanities and Sciences in the Department of Chemistry at Stanford. She's an HHMI investigator, and she's going to tell us about therapeutic opportunities in glycoscience. Welcome, Carolyn.
Hi, Sangeeta. Thank you so much for the introduction. Is my video showing?
You are
yes,
showing.
Good. Okay, good. And can I share my screen?
Yes.
Yes. Okay. So I really appreciate the invitation to join this symposium. It's a really fascinating area of medicine. And although my talk is not centered entirely on liver biology, as you'll see towards the end of the talk, there is some relevance to a particular trafficking system in the liver. And that trafficking system is something that we are taking advantage of for a new kind of therapeutic modality that will be the subject of this talk. So of the many therapeutic opportunities in glycoscience that my lab pursues, one of them is to develop new therapeutics that perform what we call a targeted degradation of extracellular proteins with lysosome targeting chimeras. And we're going to abbreviate those molecules as LITACs. There's a hand raised, I noticed. I don't know if that was by accident or for my benefit.
No, just keep going.
Keep going, all right. So first of all, let me just declare a conflict of interest, which is that I am a co-founder and scientific advisory board member of Lycia Therapeutics, which is a company that has the exclusive license to the LITAC technology that I'll describe. Also, let me just start by highlighting the two co-workers who did the majority of the work that I'm going to present today. And they are former postdoctoral fellow Stephen Bannock, who I'm delighted to say is now my colleague as an assistant professor in the chemistry department at Stanford, as well as superstar graduate student Green An, who's now in her 5th year of the chemistry PhD program at Stanford. So the story actually starts about 20 years ago when Craig Cruz and Ray Deshays and their coworkers first described an idea for developing a type of medicine with a completely different mechanism of action compared to the more conventional medicines. And so conventional medicines are molecules that typically bind to a therapeutic target of interest, and they just block its activity. They could be small molecule inhibitors of enzymes. They could be monoclonal antibodies that bind to a protein and block its interactions with a cognate ligand or receptor. But they're relatively passive medicines that just bind and block. And what Craig and Ray described back in 2001 is an idea for a different kind of molecule that actually actively stimulates the degradation of a target protein. And in that first description, they called these molecules proteolysis targeting chimeras, or PROTACs. And these PROTACs were designed to recruit the ubiquitin proteasome machinery to a target protein of interest to degrade that protein. So the PROTAC is typically a small molecule that is bifunctional, where one part of that molecule binds to an E3 ligase enzyme inside the cytosol of a cell, and the other part of that PROTAC binds to a target protein of interest, which is a deleterious protein that one wants to get rid of in the treatment of disease. So if all is working well, that degrader molecule will basically form a ternary complex between the E3 ligase and the target protein. And that proximity allows that E3 ligase to transfer ubiquitin chains onto lysine residues. ultimately forming polyubiquitin chains, and that is a mark for the proteasome to degrade that protein. And what started as a really cool idea in a chemical biology academic laboratory has, over the last 2 decades, progressed into several clinical candidates that are now showing, promising activity in humans for a number of different disease categories. But as amazing and exciting as these PROTACs are, they don't cover the entirety of the human proteome in regards to their target space. And that's because the ubiquitin proteasome system resides within the cytosol. So only proteins that are cytosolic can be targeted for degradation through this pathway. And that basically excludes what is about 40% of the proteome from this targeted degradation approach, because 40% of our proteins reside outside the cell. They are either secreted entirely into the extracellular space, or they might be membrane-associated proteins that have only a minimal domain facing the cytosol that could potentially be ligated with a small molecule drug. And there are some very important targets within these extracellular proteins that one might like to target for degradation in the context of human therapy. For example, there are a number of cytokines, immune complexes, amyloid aggregates that are out in the extracellular space that cause pathologies and that are difficult to drug in the more conventional bind and block mode. And then on the cell surface, there are proteins that are well known to play roles in disease, such as receptor tyrosine kinases, various adhesion molecules, immunomodulatory receptors like the targets for cancer immune therapies. And many of these cell surface targets have also proven to be difficult to drug using the conventional, for example, monoclonal antibody. So 4 or so years ago, when Stephen Bannock joined my lab as a postdoc, we started thinking about, could we develop a targeted degradation strategy for these extracellular proteins? So immediately we thought, how does nature degrade such proteins normally? And one of the major pathways for degradation of membrane proteins and extracellular proteins is the endosome-lysosome system. So this is the pathway by which those proteins get internalized into endocytic vesicles, and then an early endosome undergoes a maturation process, ultimately becoming a late endosome, which then fuses with the organelle called the lysosome. And the lysosome is basically a container of enzymes that perform degradation reactions. So the lysosome is a big degrading organelle. It can take proteins down to their amino acid building blocks through the action of proteases. There are glycosidases that degrade complex glycans. There's lipases, phosphatases, sulfatases, RNases. Almost any biomolecule can potentially get degraded by the lysosome. And so this is how nature does it. So we came up with an idea for basically targeting this pathway with molecules that can hijack lysosomal trafficking receptors on the one hand, and on the other hand, grab a protein of interest that we would like to degrade. The lysosomal trafficking receptor that we started with is one that's well known in the community of glycoscience. In fact, when you study glycoscience or read a textbook, one of the very first chapters will be the discovery of the cation-independent mannose-6-phosphate receptor, or the M6PR. This is a receptor that is well known to bind extracellular glycoproteins whose glycans carry this mannose-6-phosphate mark. And that epitope is recognized by this receptor when it's on the plasma membrane, which then allows basically a capture of glycoproteins and then their internalization and trafficking from early endosomes all the way to lysosomes. The mannose-6-phosphate receptor also has this interesting part of its cycling biology where it traffics between the Golgi compartment and the early endosome and the late endosome. It is in fact the major trafficking mechanism that delivers lysosomal enzymes from the Golgi compartment to the lumen of the lysosome. And it's a well-known lysosomal trafficking system for lysosomal enzymes for enzyme replacement therapy. So there are a number of rare diseases in humans where people are missing certain lysosomal enzymes, and one can infuse into those patients recombinant forms of those enzymes glycosylated with mannose-6-phosphate. And then this receptor will just grab those recombinant proteins from the system and bring them into the lysosome. So we already knew that this was a beautiful system that the biotech industry had used to treat rare diseases with enzyme replacement therapies. And then the question was, could we hijack this system to traffic unwanted proteins to the lysosome for degradation? So what Stephen did is he designed bifunctional molecules, the lysosome targeting chimeras, that had on the one hand an element that binds to a target of interest. And so we use monoclonal antibodies for many of our LITACs. So that's why I'm showing this cartoon as it is. And then we chemically conjugated to those antibodies oligomers of mannose-6-phosphate. And these are the epitopes that will bind the mannose-6-phosphate receptor. And our finding early on is that in order to get good receptor binding and clustering and lysosomal trafficking, it was helpful for us to have clustered versions of mannose-6-phosphate presented in a multivalent way, which is why the cartoon is shown as it is. So if all is working well, one could either target a soluble protein that's out in circulation or a membrane-associated protein for degradation through this kind of molecule. And the vision was that the binder would engage the target, and then the mannose-6-phosphate groups would engage the receptor, which would then pull the assembly into ultimately the lysosome and deposit it there. And I should mention, it can internalize a cargo and then come back to the cell surface and grab another one. And the way it does this is through a pH switch. So the affinity of binding of mannose-6-phosphate to the receptor is high at pH 7 outside the cell, but there's a pH drop in the endosome lysosome pathway. So by the time this complex has reached the late endosome, the pH has dropped down to almost 5. And at that pH, there's a much lower affinity of mannose-6-phosphate for the receptor. So the cargo is released onto the lysosome, it goes, and the receptor then goes back and does another cycle of trafficking. So this idea, grabbing of the extracellular target and bringing it in, this was, not so different from enzyme replacement therapy biology. What was less clear is whether we could do this with a membrane target, because as far as we know, there was no natural biology by which the mannose-6-phosphate receptor could traffic another protein that's in the same membrane to the lysosome. So that we had to test experimentally with a big question mark hanging over our heads. So the way that we synthesized our first generation LITACs was to make glycopolypeptides that built from mannose-6-phosphate amino acid conjugates. And we had two different renditions of these glycopolypeptides. Our very first version were glycopolypeptides made by NCA, which is N-carbonic anhydride amino acid polymerization. It's a kind of polymer chemistry that leads to a sort of heterogeneous polymer that can then be conjugated onto an antibody. And when I say heterogeneous, we would make these polymers where the amino acid length, or the glycopolypeptide length, could be anywhere from 10 out to about 90 residues. And the polydispersities of these were in the 1.1 to 1.2 range, which is pretty tight, but there's still some heterogeneity. We had a moderate control over the length and composition, which was sort of limited by the polymerization chemistry, but they were heterogeneous, and that made them difficult to characterize. And also the chemistry was sort of difficult to scale up. So in the next generation, we switched over to solid phase peptide synthesis to make precisely defined glycopolypeptides, again, with a mannose-6-phosphate serine building block. And these LITACs we made with much shorter glycopolypeptides of much lower valency, either just dipeptides with 2 mannose-6-phosphate groups or pentapeptides with 5 mannose-6-phosphate groups. And these solid phase peptide synthesis products gave us full control over the length and composition. They were homogeneous, easily characterized, and much more amenable to scale up. So once we started making these LITACs, we immediately wanted to do some proof of concept experiments to see whether they could actually mediate the uptake and degradation of interesting cargo. So Steve did an experiment where he tried to model the uptake and degradation of immune complexes, which are pathogenic in many disease settings. And he did these experiments in a mouse model. So for example, what Steve did is he made a LITAC built from a secondary antibody that could bind to a cargo, which in this case was just a primary antibody carrying a fluorescent probe. And he compared the distribution and pharmacology of the complex where this antibody was a LITAC, it had the mannose-6-phosphate groups, versus the immune complex that was not a LITAC, where it was just a naked antibody bound to the cargo antibody. And so after IP injection into the animals, He then did blood draw, tissue extractions, and looked as a function of time at the levels of this fluorescent tagged cargo molecule that was in the system. And so what you're looking at here is these lanes of this fluorescent gel, which is basically reading out on this fluorescent cargo, shows the abundance of that cargo in either serum, liver, or spleen. And LITAC minus is with the naked antibody, and LITAC plus is with the mannose-6-phosphate conjugated antibody. And you can see after 24 hours post-injection, there's already clearance of the LITAC immune complex versus just the naked antibody immune complex. And after 48 hours, that LITAC immune complex is almost entirely depleted from the serum as well as these organs compared to the naked antibody complex. So that was an indication that having the mannose-6-phosphate groups on an antibody could allow the targeted clearance of a cargo from circulation. But what about a membrane protein? To test degradation of a membrane protein, we focused on one whose trafficking biology has been extensively studied and is also an interesting cancer target, and that is the epidermal growth factor receptor, or the EGFR. So EGFR is the target of a number of clinically approved drugs, and one of those is a monoclonal antibody called cetuximab. So we took that approved drug, did the chemistry with the mannose-6-phosphate glycopolypeptides, and converted the antibody into a LITAC, and then asked whether that LITAC could mediate internalization and degradation of EGFR. And what you can see from these fluorescence microscopy images is that the LITAC induces a dramatic redistribution of EGFR from the plasma membrane into the lysosomes compared to the naked antibody. So these are HeLa cells that are rich in EGFR expression. And we're labeling EGFR with a different orthogonal antibody that has a pink fluorescent dye on it. So you can see the pink is all over the membrane, and the blue here are just the nuclei stained with DAPI. And when we treat these HeLa cells with cetuximab, there's no change in the distribution or the abundance of EGFR, although the antibody is bound and it would block engagement with the ligand, EGF, were that ligand present. But when you look at the cetuximab-derived LITAC, you can see that that pink fluorescence is now almost entirely driven into these puncta, which co-stain with a lysosomal marker. So dramatic internalization by virtue of the LITAC. And EGFR is degraded upon its trafficking to the lysosome. So what you're looking at here are relative levels of EGFR in the HeLa cells that were treated with the naked cetuximab, those are the blue circles, or with the cetuximab LITAC, and those are the red circles. So you can see as a function of time, the LITAC basically drives degradation of EGFR and resets the steady state levels of that protein at a level about 80% reduced compared to the naked antibody treatment. And we can't drive EGFR levels all the way to zero because there's always some de novo biosynthesis, and we're only degrading the EGFR that was on the membrane. There's going to be some protein in the secretory pathway that's in transit and not yet on the membrane. But this dramatic reduction in the level of EGFR could be therapeutically relevant, not just for this protein, but other related membrane protein targets. We also started doing some in vivo studies. In a very recent experiment, Green On asked whether she could use a cetuximab LITAC made from one of these homogeneous pentapeptides with mannose-6-phosphates to degrade EGFR in a colon cancer xenograft model, which is the HCT116 cell line model. So what she did is she injected those cells into an immune-compromised mouse, let the tumors grow, and at certain points, she then administered the various cetuximab conjugates 2 subsequent days, and then harvested the tumors and quantitated the amount of EGFR present in those tumors by Western blotting. And as you can see, these are 2 groups of mice that were sort of biological replicates. Mice treated either with vehicle, PBS, or with the naked antibody, cetuximab, you see that there's really no change in the amount of EGFR that's in the tumors, normalized to total protein. But the cetuximab LITAC injected animals have a dramatic reduction in the total EGFR levels, and this was the case in both of these biological replicates. So the next question, of course, is does this level of reduction of EGFR have an effect on the tumor biology? And those are the experiments that Green is currently performing. But in parallel, we were interested in looking at other lysosomal trafficking receptors as vehicles for LITAC-mediated degradation. And the mannose-6-phosphate receptor was attractive because it has a nice, well-characterized ligand, that being mannose-6-phosphate. But it is expressed in every cell type. It's considered a housekeeping type of lysosomal trafficking receptor. And there are times in which one might want to have a more tissue-specific activity of a degrader molecule like a LITAC. And for that reason, we became interested in the liver-specific lysosomal trafficking receptor called the asialoglycoprotein receptor, or the ASGPR. And this audience has surely heard of this receptor. It's highly expressed in hepatocytes. There can be 1 million or so copies per cell. It has the professional job of clearing serum glycoproteins out of circulation through clathrin-mediated endocytosis. And the way it recognizes these serum glycoproteins is by virtue of those glycoproteins having terminal N-acetyl-galactosamine residues, or GalNAc residues, or galactose residues. And these are the two sugars that are able to bind, usually in a trimeric fashion, with ASGPR, which then induces its internalization, trafficking through the endosome-lysosome system. And just like the mannose-6-phosphate receptor, ASGPR has a pH switch. So it binds these GalNAc residues outside the cell with high affinity, but once the pH drops through the endosome pathway, the affinity is reduced and the cargo is released. Also, the ASGPR is well known in the delivery field because it's the main receptor targeted for liver accumulation of oligonucleotide therapeutics. So many of you will know that the liver is a great organ to target with siRNA therapeutics, mRNA therapeutics, antisense oligonucleotide therapeutics, and both Alnylam and Dicerna and Ionis are making brilliant use of the so-called tri-GalNAc motif. So here's an example of one from Alnylam. And what they do is they synthesize this little dendrimer and put GalNAc residues on the end, and then connect this covalently to their RNA therapeutic. And this allows very efficient uptake through ASGPR and accumulation in the lysosomes in hepatocytes. It's a great delivery strategy, and there are approved drugs that have this tri-GalNAc motif. So taking a page from that literature, what Green did is she synthesized a tri-GalNAc dendrimer, functionalized with a cyclooctine called DBCO. And this kind of brings me to a type of chemistry that is also a hallmark of our laboratory, which is bioorthogonal chemistries between azides and cyclooctines. It's a very nice way to click a functional group onto another biomolecule like a protein. And so what Green did is she took antibodies of interest, took a N-hydroxysuccinimide ester functionalized azidopeg spacer, modified the lysine residues of these antibodies to a small extent with the azidopegs, and then these azides would undergo the click chemistry with DBCO to attach the tri-GalNAc motifs. So in her very first GalNAc LITACs, Green was doing sort of heterogeneous chemistry, scattering the tri-GalNAc dendrimers around lysines on the surface of the antibody. These were heterogeneous LITACs because there's a lot more lysines than stoichiometry of modification. And her first pass LITACs actually had quite a bit of GalNAc dendrimer, around 11 per antibody, which was a lot of GalNAc. That was about 33 molecules of GalNAc, so 3 per dendrimer on each antibody. When she tested these GalNAc LITACs in cell culture, she found that they are pretty good at degrading membrane proteins. So her cetuximab GalNAc LITAC was able to degrade EGFR in HEP3B cells, HEPG2 cells, and HA7 cells. These are all hepatocyte-derived cell lines that express ASGPR. They also express the mannose-6-phosphate receptor. Again, that's pretty much a housekeeping receptor. And so she was able to compare head-to-head how the GalNAc LITACs compared to mannose-6-phosphate LITACs, both based on cetuximab. And as you can see, both of these LITACs gave similar degradation of EGFR in these cell lines. But when she tested these LITACs in vivo in a xenograft model that was based on a hepatocellular carcinoma cell line, HEPG2, she found that now the LITACs actually performed quite differently. So these were LITACs that she made from an antibody called pertuzumab, which is an approved cancer therapy that targets the antigen HER2. And so Green made LITACs from pertuzumab using mannose-6-phosphate or the tri-GalNAc motif. And she grew tumors of HEPG2 cells in the mouse. And then at different time points, she injected the pertuzumab conjugates, harvested the tumors, and then looked at the amount of HER2 by Western blot. And what she found, and you can see here, this is the HER2 blotting. Then as control protein, she looked at c-Met, which is also expressed in these tumors and is not bound by the HER2 antibody pertuzumab. And then she also looked at antibody levels, heavy chain and light chain. So the pertuzumab mannose-6-phosphate LITAC was a pretty good degrader of HER2 in this model. Protein levels went down about 80%. But the pertuzumab GalNAc LITACs were very ineffective in this model, even though they looked just as good in the cell culture degradation studies. So only a modest amount of degradation. And what we discovered is the reason that these GalNAc LITACs did not perform well in vivo was, I think, the obvious reason. They had so much GalNAc on them that they were just cleared by the liver very quickly from the serum before they had a chance to bind to the tumors and mediate degradation of the target HER2. So the GalNAc LITAC was not good in vivo because it simply had too much GalNAc on it, and its pharmacokinetics were really bad. So to solve that problem, Green switched to a different kind of chemistry, a chemistry that allows the site-specific conjugation of much lower numbers of GalNAc dendrimers onto the antibody surface. And we had a technology up and running in our lab for site-specific protein chemical modification. We call that the aldehyde tag method. I won't go through the details, but we can basically genetically engineer aldehydes at a position of interest. And then using some indole chemistry, we can conjugate those aldehydes with polyethylene glycol spacers with azide groups. And now when we click on the cyclooctine trigalNAc dendrimer, we know precisely where it will go, and there can be either one or two, but no more than that per protein. Also, Green made different isomers of these conjugates where she positioned the GalNAc dendrimers either near the C-terminus of the heavy chain, near the hinge region, or up in the CH1 region of the heavy chain. And these are three different geometries that we thought would be fun to test because, you know, there might be one that's more effective than another. And so first what Green did is she asked the question whether these lower valency LITACs had better pharmacokinetic properties, and they did. So without going through the details, and in the interest of time, I'll just say what we're looking at here is the amount of LITACs with different amounts of GalNAc in the plasma of mice after 6 hours post-injection, 24 hours, 48 hours, or 72 hours. And those original GalNAc LITACs that had 30-something GalNAcs on them, they were cleared out of the plasma in less than 6 hours. So no wonder they couldn't function in vivo as LITACs. But the lower valency site-specific LITACs, which had either 2 dendrimers of GalNAc or 1 dendrimer of GalNAc, those LITACs had plasma residence times that were very similar to the naked antibody cetuximab. So even out to 72 hours, there's still plenty of those lower valency site-specific LITACs circulating in the plasma, similar to the naked antibody, which of course has a long plasma residence time in the mouse. And correspondingly, these lower valency GalNAc LITACs were now much better at effecting degradation of their targets in the HepG2 xenograft model. So what Green did is she formed the tumors, these HepG2 tumors, and then injected these pertuzumab conjugates, 2 doses, 1 day apart, harvested the tumors, did Western blots, and you can see that HER2 levels are now down by more than 50% in those mice that had been treated with a low valency GalNAc LITAC, which happened to be positioned at the hinge region of pertuzumab. That was the most effective LITAC in her study. And once again, the high valency LITACs with too much GalNAc on pertuzumab were completely ineffective in vivo. So what this tells us is that to have an effective LITAC, it's not enough just to have a target binder and a receptor binder. You also have to think about what's the pharmacology of your drug, just as with any other type of modality. So with that, I'll just close by saying I hope that you find the LITAC technology as promising and interesting as we do. We have a lot of work ongoing in the lab. Of course, we're focusing on cell surface and secreted molecules for which antibodies don't block function, and you really need to degrade the target in order to get a biological effect. I should also point out that although I've talked entirely about proteins, there are other targets for LITACs as well, such as pathogenic glycans or lipids or RNAs. These are all biomolecules that can be degraded in the lysosome. And then finally, one should think beyond molecules because there's toxic particles, aggregates, right? Amyloid fibers, virus particles that one might want to target for degradation in the lysosome. So there's a lot of room in the future to explore on this technology. So with that, I'll thank all the people in my lab and especially Green and Stephen who pioneered this work. Thank you very much.
Thank you, Carolyn. That was beautiful talk and way to bring it home to the liver. We appreciate
Literally,
that.
yeah.
Yeah, right. There's some questions coming in that I'm going to read out. The first one is from Hao Zhu. How many more GalNAc ASGPR pairs are out there? Are there many more that are cell type or even liver zone specific?
This is a great question. There are other lysosomal trafficking receptors for sure that are known. There's about a dozen that have been described in humans. And just to give you a few other examples, there's the LDL receptor, right, which is important in our triglyceride and fat homeostasis, and that is very liver relevant. There is the folate receptor, which is constitutively expressed by macrophages, but also it comes up in a number of cancers, which is of course very therapeutically relevant. There are neuron-specific lysosomal trafficking receptors, and probably there are many other proteins that are really good at trafficking to the lysosome that haven't been reported as professional trafficking receptors. So we absolutely are interested in taking advantage of these other receptors based on their specific tissues of interest or therapeutic value. But I think it's also of interest to discover more fully the universe of lysosomal trafficking receptors. And one can think of interesting ways to do that at the proteome scale.
Great. I have a question myself, which is about timescales. So, you know, classically, when you learn the bioengineering time constants of all the cell processes, you learn that the endosomal recycling is sort of like order 20 minutes. And it seemed to me that like the biology in your system is playing out over hours, like, or maybe even a couple days. And I was wondering, like, what is it that?
What's up with that?
Yeah, is it depletion? Is it just takes a long time to clear, you know, the dose, or can you play with the time constant at all?
This is a really good question, and I would say we don't fully understand why. And I'll even go so far as to tell you that the time constant, for example, for EGFR degradation, is actually very dependent on the antibody that you use to build the LITAC. So we have a handful of EGFR antibodies. There's lots that have been reported, and they bind to different epitopes, and some have higher affinities than others, right? We have some that are in the nanomolar KDs, others with picomolar KDs. And the rate at which the protein levels is reset at a new normal by virtue of shifting the flux towards degradation is very dependent on the antibody. So we have some EGFR LITACs, like that cetuximab LITAC, which, you know, it takes like a day or more to reset the protein levels. But then we have another LITAC against EGFR in which those levels are reset in 5 or 6 hours. So I can't tell you that we understand this, honestly. We have a LITAC against a completely different target that drives protein down more than 90% in just like 1 to 2 hours. And so, you know, right now, these are empirical observations that are causing us to scratch our heads. So I think we have some basic research to do on recycling kinetics and degradation dynamics.
Great. Well, so for the liver biologists in the crowd, that'll be a good thing to study in some of their model systems.
Yeah.
Great.
Thank you.
Well, thank you so much for joining us remotely. It's a real treat.
Thanks.
Have a great day. So our final speaker of the session is also from Stanford, Roel Noos. He's the professor of developmental biology at Stanford and the Stanford School of Medicine. He's also an investigator of the Howard Hughes Medical Institute, and he's going to tell us about signals for cell proliferation in the liver. Welcome.
Thank you very much. Let me start screening, sharing the screen. Yeah, I would like to start by thank you for inviting me for this fantastic symposium. It's really enjoyable. I'm going to be the last speaker. I'm going to talk about a theme that we have been hearing about before during the symposium, and that is how cells in the liver proliferate. So before doing so, I want to remind the audience of a couple of general principles on the cell cycle and how cells divide in tissues in general. It is the case that if you look at any tissue, that the vast majority of cells do not divide. They are differentiated, they execute the function of the tissue, and it is only a small subset of cells, we can call them stem cells or progenitors, that can actually replicate. Those progenitor cells are typically controlled by growth factors that are in the vicinity of the progenitor cells, and when those growth factors are active, they induce cells to start to divide. So how does that work? Well, again, this is in general, it has been amply documented that in differentiated cells, the cell cycle is arrested. And it happens by the binding of the RB protein, the retinoblastoma protein, to a very important transcription factor called E2F1. And when RB is bound to E2F1, E2F1 is not active, and therefore the RB protein inhibits cells to enter S phase. Now, when a growth factor comes along, it will activate a cascade of protein kinases, including the cell cycle dependent protein kinase. Those will phosphorylate the RB protein, and that will lead to the inactivation of the RB protein. It cannot bind to E2F1 anymore. And then E2F1 is allowing the cells to enter S phase. So how does that work? Well, it has been found that E2F1 is actually a transcription factor that is responsible for the activation of a large set, a whole suite of genes that are all implicated in DNA synthesis, including DNA polymerase, pCNA, and in fact, many other genes. So that is what is happening at S phase. That is, of course, important in the liver as well, but I want to also point out that in the liver, but also in other tissues, that there is another set of members of the E2F family, and those are called E2F7 and E2F8. They are much less known than E2F1, unfortunately. It has been deduced from the sequence of the E2F7 and E2F8 genes, and also looking at where those genes are present in the evolutionary tree, that they may actually be the most ancient members of the E2F family. Compared to E2F1, which contains a DNA binding domain, but also other domains, E2F7 and E2F8 only contain DNA binding domains. Now, what do E2F7 and E2F8 do? They are implicated in arresting the cells during or actually prior to mitosis. So when they are active, cells cannot go through mitosis. So how does that happen? Well, they are repressors. And what they do is that they repress a large suite of genes, again, just like E2F1 can activate a suite of genes. But the suite of genes that are inactivated by E2F7 and 8, they include proteins and genes that are implicated in mitosis. And one of them is Aurora kinase, A and B. There's other proteins involved in checking the spindle formation, and also many other genes are the targets of E2F7 and E2F8. Now, what those genes doing in the liver is actually quite well known. They cause polyploidy. So when E2F7 and 8 are active in the liver, you get the formation of these polyploid hepatocytes. Everybody in the liver field knows that there's many hepatocytes that are polyploid, and over time, over age, you get an increase in the number of polyploid cells. That is not restricted to the liver. There's also other tissues where, in the presence of E2F7 and E2F8, cells become polyploid. And one good example are the so-called trophoblast giant cells that are operating in the placenta. So the loss of E2F7 and E2F8 in the liver, but also in the trophoblast giant cells, it actually leads to diploid cells. That's quite remarkable. You would actually think that a diploid state would be the normal state, but in the liver, this is somewhat abnormal and is caused by the loss of E2F7 and E2F8. Also in the trophoblast, you see the same emergence of diploid cells. It is also important to note in the liver that polyploidization uncouples DNA synthesis from cell division. So if you are looking at the classical markers of DNA synthesis, like EDU incorporation or the expression of Ki67, you will find that there are cells that are active with respect to S phase, but those cells may not go through mitosis because E2F7 and 8 are active in those cells. So you get not the division of one cell into two, but you get instead a larger cell that contains an amplified genome. So the question is then, in analogy with what is happening at S phase, do growth factors overcome the inhibition by E2F7 and E2F8? And this is now where our work on the Wnt pathway comes in. I'm going to display you now the Wnt cycling pathway. I always do that in two different states of a cell. Where there's an off state, this cell is not activated yet by a Wnt signal. There are two kinds of receptors for Wnt, LRP and frizzled. And then in the cytoplasm is a complex of various proteins, including beta-catenin. In that complex, beta-catenin will be phosphorylated by GSK3 and also with the 8F2 scaffold proteins, axin and APC. And that will destroy the beta-catenin protein. It's going to be targeted for the proteasome pathway. However, when a Wnt growth factor comes along, I should actually point out that Wnt is a protein that is modified by a lipid and is therefore very hydrophobic. It is not a growth factor that is traveling all over tissues, but it is acting very locally. And in most cases, we see actually the production of a Wnt by a cell that is adjacent to the one that is going to respond. It is a form of paracrine signaling. Now, the consequence of the Wnt protein binding to the receptors, LRP and frizzled, is that that complex in the cytoplasm of beta-catenin will fall apart. The beta-catenin protein is now going to the nucleus, and there it will bind to a DNA interacting protein called TCF. And the consequence of the beta-catenin activation of TCF is that there will be many target genes of Wnt being activated. The kind of target genes differs between different cell types, but there's some exceptions to the pleiotropy of Wnt readout, and those exceptions include genes that are actually also implicated in the Wnt pathway itself and in the liver, but also in other tissues. Axin 2 and LGR5 are direct targets of Wnt signaling. So those genes are very interesting and also useful to follow actually which cells in the tissue are activated by Wnt signaling. Now let's look at the liver and the well-known liver lobules and the activation of Wnt and also some of the Wnt targets. It is now widely documented that in the liver lobules, it is the central vein that is the Wnt signaling center. And you can follow that here by an in situ RNA hybridization experiment that we did some years ago, where you see that the endothelial cells of the central vein, so the blood vessel itself, is actually the source of the Wnt ligand. Then there are the Wnt target genes that become activated by Wnt coming from the endothelial cells, but now in the hepatocytes, you see the activation of the Wnt targets axin 2, but also another, and for my talk, very important Wnt target gene, and that is a gene called TBX3. It is a member of the T-box family, and by an antibody stain, you can see that the TBX3 gene and the protein is actually present in an area surrounding the central vein. It may be a gradient, but for the time being, we don't know. Of course, it is known that Wnt can activate many other genes, and is in fact the primary driver of the hepatic zonation, and that many of the enzymes that we heard before during this meeting are actually direct Wnt target genes. So let's talk about TBX3 a little bit more. It is known to be a repressor of gene expression. It was also known prior to our work that TBX3 and also beta-catenin are required for liver growth during the mouse embryogenesis and also during postnatal stages. If you inactivate TBX3 or beta-catenin, there's a rudimentary growth of the liver. It is also known that beta-catenin and axin and TBX3 and other genes are implicated in liver cancer, all indicating that Wnt is indeed a driver of proliferation of hepatocytes, but how does it actually work? So we focused, after finding that TBX3 was a target of Wnt in the liver, on inactivating the TBX3 gene during postnatal growth of the liver, when the liver rapidly expands. And these are experiments that were done by Una Jin and Tenny Anbarkian in the lab. And the system that we used was to take a gene that is also activated just like TBX3, and that is AXIN2, the AXIN2 promoter driving RTTA. I want to point out at this moment that there is quite some controversy about the function of AXIN2 as a gene in the liver. I'm aware of those controversies, but in this case, we are using the AXIN2 promoter driving RTTA. So this is not a situation where the AXIN2 gene has been inactivated. This is in fact a transgene. What RTTA is now going to do is to activate the TETON CRE transgene, also present in these mice, and also as a marker of the cells where this is all going to happen, HBGFP. The CRE is now going to be able to take the fluxed form of the TBX3, also present in these mice that we have been putting together by genetic crosses, and eliminate it from the genome. So what is the consequence now of inactivating TBX3 in the zone where it is normally expressed? You can see now that, and this is again during postnatal stages of development, that instead of many cells being labeled, that in the absence of TBX3, fewer cells are labeled with HBGFP, and that the nuclei of these cells are remarkably larger. And if you do a DNA ploidy analysis on the TBX3 wild type and TBX3 mutant livers, you see that in the mutant livers that there is an increase in the number of 4N nuclei and also the higher ploidy numbers. And it indicates, together with a large set of other experiments that we have been doing in the lab, that cells do not go through mitosis without TBX3. So what are now the targets of TBX3? And Una Jin in the lab did a chromatin IP experiment, finding that TBX3 has many targets in hepatocytes, but including in those targets were these two genes that I mentioned before, E2F7 and E2F8, came out of the chromatin IP and also by direct chip qPCR experiments. And as you might expect, TBX3 being a repressor, Una found that TBX3 overexpression can lower the expression of its target E2F7 and also E2F8. And if you look at liver tissue by in situ RNA hybridization, at wild type tissue or at mutant tissue, you see that when TBX3 is absent from the liver, that there is an ectopic expression of E2F8 RNA. And together with yet other now genetic epistasis experiments, we found that TBX3 is indeed a negative regulator of E2F7 and E2F8. And that leads to the conclusion that TBX3 is a direct repressor of E2F7 and E2F8. And that led us also now to conclude that Wnt growth factors are not working on S phase, and we have also some cell culture data supporting that view, but that Wnt through TBX3 is actually working as an inactivator of the repressors that are leading to mitosis. So the players here at mitosis are different than what is happening at S phase, but in general, you see that there's a principle involved here of double or sequential repression, where again, when E2F7 and 8 are active, you don't go through mitosis, they repress the whole machinery there, the cell becomes polyploid. But when TBX3, after being activated by Wnt, starts to inactivate E2F7 and 8, cells are allowed to go through mitosis. And you get fewer polyploid cells. That brings up, of course, the question, what is now happening at S phase and how is S phase induced in the liver. And I'm now going to tell you about the experiments that were done by Abby Sarkar in the lab, who was interested in studying the consequences of fluctuation in the diet in the liver. The liver is, of course, principally involved in nutrient processing. We heard many talks addressing that point today. We also know that in the laboratory animals, the adult liver is mostly quiescent. That is related to what I'm going to tell you. But Abby also figured that in nature, food availability is actually fluctuating, very different from what is happening in the lab. Animals in the wild, they have to survive periods of starvation, and laboratory animals, they live under unusual dietary conditions. And what Abby was doing is to develop a protocol where adult mice were held in the lab under, let's say, the standard condition of ad-libiting food availability, but comparing that to what is happening after intermittent fasting, where mice were held without food for 24 hours, then the normal feeding, and then alternating a couple of days in a row. So the consequences of intermittent fasting on the replication of cells in the liver lobules were quite remarkable. We're now going to use, again, the axon 2 promoter driving RTTA. In the presence of dogs, we're going to label these axon 2 expressing cells in the adult liver, and now comparing that to a mouse in which intermittent fasting was applied. And you see that instead of just a ring of cells being labeled, that there is a marked increase in the overall number of labeled cells, indicating that these cells have been starting to divide as the consequence of intermittent fasting. So how does that work? Well, it has been documented quite amply that under conditions of intermittent fasting, there's the production of an endocrine hormone, a member of the FGF family, called FGF15. At least in the mouse it is FGF15, in humans it's called FGF19. It is made in the intestine and it responds to the feeding status. Now in the liver, there's a specific receptor for FGF15, and that is beta-klotho. It is actually a co-receptor for these endocrine FGFs, together with the FGF receptor, the tyrosine kinase. So again, in the liver, you get in the intestine the production of FGF15, and in the liver, that can now start to activate a tyrosine kinase pathway together with the beta-klotho co-receptor. So we obtained conditional beta-klotho knockout animals and crossed those into our labeling system. And now looking again at the consequence of intermittent fasting in mice that were wild type or heterozygous for beta-klotho versus mice that were conditionally mutant for beta-klotho. And you can see indeed in the absence of beta-klotho, so the receptor for FGF15, that expansion of the number of labeled cells is slowed down. A converse experiment that Abby did was to overexpress FGF15 by an AAV vector, and again labeling the cells, and you see that in a situation of excess FGF15 that there's an increase in the number of labeled cells, again indicating proliferation of the cells. So we came to the conclusion that what is happening at S phase is indeed a classical tyrosine kinase activating pathway, presumably leading through the activation of cyclin-dependent kinase, and then activating E2F1, and that what Wnt is doing through TBX3 is to regulate mitosis, so a separate phase of the cell cycle. Is the TBX3 also necessary for the replication of these cells after intermittent fasting? So this was an experiment similar to one that I showed before, where we are conditionally knocking out TBX3 in the liver tissue, again using the RTTA system, subjecting these mice to intermittent fasting, finding that in the absence of CRE, TBX3 is still active, expansion of the labeled cells, but when TBX3 is mutant, there's a much lower number of labeled cells, indicating again that the cells do not divide Also note that the nuclei of these cells now are larger, and indeed by direct measurements, Abby found that there's an increased DNA content of the cells. So lots of questions that are coming up. How general is this? Is the Wnt-TBX axis conserved? Does it play out in other tissues than the liver? It is known that TBX3 and other T-box genes, and I'll come back to that in a minute, are often expressed in a growth zone in tissues together with Wnt. It should also be noted that E2F7 and 8, they're widely expressed in tissues, and they can lead to mitotic arrest. I mentioned the trophoblast giant cells, but there is in fact many other tissues where these genes are holding cells in a state of not going through mitosis. The fact that the T-box genes may be implicated in Wnt sickling and mitosis as well is something that we find of great interest. I'm going to close my talk now, not talking about the liver anymore, but about what is happening in animals in general, and going back to the evolutionary emergence of multicellularity. And first pointing out that TBX3 is a member of these T-box genes, and the most well-known of that family is a gene called brachyury, sometimes simply called T. And just like TBX3, it has a transcriptional repressing domain. And to illustrate what T-box brachyury is doing together with Wnt, I'm going to tell you about an evolutionary tree where unicellular organisms, they can divide individually, everybody can divide, but as soon as the metazoan kingdom arose in the course of evolution, and then branching out into the sponges, the periphery, cnidaria, and the chordates, so that's us. In multicellular animals, you see that there are now unequal rates of cell division. Cell division is now going to be controlled. What has also been found is that wherever you look in the branches of the animal kingdom, in the metazoan, that early in development, early in embryogenesis, what is happening is that there is just a ball of cells, but then there is what is going to be called a local growth zone, where a small group of cells start to divide, or divide at a higher rate than the other cells. And it actually leads to an elongation of the ball into an axis of asymmetry in the developing embryo, and the emergence of an anterior and a posterior domain in the AP axis. The posterior end will, of course, form the tail. The anterior end is going to form the head. So it has been established and reviewed quite amply by evolutionary biologists, is that if you start to look at the emergence of the Wnt gene family in the course of evolution, is that unicellular organisms, they don't have any Wnt, but all of the branches of the metazoan kingdom, the ones that are that have an axis of asymmetry in particular, they do have Wnt genes, and the Wnt's are going to be expressed at what is going to form the posterior domain of the developing embryo. And the Wnt's are indeed the signals that are setting up these local growth zones. They are primarily involved in setting up the anterior posterior axis. That is illustrated here in the cnidaria. So these are sea anemones and hydra as well. You see a very local domain of Wnt expression. And similarly in the chordates, at the tail bud, you see a high level of Wnt expressed in the tail bud. And that is where the embryo is elongating due to cell proliferation. So the posterior growth zone is the consequence of local activity of Wnt sickling. Now, it is not only Wnt that is active there. That member of the T-box family, brachyury, is expressed in a very similar domain as Wnt itself, in the cnidaria, but also in the chordates. And it has been found that the brachyury, whether it is in cnidaria or in the chordates, is indeed a direct target of Wnt sickling. That the brachyury gene has binding sites for the TCF transcription factors in its promoter, and that this whole axis of Wnt going through a T-box family member, whether it is brachyury or TBX3, is actually highly conserved in evolution and is actually responsible for the proliferation of cells in many different contexts. And with that comment, I want to now acknowledge the people in the lab who've been doing this work. I mentioned the work done by Abby Sarkar on intermittent fasting, and Tenny Ambarkian and Una Jin on the interaction between TBX3 and E2F7 and 8. And this is our lab during the times of COVID, during an outdoor lab party. Thank you for your attention. Happy to answer questions.
Roel, thank you so much. Wynn is clapping on the screen. That was fantastic. We have a question in the chat about the intermittent fasting result. If you speculate, why would liver cells proliferate after fasting? Is there concurrent loss of cells elsewhere in the liver that the proliferation is offsetting?
Yeah, so what is happening after fasting is that the liver cells, not surprisingly, they become smaller. And we have some evidence that there is senescence going on in those cells. And that what is happening as the consequence of the proliferation is that the total mass of the liver is being compensated by proliferating hepatocytes.
Makes sense. Another question from Kristen Knaus. Given the Wnt gradient along the portovenous axis, do you observe an expected gradient of hepatocyte ploidy along this axis?
Yeah, that's actually Shalev mentioned that as well. So in the middle of the lobule, there's indeed a higher number of polyploid cells. So there's indeed a gradient of polyploidy in the liver lobules. So it's indeed asymmetric in the lobule.
Great. Well, there's many more questions, but I think in the interest of time, we'll thank you again and close out this session. We really are fortunate that you were able to join us remotely for the day. So thank you very much. And Sherry, I'm going to turn it over to you for some closing comments.
Great, thanks, Sangeeta. So on behalf of the American Liver Foundation, I can't thank you all enough for the amazing day we've had together. We had more than 370 participants attending the symposium today, and we are pleased that the virtual platform allowed so many global participants. Thank you to our presenters. Your research is amazing, and we so appreciate your willingness to share your work in this year's conference, along with some very challenging time zones for several of you. I also want to thank our gold sponsors, ASI and Salix Pharmaceuticals, bronze sponsors, CVS Pharmacy, and our presenting sponsors, Intellia Therapeutics and Exact Sciences. We so appreciate your ongoing support of the ALF mission. Thank you to Sangeeta, Wolfram, and especially Heather Fleming for their leadership throughout this very challenging event. Your focus on inviting the most valuable presenters, coupled with Heather's ongoing attention to detail, has really helped us present one of the best symposiums I've attended. Congratulations to Dr. Arias. It has been one of the greatest pleasures to work with you throughout the years, and the American Liver Foundation is so appreciative of your enthusiastic support for our mission and your shared passion to bridge basic science and liver disease. Finally, I'd like to thank Erica from the American Liver Foundation. Her precise attention to detail has allowed us to move through today's virtual presentation, and I'm so appreciative of her help in executing this successful day. Thank you all for attending today. Have a happy, healthy, and safe holiday season, and we look forward to seeing you in 2022 at the 31st Annual Irwin M. Arias Symposium. Thank you.
So also from Sangeeta and myself, I wanted to thank first and foremost Wynn. You have been truly an inspiration for not just us, but really generations of hepatologists, scientists, trainees, and it has been such a privilege to have you in our midst. Thank you both Sherry and Erica and the entire ALF team for not just the support here and in organizing the symposium, but really for what you do globally for patients and patients with liver disease in raising funds and raising awareness and giving advice I know personally from the patients in my clinic, the physicians in my division, what that work really means. And it's just incredible. I really wanted to thank Heather. I personally know how much work you have done versus the work that I have done. And in the process, I think you really not just shaped the symposium and the style, the outcome that we've had today, but really made me learn a lot about how to invite and engage trainees and how to really create a tremendous experience. To be fair, I think I can speak for many here. After 20 months, we are kind of zoomed out. And I was a little worried about this format and the day, and to know and to see how many people have stuck it out with us for the entire day really speaks to the selection of speakers and really the excellence of your science. And so I want to really thank everyone here who contributed as speakers, as moderators on the short science talks, and in the audience through your questions to make this an interactive and truly outstanding experience. And it happens for those of you who are in Boston. I think this must have been one of the nicest fall days ever. And we all stayed inside more or less to watch this and be participants. And that was, it was really a great reward. As Sherry said, we look forward to hopefully have this in person in Cambridge in 2022. But I have to say, given this positive experience to really build a global community, hopefully we can build it on a hybrid format that will allow us to engage with people across continents. And it would really be great to get your feedback here in the audience, what you all think about that. Because really, while many of us think that Kendall Square is the epicenter of bio and biotech and biotechnology, there's so much more we can learn from each other across the globe. And so we look forward to your formative comments and feedback there. And with that, Sangeeta, unless you have the final word, I really want to say thank you and hope to see you all well and healthy next fall.